Okay, thank you and good morning. Good morning to everyone that is in attendance this morning here at Bolivia Lodge. And also not forgetting, if ever there are those that might be watching us from the University of Limpopo YouTube channel, we also say good morning to them. We are on day 3 of the Faculty of Science and Agriculture Postgraduate Research Day. And this is where today I consider this day a very special day, and you will hear my reasons for that. First, it's because today we are going to be wrapping up the presentation sessions. And on the other hand, we are going to be listening to our very last keynote speaker. And this is somebody who will be presenting to us from a place which has a different time zone from where we are. And I think already with that being mentioned, as the faculty, we can already see that somebody had to make a very huge sacrifice to be able to be with us this morning, whereas from where he is, it's probably midnight. So thank you very much. So ladies and gentlemen, let me say on behalf of the Faculty of Science and Agriculture, for this moment, I would like to extend my warmest visual welcome to Dr. Darren Fast. Dr. Darren Fast is joining us from the province of Alberta in Canada. Dr. Fast, we would like to say to you that the Faculty of Science and Agriculture really does not take lightly the fact that you are here with us this morning. And if I'm not mistaken, probably at your place, it's midnight. So we really thank you for that, and the faculty appreciates it. So, ladies and gentlemen, today we are going to continue along the same theme, which goes advancing science through research, from lab to market, integrating science, innovation, entrepreneurship in university research. So without any waste of time, I am going to read out Dr. Darren Fast's short bio. Dr. Darren Fast is an Associate Vice President in Innovation, Knowledge Mobilization and Partnerships at the University of Alberta in Canada. His role is to develop and support a clear innovation strategy for the University of Alberta, utilizing and coordinating existing resources across campus and the community. This involves supporting entrepreneurs and innovators. Dr. Fast also works on ensuring that research of all types has a clear path to add value to and have impact in the community through enabling knowledge mobilization and partnerships with industry and community organizations. Dr. Fast serves as a liaison to funding agencies, helping to build provincial, national, and international research collaborations, as well as community research partnerships. So Dr. Fast's presentation title is Recognizing and Developing Innovation Arising from Research. So ladies and gentlemen, Dr. Fast will now have the platform to share his knowledge and expertise. And could you please join me and give him a big round of applause? Thank you. Over to you, Dr. Fast.
Perfect. Thank you so much for having me here. And yes, it is just after midnight, so but happy to be here. So Just for context, a little bit of where I am. You can see Canada there on the upper left of the map, and on the inset, you can see the province of Alberta in red, and then Edmonton, where I'm located, right pretty much in the middle of that province. So it is a very long way from you, but and just again, this is a picture. It's not this current year, but it is looking very much like this because it is fall time, and this is just a picture of our city. So just a little bit of background about myself. My career started with a PhD in biochemistry, where I was looking at lipoproteins, assembly and secretion. I then did a short postdoc, but then very quickly moved to the industry side of the life sciences and started doing work at a small biotech company doing early stage new product development, which was a lot of project management, a lot of regulatory affairs, significant amount of business development, patenting, clinical trials, and that sort of thing. I did that for about 5 years, then was fortunate to move to a venture capital firm where I became the chief technology officer, tasked with building new companies out of early stage research coming out of universities. Did that for a number of years and then became an independent consultant and got to work in many areas outside of the life sciences and really pretty much around the world, helping organizations, companies, researchers bring their new products to the market. I've also been an entrepreneur and had my own company working in the health space. And then more recently, I've been involved for the last 10 years or so in technology transfer, previously at the University of Manitoba, and then about 2.5 years ago, I joined the University of Alberta, leading their innovation efforts. And so happy to be here and share some of my experience and perspective on this. But perhaps maybe just to start, we'll start with the definition of innovation, just making sure that we're all on the same page. So the way I'd like to describe innovation is doing something new that creates value. And new is one of those words that can be tricky, but it could be entirely new, but it could also be new to the industry or sector that you're talking about, or it could be new to the organization. So it doesn't have to be entirely new. And sometimes just a part of it is new. And what do we mean by value? Value, of course, means different things to different people. And so in many cases, we think about how do we improve the quality of life? It could be things like environmental benefit, not just dollars or economic growth, which is the most common way that people view value. But I think it is important to recognize that there are many, many forms of value, and they are all valuable. So why is innovation important? I think this is a question that's worth asking. We've obviously have many challenges in our world. We've had many challenges for a long time. Those things don't seem to be going away. And there are many things that are seemingly random and unpredictable, which might be considered black swan type events. Those continue to happen. Another important piece is that, if you think about companies, there are relatively few that have been around for a very long time, and virtually none of those are doing what they set out when they first started. So there's this constant change that is there. Obviously, working at a university, we see technology advancing rapidly, and our workplaces are dynamic, right? During COVID, many of us worked from home. Now we are doing some sort of hybrid type work. And then as customers and purchasers of products, our expectations continue to change. So we are not purchasing the same types of things or wanting even the same types of things that people may have wanted 20 or 50 years ago. So there's a constant change that's happening with expectations as well. So part of the response to that is innovation. And just historically, not that many years ago, innovation was often seen as a very negative thing. Even being associated with political disruption and subversion, which is a pretty harsh view on innovation. But in about 1911, this idea of a creative destruction. So we destroy something in order to create something else. Started thinking about innovation as a positive feature, something that would help us to grow. And in the 20th century, innovation or things were largely focused on invention, not necessarily innovation as a core business strategy that it is today. But there was lots of research and lots of exploration, but it wasn't as much of a core strategy as it is today. And I think that's important to remember as well. And I've used this illustration in a number of talks that I've given, and I think it's really valuable to recognize how innovation is quite lumpy. So, for as long as historians can picture, we have moved things from one place to another. People have traveled, and they brought their belongings with them. And then, at some point, people created boxes where they could put their things in, and moved those around. Generally, they had people put these boxes on carts, and they moved them from place to place. And in about 1870, someone decided that they should put a handle on one of these boxes, and it became the suitcase that we all know and love today. Major innovation, putting the handle on that suitcase, because it allowed people to carry their own luggage without needing someone else. And interestingly, it took about 100 years after that before somebody decided to put wheels on the suitcase so that they didn't actually have to carry it, but could drag it behind them. And I'm actually old enough to remember these suitcases, and those wheels were about 1 centimeter in diameter. And as soon as you rolled over a crack in the sidewalk, it would pretty much tip over. And you think, wow, why did you just not put bigger wheels on it and maybe twist it sideways? But that took another 17 years before somebody came up with what today seems like a very obvious improvement on the suitcase. So I think it goes to show that we can't always see innovations, even though they may be incredibly obvious in hindsight. And people often think innovation is a bit of a panacea. It's easy to make money. It's something that we can just turn on and make happen. And then the other thing that people sometimes look for is they want the recipe for how to do innovation. And I think one of the perspectives that I've had is that it's really difficult to say that there is only one way to succeed, because if you're doing something innovative, you're doing something new, which goes against the idea of repeating it. Although that's not to say that there's not a lot of process that can't be applied to innovation. Another thing to sort of think about as we're talking about how we move things out of a university into public use and acceptance, there's this idea of this technology readiness level, and hopefully people are generally familiar with this. On the left is really basic research, trying to understand the principles and the concept. On the right is, the far right is when you actually have something that is on the market. So universities typically play in this TRL levels 1 to 4. Industry typically plays in the 6 to 9 levels, and there's a gap in the middle, which is difficult to fulfill and difficult to move technologies through that. It is difficult to get funding. It is difficult to attract experienced management to these companies that are moving these products forward. And so, in many cases, it's called the valley of death, and lots of technologies do not make it through this gap, and it is definitely a challenge to do that. So how do we, as people in universities who have these good ideas and have these potential solutions which could benefit humanity, how do we move them through this and into industry where they can be commercialized, scaled, and brought to people who can use them. And this is a real challenge, because scaling these technologies is often as difficult, if not more difficult in some cases, than actually coming up with the invention. It's very clear to me that this is not a solo sport. This is something that requires many players in an ecosystem to really be successful. And if you look at this chart, you can see that universities and educational institutions, yes, we're research organizations. Yes, we do a lot of work with workforce development. We may be those tech developers or create these startups. We may have some connections into the community, but there are many other players who all play in this space. And MIT has a really nice model that actually has 5 key stakeholders, which overlap a little bit with this. But there's definitely many players who need to do this, and it's not clear that any one group owns or manages this innovation ecosystem, which needs to be there in order to help create economic value and impact for stuff coming out of research. So we need all of these players to coordinate, to work together. And so I think that's a really important part to keep in mind, is how do we, as universities, make sure we are properly connected into things like investors and into industry who can perhaps help us identify some of the market needs that might be out there. And then I just want to talk a little bit about sort of different ways of moving things out of these early stages into the commercial world. There's this idea of technology push, which is really starting with an idea or starting with something coming out of research. We found something, we found a potential advance of some sort, and regardless of which field you're in, and now we are trying to find how we could apply that advance to better humanity in some way. A lot of university research, especially basic research, is really coming out of this particular lens. And there's lots of great examples of things that have come out of this. And I'll just pick on lasers as an example. It started with very basic research, and now lasers are used in an absolute multitude of industries and systems where we see them everywhere. But they really started with as a science project, and then they found a use for those lasers. The other way you can think about how things move is really this market pull. So starting at the other end, realizing that, wow, we have a problem, and we need to solve that problem. Some university research comes at things this way, not always. So it's easier to see what the problem is and what we're trying to solve for, and what would solve that. But it doesn't mean it's easier. It's just different. So there are just two ways of how we move things out of a lab, let's say, into that commercial space. One of my favorite examples, just because one of the things I do like to do is some woodwork, is this example of the SawStop. This is a brilliant invention because these folks realized, kids in shop classes were cutting their fingers off when using saws because they really weren't experienced enough to do it well. And so they set out to make a safer saw. Basic premise behind this thing is that there's a sensor that is wired to the blade, measures the impedance. If you put some soft tissue into the blade, it senses that, this is probably a finger as opposed to a piece of lumber. And then there is effectively a shotgun type shell that shoots the blade below the surface of the table, so people end up with a scratch rather than a missing finger. It's really quite brilliant. And it has really saved, especially in the US, where people are quite litigious, lots of lawsuits, and made the schools there who use these saws much, much safer. This is again just an example of how you can pull things, starting with a market perspective as opposed to starting with some basic science. And then this idea of creative destruction that I mentioned a little bit earlier. So again, there's the two parts, right? There's the death or destruction of something that's outdated, some industries. We're seeing this perhaps today in the use of AI and computer programming, where lots of current programmers are looking around and going like, wow, you don't need as many of us coding things because the AI can do some of that. And what that leads to a new industry, that whole AI industry has really started to take off. And the idea is that, people can be recycled into these new jobs. They just may need some training. Of course, this is never as simple as we describe here, but this is something that you can see over time. I think it's always important to ask ourselves, what makes a good invention? Coming out of research, what makes it good? What should we be looking for in terms of a new invention? So it's obviously got to be technically better. It's got to be cheaper. It's got to have some new functionality. It's got to have some competitive advantage in order to displace whatever might be out there today. In an ideal world, it should be able to be protected to give whoever's moving it forward some sort of competitive advantage to justify the cost of moving that development or moving that product through the development cycle, scaling it, building whatever plants need to be created to make it. So there should be some form of protection that's available to it. It's also got to have some feasible development. I'll talk in a little while about an example of some things that I've seen that were not feasible and how that basically killed them. Again, it has to have a large or significant return or profit or benefit if it's going to, if somebody is going to take the risk of moving it forward. And it really, especially stuff coming out of universities, really requires, old example for me, but I was involved with a company that was moving this compound which looks like the molecule there. And I understand many of you are chemists, so you'll see that there's a number of chiral centers in there. This had the ability, at least in cell culture, to inhibit certain kind of cancers. But the organization I was with was trying to develop this and see if it actually made sense to treat humans with it. So we could buy it, or it was harvested from a sea sponge in the South Pacific that was quite deep in the ocean, and needed specialized diving equipment to go harvest it. And there was obviously a limited amount of this that you could find. So you could buy microgram quantities of this. So we did find a chemist who had synthesized it, and you know, it was rather expensive. I think we spent close to $500,000 making about 5 milligrams, which was probably the world supply of this compound at the time, which, you know, we very quickly realized was not a commercially viable product. We thought maybe we could, you know, ultimately bring that cost down to something more reasonable. But, you know, even at 5 milligrams, we didn't have enough to treat a single mouse, much less a human. And so this company ultimately failed because it was just too technically complicated to make it. And so it remained a research project. Another example, you know, was one of these dental implant stability testing devices. So, you know, if you're aware of dental implants, what they do is they put a post in your jaw, they allow the bone to grow around that post, and then they put the crown or the tooth onto that post, and you then have a replacement tooth. It takes about 4 weeks for the bone to grow around that new post before it is stable enough to actually put a tooth on and allow you to chew and things like that. You know, a researcher had spent many years developing a prototype. He'd built a very nice device to assess this, and it basically required somebody going in periodically with this device, which would ping the tooth. A waveform was created, and by measuring that waveform over time, you could determine when the tooth was, or when the implant was stable. And he was very excited. You know, he had engaged a venture capital firm who engaged me to do some market research, and I talked to about 25 perio-orthodontists, who are the folks who do this type of work, and it was a unanimous not interested, because they would have required a special chair in their office and another staff member to assess this, and you know, of course, asked, well, like, why would you not use this? And they're like, well, of course we don't need to, because we know that the bone grows around that post in about 4 weeks, so we ask people to come back in 6 weeks. And I'm like, okay, well, that's great, but how do you know it's ready? And every single one of them said, well, we reach into their mouths and we wiggle the post. If their jaw moves along with the post, we know it's stable. If the post moves and their jaw doesn't, it's not stable, it's not ready. We tell them to come back in another month. We don't need a device to tell us what to do. So the market really dictated what they needed to do. And so this was something that again did not proceed further. So there's this idea of product market fit. And you know, it's a very simple Venn diagram to illustrate the point, but you can see that you know, the better the specifications of the product fit the market need, what people will actually pay for, the better chance of success. And in the two examples I gave just now, you know, there was not a good product market fit, and therefore that failed. So it is really important early on in this commercial process, one of the first things that we tend to do, even before we will, file patents or anything like that, is start to answer this question. This is something that gets, continually iterated as we continue to develop this forward and look for the right partners to move it forward. But this is the number one reason that startup companies fail. So it's really important to make sure that we do have good product market fit. And then, this is just basically another way of looking at this whole thing. All of the features that you see in a product, at the end of the day, people don't really care about which specs are in there. If it's a device, I mean, think about your cell phone. You don't really care which particular chip is in there or which patents are filed by which companies. All you care is that there is a bundle of benefits. It does what you need it to do, therefore creating value for you as a customer. And that is really what we are trying to get to with everything that we do. We must have something that provides value to the end user, because or else it is just really difficult to justify the cost of building it and scaling it to get it in the hands of people. And so, we file a lot of patents at our university. It's a pretty important group in our tech transfer team. But when we look at it, it is not about can we file a patent. It's should we file a patent, and how will that protect the business opportunity that exists? It is not about the technology itself. It is really about what is the business opportunity that we are trying to protect. That is the question that we are asking when we file patents. And I think that's a really important one, because it is easy, relatively easy, to get a patent. It is difficult to get a patent that creates value and actually ends up paying for itself. Another thing that I think that we always look at in our university in terms of trying to find some metrics. What should we expect? And looking at Autumn Stats, it's a group that thinks about technology transfer a lot. It looks like most universities should get one invention disclosure for about every $4.5 million of research funding. These are US dollars. It varies. Obviously awareness of faculty around the commercialization process is different. Different types of research tend to either make patenting more or less attractive. And we also notice that many universities, and the University of Alberta is one of these, where our faculty evaluations, the things that sort of move people along their career development pathway, don't necessarily reward inventions or commercial activity. And so we are, in some cases, at odds with what researchers need to move their careers forward with trying to capture value out of research. So it's an important thing to remember. And we've been working at trying to help our faculties and their evaluation councils recognize that this is truly valuable for them as a department or as a faculty, to have these impacts that are made by moving things out of the university. And again, similarly on startups, on average, it takes about $75 million of research funding in order to create a startup. And so, just as a benchmark, and again, the type of research matters. And because you're now, I would argue at most institutions, other than some of the very largest institutions, these are lumpy numbers. So it's difficult to set a target and get them accurate, because one year there might be 2 or 3, and then the next year there might be none. So, we have really moved to looking at rolling averages as opposed to year-to-year comparisons, because the numbers are so small that it's really difficult to compare within a year. So how do we create more impact? I think is one of the key questions that we need to ask ourselves. Obviously awareness of the importance of commercializing research. We spend a lot of time with graduate students, who might be looking for new careers and alternative careers other than becoming faculty, for which there are not nearly enough positions. We provide a lot of support. We have a startup support specialist who is there to help direct people to the correct resources. We have business incubators that can help. We do a lot of training in terms of teaching, and in many cases it's teaching graduate students how to do some of that market assessment prior to them starting a company, so that we can really help create companies that have more value and are more attractive to venture capital or any other sorts of funding, or even attracting good staff into those companies. We are also working on some venture studio models, which is really, so in addition to the push of the normal technology transfer that comes out of universities, pulling those market ideas and looking for technologies that can be used to support those market challenges that we see. And then, of course, we do a lot of work with businesses who can use some of the technologies and expertise that we have inside the university to really build those partnerships and, again, create more pull from the university into the community to move these things forward. So just for a quick reference, the Venture Studio is really, again, starting with that market research, identifying where there's an opportunity, and then trying to find the right solutions for that from things that already exist, perhaps, in your university or in your community, and pulling that out. It's a very hands-on type involvement. It's, there's usually a fund that provides that team, those resources, the support to make that happen. And it's something that can be systematized quite easily to really pull things out of the university. And, of course, it makes them far more attractive as venture-based companies to really help them grow quickly. So, if we look at what we do, and just starting from the top here, we get research results and we triage them, and we basically put them into one of 4 buckets. We either can create a spin-off, a new company out of that research. We might do a research collaboration with a company to sort of shift some of that knowledge and expertise into the private sector. We may license that technology if it makes sense, if it's something that we could patent. And in many cases, we'll rework the technology. And, I would say probably for every 100 technologies that we get disclosed to us, somewhere around 5 are spin-offs, somewhere around 10 to 15 are license opportunities. Most of them end up not proceeding. And then there's a few that are also good collaboration opportunities for companies that exist. So we have an inventor-owned intellectual property policy here. I have worked at institutions that do not. They have a joint IP ownership policy, but I think, it's really clear that one of the benefits that is important, that helps move this along, is having clear pathways for people to recognize how to jump into that space, which is probably foreign to them, and really help them move forward in as low pain process as possible, because this is new for a lot of people. So I thought, just as I wrap up, I will walk through sort of what our innovation strategy looks like at the University of Alberta, just to give you some ideas. And this is obviously not the path and not the process for everyone, but if there's something you can use here, I think it may be informative. So one of the things in our university's strategic plan is to create impact. And what does impact look like? Well, it looks like jobs for our graduates. It looks like economic development. It looks like philanthropy, so that people want to donate to the university. It looks like reputation. And also, it may look like royalties that come back to the institution in some way. But if you think about who actually creates these impacts, this is not the university that necessarily creates these impacts. It is high impact, high growth entities. So it is largely these startup groups that really do a lot of this work. There's some existing companies that do this as well, of course, but it is not the universities that create that impact directly. We rely on these other groups that we support to help us have that impact. So, and this slide builds, it gets quite complicated, which is why it's a bit of an animation. But if you start with research, we have lots of entrepreneurial supports. We've got our tech transfer office. We're continually building startup supports. Things in red are things that we are still working on or that we recognize need a lot of improvement. But out of that research, we can create these startup companies. We've got incubators. We have an investment fund as part of our university. We've got external funders and external incubators and accelerators that can help move these companies from that startup phase into high impact, high growth entities. And then you think we also partner with existing businesses in the community to do research collaborations using some of our core facilities, and we can help leverage with some funding programs that are available to, which helps both train people inside the institution as well as lowers the cost for those businesses to help them grow quickly and take advantage of those new technologies. We do similar work with not-for-profits and community groups on the often on the social sciences side of things. We have a program that we're calling Urban Academy, where we are partnered with the City of Edmonton to really take some of our expertise that's inside the university and help our city perform better and address some of the challenges that they face. We, of course, also do work with large multinational companies. Many cases, we license technologies to them because it is a part of something. It's not necessarily a good startup opportunity, or these large companies have the skill sets to move them forward. And we have a group called Enterprise, which does that, specifically works with those companies. And those companies, can do all sorts of things. They may do research, they may hire students, they may donate to the university, et cetera, et cetera, et cetera. They may need their own staff training, and that can help them grow and have that impact in the community. We're also doing things like hackathons with a number of student groups. We're working on this venture studio concept to again, pull things out of the institution. And then we also build these things called rapid response teams, where industry has some challenges, and we're really figuring out how do we as an institution address some of their concerns in a way that doesn't look like a research agreement, that doesn't take the 2 years for a master's or 4 years for a PhD, but something that might be done in a few weeks using some of our core facilities or other expertise that we have on campus. We are doing lots of talking about how do we support and do more for, our grad students, our faculty, how do we engage with Indigenous groups here in Canada who also need that entrepreneurship. And then we also have this concept where we've started to work pairing some of our small startup companies with some of the large multinational companies, and that's a process called venture clienting. It's out of a group in Italy that started that process, and there's a bit of a process that we can follow to really help pair up some of those, and again, it helps the startup companies grow, and it also serves a need for some of the large companies that are out there. So that is basically, in a very few moments, our innovation strategy for the University of Alberta. Hopefully it's useful. And I can kind of summarize this whole thing. If we want to create impact, you obviously need the talent and the ideas to make that happen. You need capital to make it happen, so it does require dollars to come into the system. Hopefully it will also generate dollars. And it really requires a culture of being willing to look at commercial opportunities that are different than basic research. And I think this is a really important part and my personal belief is that it's important for all researchers to be thinking about where can their research be applied, how could it be used by somebody else, not just published and put in a journal and sitting on I guess a virtual shelf at this point, but basically not having the full impact that it could. So it's really important to build that culture, and that's something that takes a long time to really do that. This is a sort of a summary of a number of the programs that we have, and you'll see by the different colors. We've got training programs, we've got resources that are available to people that they can simply access. There's a number of funding programs that exist either within the university or in the community. And then we also do quite a number of events, and this is by no means a comprehensive slide because it very quickly gets too busy, but you can see along the bottom that it really starts with this awareness and ideation of new ideas, and then moves through that market validation, proving the concept that it works, and ultimately scaling and then growing that opportunity. So I will stop there and happy to answer any questions. And if you feel like you have more questions for me after this talk, feel free to reach out to me, and my LinkedIn QR code is there, so you can just drop me a note there.
Thank you very much, Dr. Fast. Can we please give him a round of applause for that? Thank you. So this is what's going to happen now. We will open a moment for the question and answers, comments, and any contributions that the audience would like to make as a way to engage you. But I also just want to say, from my point of view, given what you have just shared with us, I looked at that example that you used, the suitcase example, as an innovation. I was laughing, actually, because I was like, you've got a suitcase, then you move on to have a very short handle, and then from there you've got the wheels. Now we see suitcases that have wheels but much more longer handles. And my question was like, what's next? So that really caught me. Okay, thank you. So we will now have the audience to engage you. We've got the microphones that are roaming. Please just raise your hand and they will get the microphone to you. Okay, so the questions are coming.
Thank you so much. My name is Thomas. I've got, maybe it's an advice that we need from you. Because in terms of technology push, where you develop, you see the gaps and then you develop and you have got a technology. Now the challenge is now pushing it to the market becomes a challenge. Because now most of the industries here in South Africa, they are reluctant to take up the technology. And then we end up starting with the small, medium enterprises. And then there is that gap of a training because they are not aware of the science that we do. So in that case, how did you handle it? Is it training on your side or is also training for the people who are taking up the technologies? The second question.
I would argue that everybody needs training. It's not just the people who are creating the technologies, but it is also the people who are receiving the technologies. One of the challenges that we face here in Alberta, and I don't think it's very different than the challenge you described, is our local businesses are not the ones really reaching into the university to pull out technology. So how do we educate them in terms of seeing the advantages of what we are doing and helping them to take those technologies? So we've done things like created some micro-credential courses that the general public can take to sort of create some of this awareness. That's one of the tools. I speak at a lot of business events. Again, just telling them about what we're doing. And in virtually all cases, people are surprised because they have no idea of the research that's actually happening right in their own backyards. And I think this is true around the world. So, that's great for a subset of technologies and for some of the startups and, getting some of the industry problems in, that's a good place to get some of that industry perspective. It's a big effort to get technologies into the hands of the right people. It's a sort of a bit of a needle in a haystack kind of problem. So, we use a variety of tools. We use passive marketing for our technologies. Just we have a website and people can come and look and see what do we have available. We also use a variety of databases to find companies that might be interested, talk to them and send them a one-pager on this is what we have, are you interested? That kind of works. We use LinkedIn extensively to make sure we're talking to the right people in the companies, because it's not only have we got the right company, but if we got the right person inside that company who is able to make that decision to move forward with the technology or not. So it's a very high touch, high effort sort of challenge that we have. We in Canada live right next to the US, which is a giant market, and so it's sometimes, but it's hard for us because we are generally so much smaller than the US to get the attention there that might be merited, and it's hard to attract those for startup companies to attract that capital. So we're finding far more success in trying to build something locally that moves it to a point where it can attract the attention of somebody bigger. So rather than trying to find the ultimate company that will have it, put it in something intermediary, help it grow a little bit so that it's got some publicity and has some traction to de-risk it and to create some value.
Thank you. One more last question. I may have missed it during your presentation. Royalties. Are you selling these technologies to industries? Or,
We do.
or some of them you get some royalties from them?
Yeah, we do, for a lot of the technologies, we do license those to existing companies. And so we will get a royalty. You know, I would say it's not enough to pay for the cost of running our office. It probably covers about, I would say, a third of the cost of running our office, but it's recognized that that's creating impact is worth a whole lot more to the university than the royalties themselves. And I think if you look at most tech transfer offices around the world, it's rarely royalties that pay the bill. Every once in a while, you'll find some. You know, but for startups, we tend to not so much license the technology. We will give them an option to assign the technology to them. So actually transfer the ownership to the company, but not right away. We ask them that they will, you know, prove to us that they are actually moving the technology forward and can attract the resources and the capital necessary to do that. And in exchange for that, we will take equity, which the venture capitalists much prefer, and it's an easier structure for any future investors rather than a license, because investors tend to not really be interested in licenses. And the other reason I would say that that's really important is, you know, many startup companies start off with a great idea that comes out of a university research, but as you know, as they're developing that, they identify that, oh, that technology needs to be changed, it needs to be adapted. And so often those initial patents are dropped. And if you have a license that's related to that patent, what can happen is the company can still continue, but there are no royalties that would then go back to the university because they are not using that original patent that they did license. So if we have a royalty stake in the, or sorry, an equity stake in the company, we still then can benefit from the upside, even if the company changes direction. But we are really careful with equity in that we do not want a controlling stake. We are dilutable just like every other investor. And you know, we're not looking for a board seat, anything like that. All we are looking for is, are they developing that technology? And typically after about 2 years, if they are moving it forward, we will assign it to them and basically relinquish all of our rights to the technology and put it in the company's hands in exchange for just an equity stake in that company.
My name is Mutabo Reginald Kumojo. Just a quick question. I'm actually fascinated with the compound that you actually showed. My question is, did you actually get to explore the extractions, like plant extractions? The second question is, you're doing collaborations and then you're actually trying to liaise institutions and all that. What is the business canvas model that you might have used to do such? Thank you.
Okay, I'm not sure I heard the question clearly. It was a bit muffled. But I think the second part of your question was, what tools do we use to assess the market and the patent for the technology? Is that correct?
Yes, that's true.
Yeah, so we use a variety of tools. You know, we subscribe to a number of patent databases, you know, that help us look at what the prior art is to determine if there's something that's worth patenting. You know, we use a tool called Wellspring Scout to help us identify which companies are perhaps interested in those technologies, i.e. they are filing patents, they are working in this space. There's press releases. They've said something. It's by no means a perfect system because, many cases companies don't want to be found because they are trying to develop something quietly before anybody else recognizes that there's a market opportunity there. So it's, we've used these tools, but it is always challenging finding, what the true market size is. And who the best receptors for those technologies are. So it really is, we have our best guess, but rarely is that information perfect. And when we're filing patents, what we will do is we'll file a provisional patent first, which gives us a year to add new information if we need to. And also during that time, we are doing some initial outreach to companies who we think might be interested. And, we've got a number of partnerships with venture capital firms who have very graciously said, send us your one-page summary of your technology. And if we know anything about the market, we're happy to give you some feedback and instruction on that market, and maybe tell you a little bit about who else is working in that space. So that type of relationship has been very valuable. They don't want to see every invention disclosure we've got. So we have multiple groups that, we try to only bug them once or twice a year so that we've got, a pretty steady stream of people we can ask questions to. And then, sometimes the technology scouts for large companies are really quite good at providing feedback as well. And, at least in the pharmaceutical industry, many of them have these people who are going out and looking for technologies. And so we, engage with them. And again, they're generally pretty good about saying, yeah, that's interesting. That's not us, but go talk to these other people. And it's really about using that network to try to get as clear a picture as you can. But it is I would say there's no perfect information that's out there when you're trying to assess the market or to find the right partners. And I'm not sure I answered the first part of your question. Maybe you could repeat that if I haven't.
Okay, my first part of the question is actually regarding the synthesis of that compound that actually showed around 22 reaction steps. So since well it's an organic compound per se, my question is what was actually costly in the synthesis of that compound? And don't you think that going into the plant extraction route might have lessened the cost of production in a way?
I'm terribly sorry. I couldn't quite understand the question.
Okay, you showed a compound that actually had less than 1% yield there. And you actually mentioned that it was quite expensive in actually synthesizing that. So my question is, what was actually expensive in producing that organic compound?
Did you hear that, Doctor?
Not quite. It's just a little fuzzy, and I can't turn my volume up anymore.
Okay, I think with all due respect, maybe we should move to the next question. Or maybe he can then use a different microphone if that will help. Okay, can we get the next question? Then we'll get back to him. Thank you.
Thank you very much, Dr. Darren. My question is also on the particular compound that you showed. You said something along the lines of stereochemical complexity of the compound. So I wanted to ask if you have been able to resolve or update such stereochemical complexity of this compound. And if so, how did that impact both the synthetic accessibility and any in vitro, in silico predictions that you were using the compound as a drug compound?
Yeah, I mean, the example I gave was about 20 years old. So at that point, we didn't have any good tools. But, you know, I know now when we have new chemical compounds that we are looking at, we do use some of the in silico modeling to see, you know, is there a way to build it? And those have actually been quite useful, and in many cases, you know, have really helped. And so anything that is chemistry related, I think, you know, that space is moving much faster than it ever has. And so, you know, we're able to use some of these new tools to either optimize or find similar active groups. So our researchers are using those, and in the tech transfer office, I know, you know, my staff use some of those tools as well to assess how feasible manufacture can be. And then, of course, you know, anything that's a toxicology related question, you know, as much as you can do in silico or in vitro, you know, is much preferred to, you know, having to do animal studies.
Okay. Good morning, Dr. First. Thank you so much for the wonderful presentation and really simplifying and sharing the model that you have at your universities. It really looks like a very good model to support research output, especially for impact and commercialization. I have two questions. You mentioned the primary reason for most failures is the product market fit. So just maybe in simple terms, what does that constitute? In practical, literal terms, what the product, how is that determined? And importantly, also at what stage would you suggest that is done, and that whatever the outcome may really more or less really indicate what would be would be in real life, what it will determine in real life. So my second question is on the benefit to the researchers. In your talk, I did not hear where the researchers fit in terms of where now you get the product to the market. What will be the benefit to the researchers in your model that you use at your university? Thank you.
Sure. So I'll answer the second question first. You know, the benefit to the researchers, you know, in many cases is prestige. You know, because I think it feels good if something that a researcher has done that, you know, can help people in the market in some way. If their product, you know, their solution is used, that's good. It buys them a lot of credibility, and you know, and in some cases, there is a monetary return as well. But I also think, you know, from the startup perspective, because we are really encouraging a lot of our graduate students to be the ones who are starting companies, not necessarily the researchers themselves. You know, they can feel proud because they will own a part of that company, but they also are empowering that next generation, and so that is the value to the researchers. And I think, you know, by doing something that has some commercial applicability, and I'll use the word commercial quite broadly, you know, they can make their research better and more applied, which you know, at least from my discussions with researchers, is something that they tend to value, because then they can see that they are actually creating an impact in the world, and that is good for them. It is, it's good for the institution. It helps them attract more research dollars because if companies see that they're working on real world problems, they are more likely to provide them funding and do research collaborations with them. So, in many cases, we see these researchers who have some success in this space have even more success, because it's there. Now, that does not mean that every single researcher should try to commercialize what they're doing, because there is some basic research that is probably simply too far away from the commercial use at this particular point. But I do think it is useful for those researchers who are doing even that very basic research to periodically ask themselves the question, how could this be used in the future? Like, let me imagine, if this research continues for another 10 or 15 years, what does it develop into? Like, what might it be used for? And that to me is a really interesting lens that sort of helps people do better research. And I think that's part of the cycle that's happening here. And then you asked as well about sort of impact versus product market fit, if I heard that correctly. And so, the product market fit is basically, will somebody buy it? And or pay for it at the end of the day, because as you're aware, none of this just happens. It requires intentionality. It requires dollars to move that forward. But so you can't have impact without getting something in some way into the market. It may be, and I'll give you an example of a thing that we've created that has very little dollars associated with it, but is having a great impact. And so it is a scale that is used to measure elderly people's ability to live on their own. And so, do they need to be in a care home type facility, or can they live independently? And this is a, validated tool that some researchers spent, many years developing these questions and saying, if people answer this way or that way, what actually happens? Are they truly able to live on their own? That is something that we license out to many. I think we've got several hundred licenses for that test. It's being used quite widely around the world. Massive impact. Not really any significant dollars that come back to the institution or the researchers, but really a big impact. But it still has that product market fit, because the physicians and the nurses that are using that test are seeing the value of that, and the patients or the people in this case who are the subjects of that test are also getting value, because in many cases people would much prefer to live independently than be placed in a care facility, and it's obviously the cost is much lower for them. So while it doesn't make us any money, it saves the system a lot of money. So then there's value in that.
Thank you, Dr. Fasten. Good morning to everyone here. Good evening to you. My question is on, you mentioned that one of the limitations has to do with awareness in the context of, driving towards commercialization of research. My question then is, when we want to foster this culture of entrepreneurship, especially in the sciences, do we do this, in your view, from the undergraduate programs, or do we then just force postgrads to start thinking as entrepreneurs when they get to postgraduate levels. And if we then do the approach of inculcating this culture from the undergraduate programs, do we then incorporate it into the already existing modules, or do we then move towards a more specific approach to generating maybe a program for science entrepreneurship more specifically? Thank you.
Yeah, if I had, the ability to change the education system, I think I would start teaching entrepreneurship in elementary school the first time, a child enters the school system. That's not necessarily possible, but I think earlier is better than later. So I would definitely include undergraduates in this. And it's different levels of training for different stages of where people are at, but building that entrepreneurial mindset and that entrepreneurial thinking into every program, and I personally don't think you need to create special programs to teach these skills. I think you can incorporate that into, every course, every program should have this entrepreneurial thinking built into it. And that really means teaching faculty how to sort of have these skills themselves, which, most researchers haven't necessarily been exposed to this. So, we as university administrators and leaders have to sort of make sure that our faculty understand this stuff just as much as the students who are taking it. So it really requires building this into the entire culture of the institution to really have it be successful.
Okay, we will take the last question so that we can wrap up this session. Are we done? Okay, thank you so much. Thank you, Dr. Fast, and thank you for the engagements. You can see the interest that is coming from the audience. Thank you, and I'm going to allow Dr. Fast at least a minute so that he can, if he's got something to say to the audience as he says goodbye to us. Please, you can go ahead.
I would just like to thank you very much for the opportunity to share my experience, and really appreciate the questions. I hope I understood and answered everyone's questions, and I hope that you will have a really great rest of your conference, and thank you again for this opportunity. I really appreciate it.
Thank you.
Take care.
Okay, thank you very much. So if you check in the program, you will see that the next item is announcements, and I will quickly run through that. The first one, for the past 2 days, what would be happening would be that we have the plenary talks, but you can see the plenary talks are done. But we also have 2 talks that are lined up, and this is to the entire delegates. But Prof. Masuku will take care of that item. So the second one is just to remind the audience, the presenters, about the halls where they have to go. And please, when you leave here, grab a bottle of water. And those who are going to present, let's have the talks loaded on the system so that once we start, we don't have to stop. And the last one, very important, in the foyer, remember, we've got the exhibitions, so you have to visit those stations during breaks and whenever you can. So thank you very much, and over to you, Prof. Masuku.
Good morning, colleagues. For this short session, we have two colleagues from the university to share with us their services at the university. Both divisions have colleagues as part of our exhibitors. Please visit their stalls for more information. First to present, we have Mr. Thabo Lebelo, the Technology Transfer Office at Digital Hub Manager. And his talk is the role of Technology Transfer Office within an intense research university. Please assist me to welcome him on the podium.
Good morning, everyone. My name is Thabo Lebelo. I'm the University of Limpopo's Technology Transfer Office Manager. First and foremost, I'd like to say to the acting dean, Professor Mampuru, it's an honor to be given an opportunity to be here to share some of the insights that we have to our researchers from the university, sons and daughters of the soil, our young researchers. It's really a pleasure to come and share information that we know and that would also advance your research work. You know, the previous speaker talked about the difficulties that are there in terms of commercialization of intellectual property. Commercialization of intellectual property is not a walk in the park. Having have worked at the Department of Science and Technology, where the aim was to commercialize intellectual property, I can tell you simply that in the years that I was a bureaucrat behind the desk asking for universities to comply. I said to myself, let me go into these universities and go and work and implement the same legislation that I was telling them that they should comply to. My talk today is going to be really based in terms of saying, how do you use research for economic development of a region? And that is the key, because if you don't form ecosystems within a region in which you are in, you are going to be unable to commercialize the intellectual property or research output that you have. As big as the big cities are, where you find the big universities, Wits University, in Cape Town where you find UCT and the rest, they'll tell you it's very difficult to commercialize intellectual property that comes from research. And the reason being that, imagine if UCT is struggling, us as a very rural province, how are we going to be able to commercialize when we don't have the necessary industries within this region? So my talk is to look at how university knowledge created can be turned into creating industries within the same region that we are in, so that for future purposes, it will be then easy to translate university research outputs into services for industries that have been created because of the knowledge from the university. Maybe my starting point is to maybe share with you this slide. This slide simply gives you a tech transfer research life cycle. You sit in a laboratory as a researcher. you create, or you have your eureka moment whereby you've created something. Once you've done that, you come to us and you say to us, colleagues, I think I've found something here. Can you assist me? Now, our jobs are different. In a tech transfer office, you have two divisions mainly. You have an IP division that looks into interrogating whether the technology that you claim that you've created is new. And secondly, we have a commercialization unit. Embedded in it, it's a unit called the Business Development Unit, which does your market assessments, for example, takes your product to market as well. So when you look at this representation, it tells you about the life cycle of your research. Once you've done research, up to the point then when your research output is taken to market. But I'm not going to bore you too much about that. So what is the role of a technology transfer? So the main role of a technology transfer is to really manage how to take research outputs towards the market so that, or towards society so that it can solve societal problems. Remember, knowledge generated cannot just be generated in a vacuum. Knowledge should be generated in order to solve a particular crisis within society. And again, the knowledge ensures that knowledge and discoveries benefit people beyond just academia. Academics, as it has been said, would want to publish, and that's how they can then be ranked, you know, as NRF-rated researchers or whatsoever. But the call that the Department of Science and Technology did in 2008 in the implementation of the IPR Act was to say that universities, in terms of the mandate that they have for teaching, learning, and research, does not really just end there. They must also, as universities, also commercialize the output of their research. And therefore, the discoveries that are there must go beyond just academic activities. And the key role in building economic ecosystems. And I was saying, the university sits at a very crucial point within a region as vast and as undeveloped as Limpopo, in the sense that for you to create economic innovation and economic ecosystems, you need to be on the ground and work with the people on the ground. I'll show you a model that we use, which is called the quadruple innovation model to economic development. So what is a research-intense university? I think it's a big word, and I saw in the program we did a mistake. We said intensive research universities. But in essence, a research-intensive university is a higher education institution where research is a central mission. So research must really be considered to be the pillar of an institution, because through research is where you create the prestige of a university, is where you get the rankings of a university, is where even the researchers of university are then taken to be people of knowledge creation within a particular field or discipline in which they are in. So it is quite important that it should be a central, and being a central entity or aspect of university, it means then that the university must take reasonable care in terms of nurturing research, and that means also putting money into niche areas of the university so that in those niche areas of the university, you can then bring excellence in terms of innovation. And people would say that this research, central mission of research, is not just an add-on to teaching and other things, but it's a powerhouse for innovation, discoveries, and academic inquiries across disciplines, from science, engineering, to the humanities and social sciences. But what are the characteristics of a research-intensive university? For one, a research-intensive university should be able to get investments for its R&D at a very high rate. So, because if you are now prestigious and you are known, you are not going to beg for money. Money will come to follow you in terms of the university in which you are in. Secondly, high research outputs. Faculties and students regularly publish in academic journals, contribute to conferences, and secure research grants. So within a university setup, as you are sitting here now, you are presenting in a conference, hoping that someone might see your research and say, wow, I'm interested. Maybe in the next years to come, working with the faculty, we might just try to find investors before the research faculty day happens that would come and listen to your, presentations. Who knows? Maybe some of them might be interested in the work that you do. And graduate programs. A university which is research intensive must have a strong emphasis on master's and doctorate offerings within the university, which would also, by themselves, attract robust funding. And there must be mentorship programs within research-intense university. Then that also means that, your researchers themselves must be at the level whereby they can mentor you as much as they were mentored to be in the position that they are in. Cutting-edge facilities. You cannot be a research-intensive university if you don't have the state-of-the-art in terms of your laboratories, your equipment. These are key, and this is something that a university must actively try, by all means, to secure such facilities, because those will be the drivers to the status of a university. Global reputation. If you are a research-intensive university, you'll have global reputation that, your rankings will go up, you'll get international recognition and all that. And that is key in terms of attracting people that could come and take your research. Remember, if you do research and no one knows about you, no one will come to you. Faculty researchers and professors, these are people who are scholars, and then their work is to train future academics, and it's key to have people with capabilities to do that. I want to talk about the theme that or the topic that I was saying, that mine is to say, how do you as a university become relevant within the region in which you are in? And how do you then harness that to make sure that your research outputs are taken up in the region that you are in? It goes without saying that Limpopo is a rural province. You find that only about 8% of its residents live in the urban areas, and about 82% live in the rural. And it is in these rural areas that some of the economic activities of the province are taking place in. And for example, if one looks at the Limpopo Development Plan, 2025 to 2030, it revisits the Limpopo Development Plan 2020 to 2025, which highlighted agriculture, mining, tourism as the key components of economic development within this region. And now the question is, as a faculty of science and agriculture, this is where the university should really concentrate in niche areas that talk to agriculture, that talk to mining, that talk even to tourism. Let me give an example about agriculture, and let me see how you would react to what I'll say. In my engagements with farmers around, I was intrigued to find out that, as a university, we still have a long way to go in terms of affirming our strong foothold within the province. One day I was discussing with some farmers who plant macadamia, and they said to me, we have a problem. They are in Mopan. They said, Mopan is a tropical area, and if we are planting macadamias there, they grow very tall. And as they grow very tall, it becomes a problem in terms of pest control, because now you have to engage services of drones, agricultural drones, and it's very expensive to use such. So they said, if the university could help us with making sure that the stems of the macadamia are shorter. And if they're shorter, it means the tree is not going to grow too tall. Therefore, the yield becomes what they expected. So, just by saying that, for me, it said, wow, our horticultural people within the university, if they could hear what I'm hearing, and if I take them to these commercial farmers, they would then be in a position to first do research that will talk to the shortening of the stem of a macadamia plant, by virtue of doing that, you automatically also now own the value chain in terms of propagation of those stems. And that talks to creating a company that now supplies these commercial farmers with these short stocks. So already you can already, as a researcher, spin out a company that when it lands into the market, it services the commercial farmers. And that is the key of economic development. The university knowledge must go and target where there are problems and create solutions. And by creating solutions, then you are creating economic viability. In terms of mining, for example, maybe in the next slide I'll show you something about mining, which is quite important as well. But that being said, I know the university has niche areas. I don't want to dispute that they are capable and they do a good job. There is what is called currently the sunrise sectors. And these sectors are not in any way meant to move the province away from the initial or the primary economic drivers, for example, your mining, agriculture, and also tourism. But these are meant to also assist in terms of what is relevant now and how can the relevance of these sunrise sectors help propel the economy further. For example, in terms of automation, you know, when one looks at the mining industry in the province, which is largely, you have your PGMs, which are platinum group metals. The platinum group metals that are mined here, if the university plays an active role in such, we might just see in the province beneficiation of some of these minerals. For example, in what do you call, in terms of remission or reducing of emissions of cars, for example, the PGMs can be used in catalytic converters, which trap the emissions so that you don't have a lot of emissions out there. And this is important. We have iron ore. Iron ore is used mostly in terms of the production of vehicles and vehicles components as well. So just imagine, if the provincial government was able to negotiate with the likes of your BMWs and say to them, why don't you come and have your plants in the province that will then do these components of cars, and the national government then cuts taxes on these entities. In a way, we will also stop the outflow of young men and women from the province into Gauteng, where there are jobs, but we'll retain them in the province. We also look at the issue of cannabis and hemp. I'm proud to say that as a university, we actively are taking part in the development of the strategy for cannabis and hemp production and industrialization, which is also one of the upcoming industries that, you know, the university is doing a lot of work here in terms of research. I can quote one researcher, Dr. Hadebe, who's doing quite good work in this space. Renewable energies. In terms of, you know, renewable energies, here again, in terms of the sunrise sectors, the university has a very important role it can play in terms of, you know, your production of hydrogen, conversion of hydrogen into electricity. We have crises in the country in terms of load shedding. Again, the minerals again that we find in the province, like magnesium, can be used again in battery materials, which Professor Ngwepe is doing. So this is just to say, we can actively turn around the province of Limpopo to be an economic powerhouse using knowledge from the university. We look at, again, issues like, for example, the wild economy. There's a lot that we can do because tourism is one of the biggest contributors of the GDP of this province. There's issues of breeding, for example, which, you know, our agricultural students in terms of animal production can work together with industries, your game farmers and things, to breed for them. Now, I was talking about this model for innovation or economic development within the province. What we have done is to say that we as academics should work with private sector, public sector, and communities. And one of the projects that we are doing in the Mopane region is to work with the municipality there in terms of their local economic development and try to find out how can we turn around the economy of Limpopo using knowledge from a university point of view. I'm pressed for time this side. And then what are the benefits of creating such ecosystems with different parties? The benefits for one is that, there's attraction of funding that comes through to the university, contract research, recognition of academics, university revenue streams, access, you give industry access to cutting edge technologies, and also you create jobs, circular economies within rural settings and technology benefits. And in conclusion, I'd like to say, don't just do science for the sake of doing science. Do science so that the application of science can create economic development within the province of Limpopo. As I wrap up, I would say we are hosting next month the regional Moringa conference with the Green Biotech unit within the university. It is stretched across 3 tracks: academic track, entrepreneurship, and innovation. Do register. Thank you very much.
10 minutes turn out to be 20 minutes. I'll take one quick question. And the rest of the questions, we do have some colleagues from TTO office outside as part of our exhibitors. So I'll take one quick question. Yes, honestly. I guess they are well informed by your colleagues outside. And feel free to talk to the exhibitors outside from the office. And Mr. Lebelo will also be around. Oh, all right, he's leaving to attend another conference at the ranch. So, but he is around at campus. Maybe he can share where he is because.
If I may
the
share,
office.
there's this office here next to MO residence. It's the only office I think that is in the middle of a residence or a student residence. So that's where you'll find our offices. You can come there if there are any questions. Thank you very much.
Thank you, sir. Our next presenter is Dr. Yvonne Maila, Research and Development Coordinator at LATS. If she can try to stick to the 10 minutes.
Greetings to our professors, doctors, researchers, and students, and the House at large, all protocols observed. My name is Yvonne Maila. I come from the Limpopo Agro-Food Technology Station within the University of Limpopo. And today I'm going to talk to you about how you can add value to your research project and develop it into a marketable product. Hence the theme from lab to market. The previous speaker was talking about how it can be a challenge to commercialize your research. And for me, I'm going to talk to you how you can develop your findings into a product. We know that researchers are trying to find solutions to various industries. It can be the pharmaceutical, it can be the nutraceutical industry, and also agriculture at large. So, a bit of background on our department. We are a center of excellence that has been established in 2008. We are within the University of Limpopo, but serving under the School of Agriculture and Environmental Sciences. We are under the Technology Innovation Agency, and we are funded to service the SMEs in Limpopo province, which turn their agro crops into marketable products. Our vision is to be a center of excellence that improves SMEs agro-processing services in Limpopo province and other African countries by means of using the cutting-edge technologies that we have. And our mission is to improve agro-processing services rendered by SMEs, mainly in Limpopo province and other parts of South Africa, through modern technologies and partnerships. Our objectives is to provide services to SMEs for their agro-processed products to comply with the national and ISO standards, and also to enhance entrepreneurial skills for existing and potential SMEs. Also to strengthen research and development capacity in agro-food processing and value addition sectors within the Limpopo province. How do we do that? Our department, our station comprises of 4 departments. We have the Food Technology Department, the Food Microbiology Department, the Food Analytical Chemistry Department, and the Research and Development Coordination Department. That's where I am. And it is through this department that we are able to offer these services. The Research and Development Coordination, whereby we offer postgraduate mentorship under the Department of Plant Production, and we also offer training and supervisions of postgraduate students, masters, PhDs, and honors, and also we do technology demonstration to our students that are coming also from the Department of Chemistry, Microbiology, Biodiversity, and also Agriculture. And with that research, we are able to develop publications, and some of the findings, we do present them at conferences. We offer testing and analytical services in our food and microbiology lab and analytical services to students and also researchers and SMMEs. Some of you has already visited our station. And in our food development department, we do develop product, and also we improve process development in case you have a product that you need to develop or an idea that you want to develop into a food product. We are able to assist you through our food technologists. And we do also consultation and technology audit. We are able to achieve that through our various equipments that we have in different departments. In the food technology department, we have an industrial cold and freezer whereby we can store our samples until they are analyzed. We have the juice pulping machine, industrial blender, and industrial dryer, and the grinder, and the microwave. These are some of the equipments that we have in the food technology department that can assist to develop products for our SMEs and researchers. In the analytical chemistry as well, we also have the state-of-the-art equipments, the GCMS, the freeze dryer. Like for those who are drying their plant material using the oven, we encourage you to start looking into the freeze dryer because your freeze dryer will not degrade some of your micronutrients. We have the ICPE, the AC600, TrueSpec, and the water bath, and other minor equipments. In the food microbiology lab, we have also various equipments that can be used to detect your microorganisms, and also equipments that can determine the shelf life of your products. We are proud to say that the past 8 years we have made an impact. We have assisted 1,991 SMEs across the province. We have also trained them to comply with the food standard, 595 SMEs, and we did 476 testing and analysis within the university and also to our SMEs outside the province, outside the university. We developed 234 products that some of them you meet them in the market. And we have assisted 219 postgraduate students across different departments within the Faculty of Science and Agriculture. Some of the SMEs that we have assisted, for sure you have met the TM Food. They developed a beetroot using our cutting edge technologies. They come with different flavors. They've got the chutney, the hot, and the garlic. They've employed 15 employees, and they supply A1, Spar, Cash and Carry, Raza and Broadway supermarket, and also the communities around Polokwane. That's how we assist our SMEs. And we also have Harose. Harose is one of the SMEs that we are proud because he came there with an idea of developing a sweet potato yogurt. Through my office, we developed a research, and with the assistance of the Department of Plant Production, we had a student who researched the type of sweet potato that he can use to develop an antioxidant-rich yogurt. Now he's doing very well. The yogurt is there in Spa, Hosiami, Nesta Food, A1, and the government has already given him a site to do this in huge production. We also have Rave About Sauce. They are doing the sauces. Their products also are found in Spa and other selected supermarkets. They develop sauces and jams that were assisted at the station. Some of these products, you will meet them in the shops. We have our past student, Dr. Kweteps. He calls himself Dr. Kweteps, but he's not a doctor. His company is his company is doing a range of moringa products, hence the conference, the first regional moringa will be presenting there, I think. So he is developing products from his master's research. He was working with moringa plant. Now he's developing some moringa powder, moringa scones, moringa juices. We assisted him in developing this product, and now he's selling them, and he's also been funded with a facility to develop them around Mankweng. We also find his herbal moringa in a local pharmacy in Mankweng. So we have the African Nosh, they are developing ding, the fermented sorghum that you use it for your morning breakfast, most of you. So we have assisted them in developing this thing, and it is also found in Spa and Hosiami, and they are proud that they have employed several youth to work with them in their facility. And in the Research and Development Coordination Department, we have developed research with one of my colleagues, Dr. Bango, who happened to be a student in the Department of Plant Production, through his research in Jatropa iced tea, whereby he was domesticating this indigenous plant which had tea properties. So out of it, he developed this iced tea, and we found that it has it has a potential for being registered its novelty. So we filed for its novelty through the TT office, and it was accepted that it has novelty. So we are waiting for its commercialization. So we also assisted this SME with the indigenous Setosa instant pumpkin powder. He has developed a pumpkin powder that can be used as a morning breakfast also for all age groups, and also it has novelty, it has passed all the processes, and it is also awaiting commercialization. So we are proud about those two researches that we did. So thank you so much for your attention, and our stall is out there. If you want to know more about LADS, you are so welcome. Thank you.
Thank you, Madam. Exactly 10 minutes. We have 2 or so questions. We have time for questions. How? You know where LADS is? Yeah? You know where LADS is, colleagues? Okay, you have used their services? There's a hand there.
Morning, everyone. My question is going to be simple. Are the services free at that lab?
Okay, I wish I can say yes. They are 50% free for SMEs because we are funded to assist SMEs to develop their products. But hence we are within the university, we offer also our services to the department. But we know that the universities have got research money, so it is not free.
Just a follow-up from what you asked. Since you mentioned that they are 50% discounted for people who are within the institution, but what about SMEs that are from external people and are trying to somewhat build themselves as entrepreneurs and they don't have the necessary funds to uplift themselves? So how do you go about with that process from external people?
For the SMEs, the services are offered depending on their business. If you are still starting and you don't have any form of funding, they will request paperwork to prove that really you do not have, and LATS will assist you in such to establish your business. But as you continue to make money, then you start to pay something towards the services that are offered.
Thanks.
Okay.
I'm just jealous to hear the great impact you have just demonstrated, the impact you have had on SMEs, potential SMEs. And I'm wondering, how do you raise awareness among members of the community at the grassroots who may not be aware of the wonderful interventions you are able to make, just judging by the examples you gave? How is awareness penetrating into communities, especially the poorest of the poor, because there's great potential here? I'm so impressed. Thanks.
Okay, we are really working with those ones at grassroots level. Our food technologists and our technicians are always every day out there looking for those SMEs that needs help. These are the SMEs that are at grassroots level that you have seen here. We do workshops also in collaboration with the Department of Agriculture at government, also with the Technology Innovation Hub. So awareness is always there. And we have a huge database of them that we are assisting. So yeah, we are reaching them there.
Thanks, colleagues. They also have exhibitors from LATS are also outside. Let me quickly acknowledge the presence of Professor Lisibana Rafapa, research professor from the Faculty of Humanities. You are welcome, sir. I'm not sure if Prof. Manduana will want to make some announcements, or that was all. So from here, how many minutes for tea? I know you guys, you are looking forward to tea. Ne? $15, $20? 20 minutes, 20 minutes tea break. From there, go to your halls, please. Oh, take your water bottles, please.
influencing informal food traders' awareness of COVID-19 support services in selected areas of Polokwane Local Municipality, Limpopo Province, supervised by Dr. Lefopani MH and Dr. Baloyi VA. These are the presentation outline that will be covered in this presentation, starting from the problem statement and conclude with the references. COVID-19 pandemic has disrupted the livelihood of informal food traders, causing loss in income, business disruption, and increasing financial vulnerability and threatening the socio-economic factors and threatening the socio-economic well-being. In response, the government have implemented various support services aimed at mitigating the advance in which the informal food traders have been impacted by the COVID-19, such as the COVID-19 loan guarantee, the SRD grant, the Spaza Shop support scheme, and the scheme facilities and the loan COVID-19 support services. Sorry. Much uncertainty still exists on the implementation and awareness of COVID-19 support services on the intervention. More specifically, existing studies focus on the income loss and also the food insecurities and the mental health that the informal food traders enjoy during COVID-19. In response, they didn't focus much more on the awareness of the informal food traders that the COVID-19 that the informal food traders have to access. So this necessitated this study to focus on analyzing the extent to which informal food traders have been aware of the support services implemented to mitigate their lives and to improve their welfare. The scope of the study is that the study aimed to analyze the factors influencing informal food traders' awareness of COVID-19 support services in selected areas of Polokwane Local Municipality, and that the research objectives were to identify and describe the socio-economic characteristics of informal food traders in selected areas of Polokwane Local Municipality, and the last objective was to determine the socio-economic factors influencing awareness of COVID-19 support services by informal food traders in selected areas of Polokwane Local Municipality. And their research hypothesis was that socio-economic factors do not influence awareness of COVID-19 support services by informal food traders in selected areas of Polokwane Local Municipality. So coming to the methodology, the study was conducted in selected areas such as Mankweng, Paledi, and Polokwane Central Business Districts, which are areas that are located in Polokwane Local Municipality under the Capricorn District Municipality in Limpopo Province, South Africa. So the study used primary data as a data collection using a questionnaire that was structured, and the sampling procedure were the purposive sampling techniques, and then the sample size, only 90 informal food traders were interviewed. Coming to the analytical techniques, descriptive statistics was used to address the first objective, and then the binary probit regression was also used to address the second objective, and this is the model specification for the binary probit regression. So coming to the result and discussion, as you can see, there's a table of the descriptive result on the socio-economic factors. We see that age, it is part of the socio-economic factors, and that 37 informal food traders, they had age that ranges from 26 to 35. That shows that these are the youth that most of the time when they are done with their school, they resort to informal trading. And others, we have genders, which female and male, and we see that female are the ones that are dominating the informal trading. And lastly, the informal food traders were, among the others, we have the trading permit or license. The informal food traders were asked if they have the trading license, and only 57 informal food traders were asked if they had only the 57 said they have no permit. And that we have the continuous variable, such as the household with a minimum of 2, maximum of 10, and a mean of 5, and a standard deviation of 2. So also the trading hours per day, it has the minimum of 1, the maximum of 10, the mean of 4.1769, and a standard deviation of 2.34096. So these are the continuation of the descriptive results where the informal food traders were asked if they were aware of the COVID-19 support services, and only 35 informal food traders were asked, and then they said that they were aware, and 55 of them, they said that they were not aware. Among the others, we have the types of government support services, government support that they received during COVID-19 or maybe prior to COVID-19, and only 7 of the informal traders said that they received technical and training, and others said that 3 of them, they said they received business management, and others, they said that they received the protection from the officials' harassment, and then 70 of informal food traders, they said that they have no response regarding the types of government support. So coming to the binary probit regression model, we have the variables that were significant, the level of income, the access to information, trading hours, harassment, and we found out that the level of income, it has an estimate of coefficient that is positive, meaning that when the level of income increases, the awareness of informal food traders also increases. And that implies that the informal food traders that are ranging between 1,000 to 2,000, and also the informals that are ranging from more than 2,000 level of income, they are more aware of the informal food traders. And then also the level of income, access to information, trading hours, these are the ones that have a positive effect on the awareness. As they increase, also the awareness increase. So when it comes to the harassment, it has a working estimate of coefficient that is negative, meaning that when when the harassment decreases, the awareness increase, so it has a negative relationship. So in that, there are also insignificant variables, and that the following, the log likelihood and the COGS, this one shows that the model is fit for describing the dependent variable, which is the awareness of COVID-19 support services. In conclusion, the study had only one hypothesis, which stated that the socio-economic factors do not influence awareness of COVID-19 support services by informal food traders in selected areas of Polokwane local municipality. And this hypothesis was rejected as the results showed that there are socio-economic factors influencing awareness of COVID-19 support services by informal food traders. And these factors, as I've shown and I have described, are the level of income, access to information, trading hours, harassment, types of COVID-19 support services, and the type of harassment. In general, the study concludes that the awareness of COVID-19 support services by informal food traders is determined by the socio-economic factors. Coming to the recommendation, since most of the informal food traders were not aware of most of the COVID-19 support services, the government should provide offline communication options like local radio, printed posters, and SMS updated, not just website or social media, and that the government must set clear rules protecting traders' rights and train municipal officers to handle enforcements with respect in case like when the informal traders are harassed, and that they have to implement paperwork and online forms that should be short and easy to understand, and to provide help desk or kiosk where traders can get assistant filing in online forms. So I would like to acknowledge my supervisors for the unwavering support and guidance throughout this journey, and that and also to acknowledge the University of Limpopo for giving me the platform and resources essential for this, and also the informal food traders. These are the references that I used to compile the presentation. Thank you.
Thank you very much, Palisa. next time the presenters, if you see me standing, you must just know that you are now on the red line. Eh? Can we give here another round of applause for this wonderful presentation? Can we open this time for questions and answers? I see Dr. Lidwawa over there.
Thank you very much, Palisa, for the wonderful presentation. There is one part of your results that caught my attention, which makes me wonder, haven't you find any reason to ask why about that? The trading hours. There is one trader who only traded for 1 hour. What was the reason? Thank you.
I think this was a mistake that time when I was compiling my presentation. So the trading hours, they have a slight mistake. They're supposed to be 7 because most of the informal food traders, they are working more than they can, they work more between maybe 7 hours to 10, maybe 15 hours so that they can improve their income.
Thank you for that response. Any other question? We can take 2 more questions if you have questions. Can you move the mic over there?
Thank you, Palisa, for a nice presentation. Please advise me, because COVID-19 is gone now, and your interventions, you think they would be appropriate at this point in time because we are no longer facing the COVID-19 pandemic?
Okay, thank you for the question. Yes, the intervention, that's so important for all of us to know, and also for policymakers to know in case like future crisis on how they can be able to combat and how to know which areas they can help the informal food traders, such as the awareness. As you can see that there were studies that were only focusing on the access, but they didn't look at the parking. The drives of the welfare, the drive of the awareness.
Thank you very much for that response. Can we give her a round of applause as she is going to sit down? Thank you very much. Can we move and call the second presenter, Mr. Sithole? Can we give him a round of applause as he's ascending the stage? Mr. Sithole is going to talk to us about the recovery technologies in acid mine drainage. Thank you very much.
Display.
The impediment to action advances action. Good morning, ladies and gentlemen. Welcome to my presentation. I'll be presenting a review as a first-year master student in water and sanitation. My title of my review was dealing more on reclaiming resources from acid mine drainage using eutectic freeze crystallization. This is my presentation outline. So now let's start by understanding our context on acid mine drainage. According to studies, South Africa is a rich mineral resource base and plays a crucial role in our economy. Mr. Lebelo recently spoke about the importance of looking at key factors in economic growth. So figure 1 shows our problem now, where we are looking at the schematic diagram of the formation of acid mine drainage. So we can look at number 1 on our figure. We have precipitation and mining operations. So when this mining operations releases water, it's not initially acid mine drainage, but during its runoff, it experiences leachates on the sulfates, bacteria in presence of oxygen. Then number 2 shows the result of acid mine drainage, which has a low pH of less than 3. As a result of that, when it continues to run off, number 3 shows when it encounter with fresh water. On Monday, someone was presenting on how they were experimenting on goats, giving them acid mine drainage. So if maybe you have checked that. So number 4, as a result of that, it results in toxicity toxicity accumulation in plant and aquatic life. Number 5 shows our yellow buoy now. We call it yellow buoy because of its yellowish color. And above is the chemical reaction. So this was with the help of Mafane et al., my colleague in water and sanitation. You'll be dwelling more on that. So this is my recent visitation. Yesterday, I went to Mpumalanga to review exactly if the studies are correct on this. So it was a correct indication and a perfect review. So I'll be looking at the objectives on how to review the current approaches of acid mine drainage so that I can introduce my EFC and then how it can be applied to acid mine drainage. The current approaches, they use technologies such as ultrafiltration, reverse osmosis, and direct freeze crystallization. However, these methods are expensive and large applicable in large scales. But then as a result, they went on to then approach this problem by using abundance enough conventional treatment. So as a result, their remediation was that they will need to advance to immediate action now, such that figure 3 shows how this method resulted in terms of releasing a high wastewater which has high sulfates and heavy metals. So this now why we need eutectic freeze crystallization. Firstly, let's understand what is EFC. EFC is a technology that uses temperature to recover and remove solids. So EFC operates by reaching a eutectic point, which is called the freezing point, so that we can have water security and mitigate health risks by making sure that our water is clean and very sanitated. There was an approach in United Nations conference where they now spoke about SDGs, which are very important now in our generation. Therefore, EFC as a result is a cost-effective method. You can apply it at home, and then it will give us valuable resources and potable water. How it works, looking at figure 4, we have on our left-hand side, that's a reactor where now we have ice and salt. Then the right-hand side, we have the graph and the principle of EFC. So on the principle of EFC from A to B, that's where now our solution is introduced so that we can drop the temperature to a certain temperature. So studies such as done with MAL in China is that they drop the temperature to -4 degrees so that they can have ice separation and salt. Then why application of EFC is very important to acid mine drainage? Not specifically, but then why is very important to acid mine drainage is that acid mine drainage contains high concentration of metals, sulfates. As a result, it converts the wastewater, which is our AMD, into useful resources, such as iron, aluminum, and magnesium and copper. So magnesium is very important, as now I'm presenting to people which are older than me, which now some of them experience blood clots, but then that's where I come in. Then the products that may also be metal deficient in terms of this. So case study review was done, the one internationally and locally. So the purpose of reviewing my international reviews is that I'm showing you how this is also applicable locally and international. There is an interaction. So number one shows that they use pilot studying, testing for EFC, and the outcome is that they supported a circular economy. But then the gaps is that the long-term economy and this real case is still a problem. Case number 2, which was done by Mogashan, a very interesting author, focused more on AMD brine, where the result was good, pure water and gypsum. And problem was that the limited integration with pretreatment. So I'll take you to number 5, since those one explained the same as Mogashan. So number 5, which was done in China, is that their AMD was using iron and copper and magnesium salts. So they recovered high purity of metals reclaimed. But then problem was that now it needs further optimization to have the purity of metals and water. EFC comparison with other methods is that it reclaims salts, no membranes required, low energy. The challenge is that now reaching the freezing point is very difficult, as it needs careful consideration. So the initial capital and operational cost of this method is still a problem, and scaling up to industrial level is still a problem in this generation, and also optimization of our recovered valuable resources. The conclusion is that AMD is both challenge and opportunistic. We don't have to see AMD as an enemy, but then as an opportunity to grow our economy. By doing that, EFC offers a promising solution for this problem by water recovering, which will be large amount and valuable salt and metal extraction. And also, the most important thing, we have to look at zero liquid discharge. Still, even though when you are dealing with wastewater, you must ensure that you reach zero liquid discharge. No amount of thing should be harmful to the environment. And then with further optimization, EFC can transform our AMD management. Then future studies should look at the following: scaling up to industrial level, and then develop integration systems where now they combine neutralization using lime and EFC all together. So now we can solve more problems now, so that we can reach SDGs number 3, 6, and 15. Number 3 talks about the human well-being, and number 15 talks about the innovation, so that the youth can also prosper in this. And also looking at investment is very important. I don't know if it's my observation or what, but in South Africa we lack collaboration and communication. We depend more on people's methods, but then we are lazy to think critical. So strength, and as I said, strengthening collaboration is very important, so that in academia, also for commercialization, sorry for that, then these are my reference I used to conduct my review, and acknowledgements goes to the below. Thank you. I would love to take any questions.
Thank you very much for your wonderful presentation, Mr. Stoller. Can we give him another round of applause, please? He is very confident. He's challenging you guys to ask questions. Can we ask questions to Mr. Stoller? Guys, are you intimidated? Dr. Duava there.
Thank you, sir, for the wonderful presentation, though I'm not from your field. I can see that you're saying the EFC, the problem with it is that it is expensive. Then my question is, from the recommendation where you're saying you recommend that it be distributed or scaled up, is it a sustainable approach for one to scale it up? Because the only limitation that you have indicated is that it is expensive.
Thank you, Dr. Who? Ledwaba. Okay, thank you for this good question. And then I didn't say it's costly, but I said initially when you study this operation, it's very costly. But going on compared to the other methods I reviewed, it becomes sustainable. Okay, thank you.
Can we have another question? Any more question? Maybe a question from me, Mr. Stolle. South Africa is a water scarce country, and mining actually requires or uses a lot of water. Do you think we should continue mining, also taking into account the damages that mining actually bring to the natural resources? Thank you. He said I must repeat it. He didn't hear me well, but he's closer to me. I want you to share with the audience whether South Africa should continue with mining, because mining requires a lot of water and we are a water scarce country. And also taking into account the damage that mining brings to the natural resources, including the land. Thank you.
Okay, thank you. Doc, what's your name? Okay, so the thing is that when someone asks me a question, I need the name first, so that, because they know me. Yes, South Africa should continue with these mining operations. You know, South Africa has systems where and the rules that they follow after operation and extracting resources. And then they again go back so that they clean the land and make sure that it's very in good conditions. So, and mining is the second sector now. By now you know that it contributes to the economy after agriculture. Am I right on that? Yeah, I'm right. So they should continue, and it helps us a lot, so we have to.
Thank you very much, Stolle. Can we give him a round of applause? Can we move on to the third presenter, Salome Letswalo? Salome Letswalo, are you around? Yes, can we chair her up as she is ascending the stage? Salome is going to talk to us about the impact of edible coating on the maintenance of physiochemical properties in tomato fruits.
Thank you, Chair, and greetings to you all. As I was already introduced, my name is Salome Letswalo, and I'm a Master of Agricultural Management student specializing in plant production. Today I'll be presenting my research titled The Effect of Citrashine, Edible Coating, Dipping Time and Storage Duration on the Maintenance of Physicochemical Properties of Classic Ground Tomatoes. This research is part of my study level, and it is supervised by Dr. MJ Mothapo with co-supervision from Prof. N. Matava and Dr. Kesh Kwampa. Tomatoes are widely consumed fruits worldwide due to their nutritional value and health-promoting properties. However, they are highly perishable, which limits post-harvest storage and their marketability. To address this, the post-harvest treatments such as edible coatings have been explored. One of them is Citra Shine, which can extend the shelf life and maintain the tomato quality. The aim of the study was to investigate how different Citra Shine dipping time and storage duration influence the physicochemical quality of classic ground tomato fruit during post-harvest storage. The objective was to evaluate changes in key physicochemical properties of Citra Shine coated tomatoes under varying dipping times and storage duration. This study used the classic round tomato fruits harvested at the red maturity stage from ZZ2 Mweketi farm, and the experimental design used was a 4x4 factorial design arranged in a completely randomized design. The dipping time applied were 0, 2, 4, and 6 minutes, where 0 served as control or uncoated fruits, and the storage durations were 7, 14, 21, and 28 days. The tomatoes were stored in a cold room at a temperature of 10 degrees Celsius throughout the study. All data analysis was performed using GenStat. The parameters measured on the physicochemical properties include the weight loss, firmness, decay, the total soluble solids, titratable acidity, TSS to TA ratio, and also the color values such as lightness, redness, and brightness. The results showed a significant difference between the coated and uncoated fruits, and overall the coated fruits performed better. They had a lower weight loss and decay, lower total soluble solids and TSS to TA ratio, higher firmness, color, and acidity. There was also a significant interaction between dipping time and storage duration, and the most effective dipping times were the 2 and 4 minutes. They're the ones that managed to maintain the tomato quality than other dipping times. Which means that Citra Shine coating successfully slowed the deterioration and preserved the quality and nutrition of the tomato fruits. The proper dipping time of 2 and 4 minutes is the one that maximized the benefits. The reduced weight loss and decay can be explained by the coating, can be explained by the semi-permeable barrier formed by the coating that reduced the moisture loss and microbial growth. The higher firmness and color retention is likely due to the coating slowing the enzymatic degradation and respiration. The lower TSS and TSS to TA ratio in coated fruits suggests that the coating slowed the metabolic conversion of acid to sugars. And the optimal dipping times of 2 and 4 minutes is provided the sufficient coating thickness without oversaturation or negative effects on gas exchange, unlike the longer dipping periods such as 6 minutes, which was less effective, likely due to oversaturation or reduced gaseous exchange. These are the graphical representation of the results from figure 4 up until figure 10. This study contributes to post-harvest science by showing that Citra Shine edible coating can effectively maintain tomato quality and identifies the most suitable dipping times. It also addresses the consumer's concern as edible coatings are safer alternative to chemical treatments, ensuring healthier food products for consumers. The study concluded that the application of Triage and wax coating, particularly the 4 and 2 minutes dipping times, effectively retained the quality attributes and physicochemical properties in tomato fruits during cold storage. Compared to control fruits, Triage and coated fruits showed lower weight loss, better firmness and color retention, and higher level of acidity. The storage duration also had varying effects depending on dipping time. And overall, the Triage coating improved the post-harvest quality of the classic ground tomato fruit. Those are some of the references cited in the study. And I would like to acknowledge my supervisors and co-supervisors, the University of Limpopo, and everyone who supported me throughout the study. Thank you.
Thank you very much, Salome, for such a wonderful presentation. Can we give her another round of applause, please? That also opens the time for questions. There's a hand at the back there.
Good day, ma'am. My question is, did you test at different color stages of the tomatoes, since we know that tomatoes have color break, red, and also a green stage? Did you check at like different color stages?
No, we did not check at different color stages since the tomatoes were harvested at the red maturity stage, so we only checked the one that are at the red maturity stages.
Okay, and my other question is, why did you choose the Triage wax instead of other edible coatings?
The Triage edible coating is already approved commercially, and it is also safer, and also there are less researches which investigate its effect on tomatoes. So mainly mostly it is applied on citrus. So there's only less researches that investigate its effect on tomatoes. So we also wanted to check if it can also work wonders on the tomatoes.
Thank you very much. I will advise that we give one question per person so that we spare the time for others to ask as well. The guy there, yeah, go ahead.
All right, thank you. I just want to ask a question. I'm not from this field. The choice of your time series, because you simply started from 2, 4, 6. Why didn't you at least try 1, 2, 3, 4, 5, 6? I mean, it was going to give you a better understanding, because I can imagine if you are coding for 1 minute, it performs better than 2 minutes, as well as that 4 minutes that you concluded to say between 2 and 4 minutes, your coding works well. So my question is, why did you skip 1? Because 1, you might find that at least with a lesser time, you could achieve a better result. Thank you.
Okay, thank you for that question. We started at 2 minutes because I think 1 minute is just close to 2, so there's no much different on that. And also we wanted to check both short dipping times and longer dipping times and find the balance, which one can perform better.
Thank you very much. If we don't have any other question, we can give her a round of applause as she's going to sit down. Thank you very much, Salome. Let me remind those that are going to present next that please make sure that you load up your PowerPoint so that we don't waste time. Our next presenter, ladies and gentlemen, is Nomcebo Malaza. Can we cheer up as she is ascending the stage? Nomcebo is going to talk to us about the evaluation of extreme weather events that impact domestic water abstraction in the Maruleng municipality.
Please be sure to receive my very humble greetings. My name is Nomcebo Invombega Malaza, presenting my study, which is titled Evaluating Extreme Weather Events Impact on Domestic Water Abstraction in the Maruleng Local Municipality of the Limpopo Province in South Africa. This study was carried out under the following supervision. Just to give you a bit of a background, let me paint a picture for you. Imagine waking up to extreme rainfall today, extreme low rainfall the next day, and with scorching heat in between. That's the reality of weather today. While the whole world feels this, the sub-Saharan Africa here carries the most burden of this extreme weather. So these extreme weather events, they don't just change the weather, they affect our water resources, affect domestic water abstraction, and also affect our progress towards the SDG goal number 6, which is focusing on availability of water and management of water for all. The problem that is driving this research is the heightened risk of extreme weather events, which is brought up by climate change, which was noted by Isaac and Musenwa in much of Africa, especially in the semi-arid regions. Hence, the already strained water resources, they are predicted to face even more challenges in the future. So villages like the Oaks, Haskororo, and others in the Maruleng local municipality are already facing water supply problems. Why this study? I'm doing this study because it is very much imperative to navigate through the problems that are related to water supply. Hence, this study focuses on ascertaining the extent and the nature of water abstraction from the source, challenging the challenges that emanate from extreme weather events in the Maruleng local municipality. This is because researchers such as Raymond and others in the year 2020, they stated that it is very much imperative to comprehend the effects that these events come with in our water systems. And studies, they also emphasized further that we need strategies that are based on science that will help us to rather combat these problems that comes with extreme weather events. Therefore, this study comes into the place that will further assist water managers in making informed decisions going forward that will ensure domestic water security to vulnerable communities. Which brings me to my main aim and my objectives. This study aimed to interrogate the extent to which extreme weather events affect domestic water abstraction in the Maruleng local municipality under the Mompane district in the Limpopo province by analyzing the trends of extreme weather events over the Maruleng local municipality and investigating the domestic water abstraction patterns from the Mamejas Kororo water treatment works, and finally to evaluate the relationship between the extreme weather and the domestic water abstraction. My study area is the Maruleng local municipality, focusing specifically on the Mamejas Kororo water treatment works, which is where they get most of their water. So for data collection, the climate data was collected from ARC and South African Weather Service, and the domestic water abstraction data was collected from the Mamajas Kororo Water Treatment Works. For data analysis, for hydroclimatic trend analysis, I used the non-parametric modified Mankendal test to identify weather extremes. I used the EVT method, specifically the peak over threshold method. For domestic water abstraction patterns, I used a simple Excel analysis. To evaluate the relationship between climate variables and domestic water abstraction, I used the Spearman rank correlation coefficient. Now coming to the results. In the study, rainfall records show no significant trends, as you see in table number 2, meaning there is insufficient statistical evidence to conclude that there is an increase or a decrease. Such results were expected given the highly variable nature of rainfall in semi-arid regions, where fluctuations are strongly influenced by sporadic convective storms, as per the literature that I have engaged with. Now coming to table number 3, the minimum temperatures also tells a similar story. They are rising but without statistical significance. A clear standout of the findings now come in table number 4 with the maximum temperatures. With the maximum temperatures, with stations like the Hoedspruit station and the Met stations showing a clear, significant warming. This is echoing the past studies that reported that there is an increase of about 1.3% in the maximum temperatures over my study area in the recent decade. So, table number 5 summarizes the peak over threshold analysis for the rainfall over 7 stations. Each station shows the threshold that has been chosen, the number of exceedances above the threshold, and the estimated shape parameter and scale parameter of the generalized Pareto distribution model. The shape parameter for the rainfall, it indicates a heavy tail with positive values, suggesting that there is a potential for more extreme rainfall to happen in the future. And then the scale parameters is also larger, which suggests that the extremes, they are widely spread above the threshold, meaning they are variable. Now coming to the extreme value theory result of the extreme temperatures. The shape parameters in all the stations is negative, indicating a bound upper tail, suggesting limits of how hot it can get. The scale parameter is smaller, suggesting that the extreme temperatures, they cluster closer to the threshold, implying less variation. Now for the domestic water abstraction patterns over my study area. The graph for the wet season, it shows a slight upwards trend, peaking nearly at 1,800 meters squared on the highest day. Now coming to the dry season, water abstraction is also variable, but with a lower peak of about 1,600 meters squared, much less than that of the wet season. This might be attributed to the fact that during wet season, there was abundant water to be abstracted than in dry season. But dry season has a steeper rising trend of water abstraction, reflecting a growing trend and a growing demand in drier seasons. So now, determining the relationship between the climate variables and the domestic water abstraction, they were not statistically significant, meaning there was no relationship between the two. To wrap up, the Maruleng weather is telling a very interesting story. Rainfall shows no clear trend, but the temperature, especially the maximum, is showing a very significant increase. increase, signaling heat waves in our study area. Yet, surprisingly, the rainfall and temperature alone, they cannot be conclusive enough for us to conclude that for us to conclude that them alone are affecting domestic water abstraction in our study area, which is why in the future we have to look to other factors such as the institution, political influence, and others. Thank you.
Thank you very much, Nomcebo, for such a wonderful presentation. Another round of applause, please. Can we open this time for questions and answers? Questions and answers. While you are still thinking, perhaps let me come in first. Nomcebo, you were talking about the SDGs, which are more global. Can you share with us some of the NDP goals that your study seek to address?
For the conceptualization of the purpose of the scope of my study, I didn't really look at the NDP goals, but I based it on the SDG goals because they all affect the world. It's not just about the world, it's even about South Africa. We are part of the world, so that's why I based my study on it.
Thank you very much. Next time you must also try to localize it, because at a global level it might be flying up high. You find that we are actually not addressing the SDG goals, but we have our own that are formulated based on that. The NDP document is very important. Any other hand, guys, before we release Malaza? Aibo, you don't have a question? Okay, one last one. Why Maruleng and not Polokwane municipality? Can you justify why the choice of your study area?
The choice of my study area is because this is a follow-up from my honors study. I was also looking at the Blade River, and I've realized with these people from the Maruleng municipality, specifically the Mambejes Korolo, they abstract their water from the same Blade River that I studied before and was affected by sedimentation. So now I wanted to broaden my scope around that area and gain even more understanding.
Thank you very much, Nomcebo. Can we give you a round of applause, please?
There's a question.
Oh, sorry. The guy at the wall there, yeah.
Hi, I'm from Woodspruit under Maruleng municipality. So I want to ask, how does your study benefit the people from Maruleng?
My study benefits the people from Maruleng municipality in a sense that now they know that extreme weather events are indeed happening and they are affecting their water. But it's not only them alone that are affecting their water. They should also look at their political influence, which is what I've also added in my conclusion.
Can I have a follow-up question? We already experienced those conditions and we already know, so we want a solution on how we are going to get water because we already don't have water. So what solutions do you have for those people?
One of the solutions I have is that, okay, let me start by saying this. When I went for my data collection, I've realized that the plant is mostly controlled by people who don't have knowledge about water, but people who are from political structures. So my solution is we should start hiring people who studied water so that they can give us solutions that are water-based, not because, they are part of a political structure that is dominating in that time.
Thank you very much. I can see some hands. I think you will discuss that later. Can we give you a round of applause, please? You are safe. Thank you very much, colleagues and students, for giving us an ear. I think we really appreciate, and I enjoyed the presentation, and I guess you also enjoyed the presentation. The next session, I think Dr. Dwaba, my colleague, is going to chair it. Thank you very much, and thank you very much indeed.
Good morning, researchers and our professors. And let me welcome you to the next session and thank Dr. Mgovani for handing the baton. We've got 6 presenters that are lined up for this session, and the rules and procedure as they were outlined by Dr. Mgwabana will apply, because when you look at the calendar, it says we should have started a long time ago, so we no longer have time. So on that note, allow me to call the next presenter, which is Diti Romafani. And Diti Romafani is going to share with us the research on comparative analysis of optimal growth and activity condition for sulfate-reducing bacteria in Kwezela coal and cheesy copper mine waste water. Let's welcome her.
Water is a driving force of nature, yet when it is poisoned, it becomes a threat to life in earth. Greetings, my name is Diti Romafani, and today I'll be taking you through my master's study, which is titled The Comparative Analysis of Optimal Growth and Activity Condition for Sulfate-Reducing Bacteria in order to test their effectiveness in treating Kwezela coal and Chesi copper mine wastewater. This is my presentation outline. So mining industry, which is a fundamental pillar for global economic development, faces an unavoidable challenges of mine wastewater. This mine wastewater, also known as acid mine drainage, it's one of the most pressing environmental challenges that is faced both in South Africa and France, associated with coal and metal mining activities. So this acid mine drainage is characterized by elevated levels of dissolved heavy metal, such as iron, magnesium, and many more, and also highly acidic wastewater with a pH that ranges from 2 to 3.5. So figure 1 is a clear representation of how AMD looks like, and it is formed when pyrite minerals react with water, oxygen, and bacteria such as ferro-oxidizing bacteria to form dissolved iron and sulfuric acid. Then some of this iron can then now precipitate and form the red yellowish sediments as shown in the figure. So one might ask, why the use of biological sulfate-reducing bacteria? This is because the current conventional treatment methods which are used to treat acid mine drainage have been proven to be inadequate in reducing sulfate concentration, and they also produce large amount of sludge, therefore causing secondary pollution. So now we are trying to divert into finding a natural approach that is not only cost-effective, it is environmentally friendly, easier to operate, and also produce less amount of sludge. And that's where I come in with a solution of using sulfate-reducing bacteria. So figure 2 shows the mechanism of how the sulfate-reducing bacteria uses their ability to convert sulfate into sulfide, which is less toxic, and precipitate metals in order to neutralize acidity. So what is the problem? So the problem is that the discharge of this untreated and highly acidic mining wastewater continues to grow and damage our environment. The release of this acidic water with high concentration of heavy metals, which I've mentioned, into the surrounding ecosystem lead to severe ecological degradation, soil contamination, and also have a negative effect on the surface and groundwater pollution. So some of the negative effects that we've recorded is the death of many fish species, which was caused by the spillage of mine wastewater in Kwezela into the neighboring river, therefore causing the loss of biodiversity. And another issue is the leaching of minerals such as malachite, which then caused soil pollution in Chelsea-less copper mine. So the aim of the study is to compare analysis of optimal growth and activity condition for sulfate-reducing bacteria in order to treat Chelsea and Kwezela AMD. So the objective were to investigate the optimal condition for the growth and activity of these sulfate-reducing bacteria in order to eliminate or reduce sulfate concentration and increase the pH of the mine wastewater. So this study was conducted using AMD from Kwezela, which is located in Mpumalanga province, and Chelsea-less copper mine, which is located in France. And these two mines have demonstrated different geological and chemical diversity. So for AMD characterization, in order to determine the pH concentration and electrical conductivity, we used a multi-parameter. And to determine the metal concentration that were present, we used ICP-OES. So sulfate kits with the spectrophotometer were used to determine sulfate concentration in the water. And equation 2 shown, it was used to calculate sulfate reduction in order to test the effectiveness of this SRB to reduce sulfate concentration. So the inoculum that was used in this experiment was collected from the French Geological Survey operating SRB bioreactor system, and each batch bottles, as shown in figure 6, was supplemented with yeast extract for nutrients, glycerol as a source of carbon, and finally hydrogen gas as a source of electron. And they were incubated at a temperature of 25 degrees under strict anaerobic conditions. So in order to analyze for the sulfate communities that are present in the inoculum, we used the protocol shown in figure 7, where the collected samples were filtered using sterile filters and then stored in a refrigerator for a period of 24 hours. Then after we did DNA extraction, then PCR amplification using the purified DNA. And then after, in order to prepare for sequencing so that when we can be able to have a data that is processed in order to plot the bacterial relative abundance graph. So the ICP-OS results showed that Kwezela AMD was highly acidic with a pH of around 2.4 when compared to the Chelsea. And when we initially used this AMD treatment using the SRB, the active SRBs, almost all the bacteria died. Therefore, we had to pre-adjust our AMD using sodium hydroxide to a pH of 4.5, which has been proven to be an ideal pH for SRB growth and precipitate some of the metals. So moving on to the results that are shown below. So in order to understand the relationship between pH changes and also sulfate evolution, we observed different treatment efficiencies. As you can see by the Chelsea AMD treatment shown in figure 8A, we managed to achieve around 93% of sulfate reduction and effectively neutralize the pH from 6 to 8, therefore improving the water quality for environmental standard discharge. On the other hand, Kwezela AMD, the sulfate reduction was not that effective, as we managed to only reduce around 50% of sulfate, but the pH was high within the 70% of inoculation. This proves that in this inoculum, there was incomplete bioremediation. So for bacterial community, the most dominated bacteria that we recorded were Desulfosporocinus and Desulfovibrio. And these specific genera are known for their sulfate-reducing capabilities, and they thrive well under acidic conditions. So the image also in B, it also shows that the inoculum was not only dominated by one specific kind of bacteria, but it was dominated by a diversity of bacteria. So when it comes to the Kwezela AMD, we also observed a similar trend of bacteria. As you can see that it was also dominated by Desulfosporocinus, as shown in the graph, and also the observation in B. But it was only dominated by one specific, based on the bars that were recorded. This is why we observed less sulfate reduction, meaning that the conversion of sulfate to sulfide was incomplete, as the dominance was only given to one specific bacteria. So in conclusion, we can say that sulfate-reducing bacteria is indeed a procedure that we can install to effectively treat our AMD, as we manage to effectively grow a diversity of community of bacteria and achieved around 93% of sulfate reduction. Not only that, but around 99% of iron removal in chassis. And even though we maintain the same condition in both the inoculums, for Kwezela, we only monitored a moderate, as I mentioned, 50%, but iron removal was at 96%. This study therefore supports the Sustainable Development Goal of 6.3, which aims to improve water quality by reducing pollution and enhancing treatment of wastewater, thereby contributing to sustainable and effective acid mine management. So for future use, we can integrate SRB bioremediation with advanced metal recovery technologies, such as freeze crystallization, desalination, in order to treat sulfate and recover valuable minerals that can be readily commercialized into the market and improve the economy of South Africa. So I would like to express my appreciation to my supervisors, Prof. Mabel and Dr. Lebelo, and heartfelt for EWCTA for financial support, and finally BRGM for giving me the facilities with the laboratories. These are the references used. Thank you.
Thank you very much, Didiro, for the wonderful presentation. Now we open the floor for questions and comments. Can you take questions from the floor? One person, one question, please.
Hi, Didiro, how are you?
I'm good, and how are you?
I'm okay. They've just dropped me with one question. I'll ask you one question. Here you are using the bacteria.
Yes.
It's a question and it's a comment or maybe suggestion. Do you know the term bioflocculation?
The term biofiltration.
Bioflocculation. When you are using a term bioflocculant, have you ever considered to use that bacteria because it's a genera of various bacteria?
Even this one, it's a genera of different kind of bacteria. That's just that only they focus on sulfate reduction, but also we can see that they also reduce some of the metals that were present and at a very high rate.
But it does have negative and positives, isn't it? On suggestion, maybe in future, take those bacteria, check their bioflocculation activity, instead of using the bacteria itself. Try to use the bioflocculant as a product. Maybe check what will happen.
Okay, can I add something? in this
Yes.
study. So this is the batch mode of my experiments. This is whereby we're just testing it in a very small bottle in order to remove it into a bigger bioreactor, whereby there was a material that was used in the bioreactor to help for the biofilm formation in the bioreactor. So in that, we monitored a very effective compared to when it comes to the batch, because in the batch there was no material that was like keeping the communities like intact. Yeah.
Okay, that's why Gary in future maybe try that.
Yeah.
All right, thanks.
Thank you.
Thank you. I just want to ask you something. Have you ever checked the precipitation of sulfate, ranging the pH? Because I'm sure that it will precipitate at the pH between 4.5 to 5. And in your case, you adjusted your pH because of your bacteria were dying at pH 2. You had to adjust using sodium hydroxide, adjusting your pH to 4.7, isn't it?
Yes.
So I'm worried, have you ever checked the precipitation of sulfate without using any bacteria? Because you might find that you are assuming that the bacteria, it is the one that reduced your sulfate, only to find that precipitation took place there. Thank you.
Thank you. I think I didn't like fully get your question, or where is it directed to? But what I would say is, yeah, it's a good suggestion. But also, I think the main reason why we didn't monitor like a high increase in sulfate reduction was that there was accumulation of, because remember, when you use sulfate reducing bacteria to treat AMD, so the sulfate is converted into sulfide, which then the sulfide then react with the dissolved metals that were present to form metal sulfide. So the accumulation of that metal sulfide, it's the main limiting factor of why the reduction of sulfate was not high in this condition. Because the batch bottles were kept in an anaerobic condition whereby there's no entry of air, and even when you do sampling, it's, yeah.
Thank you very much. I think we can release it. And our next speaker is Tato Seleka. Research topic: analysis of smallholder goat farmers' perception and adaptation strategies on effects of climate change towards production in the Palale local municipality, Limpopo province. Can she hear up as she starts?
Good day, everyone. My name is Tato Seleka, and I'm doing my second year master's. I'm here to present my ongoing study titled Analysis of Smallholder Goat Farmers' Perceptions and Adaptation Strategies on Effects of Climate Change Towards Production in the Palale local municipality, Limpopo province. Animal production is negatively impacted by changing climates either directly or indirectly. The unfavorable weather often gets worse and increases the volatility of animal productivity. Extreme weather events will occur more frequently in addition to the previously changing patterns of rainfall and rising temperatures. Studies conducted on effects of climate change and adaptation strategies in agriculture have focused more on crop production. However, there's a gap that remains on effects of on the perceptions and adaptation strategies on animal production, particularly goat farming. The aim of the study was to analyze the smallholder goat farmers' perceptions of effects of climate change towards production and their adaptation strategies in the local municipality. The objectives of the study was to profile the socio-economic characteristics of smallholder goat farmers, to determine smallholder goat farmers' perceptions, and to analyze factors that influence smallholder goat farmers' perceptions, and finally to identify and describe smallholder goat farmers' adaptive strategies to climate change. The hypotheses of the study were derived from the above objectives. The study was conducted in the Palaleo local municipality under 4 villages. And the study used a multi-stage sampling to select 138 farmers. It also used primary data through a structured questionnaire. Then analytical techniques. Descriptive statistics was used to address the first and fourth objectives. A Likert scale was used to collect information about smallholder goat farmers' perceptions of effects of climate change on their production, thus addressing second objective. The third objective was addressed using binary logistic regression model. And the results show that most of the farmers were old and experienced. Again, most of the farmers were male, married, and unemployed. None of the farmers indicated to have access to credit extension services or either being familiar with the use of technology in their production. Only 5 members are involved in agri membership groups, while 2 of them have received agricultural training. Regarding education level, large percentage of the farmers had secondary education, and also they survived through the monthly income of between 1,000 and 5,000 rand. Here are the experiences and on effects of climate change and their adaptation strategies. All of the farmers indicated to have experienced drought and heat, and a large percentage indicated to have experienced ticks. However, the experience on floods were rare. Their adaptation strategies for drought, most farmers relied on feed supplements, drought-resistant crops, and destocking. For heat stress, they provided shades, provided cool drinking water. Tick infections, they used dipping and spraying methods. However, those who indicated that they have nothing indicate that the farmers lack resources and knowledge. The study used a 5-point Likert scale to assess the perceptions of the farmers, with a mean of 3.17 as a cutoff and overall mean. Although the result shows that the farmers really perceive climate change, although their awareness differs. State of production had a low score, which means that farmers really do know that, do agree that their production had declined, and rainfall pattern had a larger score, which shows that farmers really can indicate that rainfall patterns do shift. Here is the percentage by age, gender, and education. The youth and the middle-aged farmers were the most group that perceived climate change, while the older farmers indicated that they do not perceive climate change. And for gender, female farmers perceive the effects of climate change more than male farmers. Or education level, those with higher education perceive more climate change effects more than those with less education level. And the table shows the results for binary logistic regression model. And education level was significant at 5% level, and it's actually reduced the likelihood of perceptions. Farmers with more education were about 80% less likely to perceive climate change as a threat. This could be because farmers often diversify their income or rely less on goats, so they don't feel the heat of climate change. And married farmers, on the other hand, were two and a half more likely to recognize climate change impacts. This may be linked to the family responsibilities, the need to secure household food and income, making them more sensitive to production risks. Oh, sorry. And for employment reduced the likelihood of farmers perceiving climate change. This could be because of farm employment, like farmers who are employed out of the farm do not rely more on the generation of income from the goats. Agric membership, although it was marginally significant, it had a greater effect because farmers were more likely, about 10 times more likely to perceive climate change. This means that farmers in groups or peer socials are perceiving climate change positively. In conclusion, we can say that farmers do really perceive climate change. Therefore, the proposed hypothesis, which said no difference in perceptions and no factors influencing perceptions, they are rejected. For recommendations, this study recommends expanding extension and training programs to farmers, improving access to adaptation resources, enhancing early warning systems, more like whether information should be available, reliable, and farmer-friendly, promoting inclusive policy support, more like local government should integrate farmers with access to credit, subsidy, and insurance programs. I'd like to acknowledge the following: my supervisors, Lepala Local Municipality Goat Farmers, and National Research Foundation for the funds. These are the references used for the study. Thank you.
Thank you very much, Tato, for the wonderful presentation. Let's give her a hand of applause because she made it to the finish line. Now we can open for questions and comments from the floor. 1, 2, 3, sure, 4. Let's try to be as short as possible and straight to the point.
Okay, thank you very much. Mine is very simple. I just want to know what informed you to focus on Lephalale local municipality goat farmers? And my second question, Koro, which type of farmers did you, oh, sorry, thank you.
Thank you for that question. Well, I would say, personally, I'm from Lephalale, and I have seen the challenges that the farmers are facing like almost every day. So this study is more like a continuation from my honor study, where I was analyzing the factors that determines the head size of the good farmers. So on this case, I'm now focusing on climate change perceptions to find out whether they perceive or not the climate change effects.
Okay, thank you. My question is, okay, as you were trying to justify your study, you said that a lot of studies that work on climate change, they usually focus on crop production instead of, therefore there's a lack of study in animal science. So my question is, what is the impact of this climate change on a goat? Think of it in this way. Let's say you have a goat here, and then there's climate change here. Does it affect it directly or it affect it indirectly? In this sense, if right now it affects crop production, which the goats rely on crops or the plants themselves to feed, then it affects it indirectly, not directly. Therefore, justifies why, when you look at climate change, we mainly focus on crop production instead of animal production. Thank you.
Was that a question or a statement, a comment?
The question is, what is the impact of climate change on a goat? You have a goat here, and then there's a climate change. Tell me what's the impact there?
Well, you have mentioned that it affects it indirectly. I agree, and I also say it affects it directly. In what sense? Well, let's say higher rainfall, high rainfalls can cause the goats to develop the tick infections, because the food when they on the wetter conditions they tend to develop the ticks, and again the heat stress can also affect goat farmers. It can affect their appetite. Yes.
Hi, thank you for the beautiful presentation. Mine was on the recommendations. There was some point where you recommended training and also spoke about the weather information being available. So my argument would be that there is some training that is provided, but my question to you would be, how do you think, what would you recommend to make sure that those trainings and stuff are actually reaches those communities? Because I also was under the impression that they are not provided to local until I read the government reports, and you find that they actually list certain trainings and they give people livestock and all of the shebang. And then the problem is that it never, or our communities don't have access to that information. So my question would be to you, how can it reach,
I think the trainings maybe from the government or local government can reach the farmers through extension services. Like for instance, on this study, all the farmers indicated that they don't have access to extension services. So I think that is the reason why they lack access to more things.
Okay, thank you. I heard you talking about the multi-stage something under your sample size that you used 138 farmers. So I would like you to briefly explain how the multi-stage method works using your example of 138 farmers.
Firstly, under multi-stage sampling, I would say 3 sampling techniques were used. firstly being purposive sampling. Purposive sampling was used in order to select Lephalale local municipality out of all municipalities in the Waterberg region. Then the second one is convenience sampling. The convenience sampling was used to consider the selection of all the villages under Lephalale. Then, based on the availability of the farmers. Then the third one is simple random sampling. That one was used to select the farmers in selected villages to reach the targeted sample size.
Thank you very much. Let's clap hands for her courage and the experience learned today. Ma'am, you are released. The next presenter is Johanna Mashita. And the title is Analyzing Challenges Faced by Smallholder Poultry Farmers in Selling Eggs during avian influenza outbreak in 2023, a case of Sekhukhune District Municipality.
Good day, everyone. My name is Mashfana Johanna, second year Master of Agriculture Management student, and I'm here to present my current study titled Analyzing Challenges Faced by Smallholder Poultry Farmers Selling Eggs During Avian Influenza Outbreak in 2023, a case of Sekhukhune District Municipality, supervised by Prof. MP Signolo, co-supervisor Prof. A. Beled. In early April 2023, South Africa experienced an outbreak of avian influenza, which is a highly contagious viral disease mainly affecting poultry industry, with smallholder farmers producing layers being mostly affected. It led to changes in consumers' behavior, reduction in egg sales or profit, business shutting down or retrenching their workers. Hence, most of the smallholder farmers resorted to setting higher prices to cover up for the increase in cost of production, brought by increase in feeds or introduction of biosecurity measures. Smallholder farmers in regions like Sekhukhune are characterized by limited access to resources, infrastructures, and support services. Additionally, avian influenza led to culling of millions of chickens in the country, which led to a 30% drop in hatching eggs production. It posed trade restrictions and bans on poultry products, making market access difficult for smallholder farmers with limited alternative sources of income. This worsened the circumstances of these farmers as they were already battling with the effects of load shedding. Hence, the study is aimed at contributing to the existing literature on smallholder farming and disease management by providing context on the specific challenges faced by these farmers in Sekhukhune District Municipality. The study was aimed at analyzing challenges faced by smallholder poultry farmers selling eggs during avian influenza outbreak in 2023 in Sekhukhune District Municipality. Objectives: to profile the socio-economic characteristics of smallholder poultry farmers in selling eggs in Sekhukhune District Municipality, to identify and describe challenges faced by smallholder egg farmers in Sekhukhune District Municipality during avian influenza outbreak in 2023, to analyze factors influencing the number of challenges faced by smallholder poultry farmers in selling of eggs during avian influenza outbreak in 2023 in the study area, to assess whether there is a difference in the strategies employed by male and female smallholder poultry farmers to cope with the challenges faced selling eggs during avian influenza outbreak in 2023 in the study area. The objectives were developed from the hypotheses were developed from the last two objectives of the study. The study was conducted at Sekhukhune District Municipality. It used primary data to collected from 73 respondents using combination of purposive and snowball sampling procedures, which is a non-probability procedure that is used to select an individual for inclusion in the sample based on specific characteristics, while the snowball is used to identify initial respondents when the target population is hard to reach or access. In this study, the combination was used to first identify the primary smallholder farmers based on characteristics that they were selling eggs in 2023, and at least have 5 years selling eggs, which allow them to have knowledge of network about other farmers, allowing them to recruit them or mention them. And this process was repeated until the study reached a total of 73. The following analytical techniques were used in the study to address the objectives of the study. Descriptive to address the first and the second objective of the study, while the chi-square was used to address the differences in the strategy employed by male and female smallholder farmers. Binary logistic regression model was used to address the third objective of the study. From the result, the youngest and the oldest were 22 and 76 years old with a mean of 41 years, indicating that most of these respondents were of mature age or were older farmers, and they've been selling eggs for at least 5 years, indicated by the mean of 5 years. For the gender, only 44% of them were females, and most of them were not working. The marital status indicated that most of them were single, followed by married with 40% and only 1% divorced. From the number of challenges faced by these farmers, majority, which is 58%, faced more challenges, as most of them operate on limited resources. From the places where these farmers sell their eggs, direct from the market was dominating with 34%, followed by local source with 21%. From the follow-up questions, the farmers indicated that it is easy to sell direct from the market and it saves transport costs of moving the eggs from one place to the other. From this table here, 61% of them had a monthly income of more than 3.5, while only 33% received training regarding outbreak of diseases, and only 19% had access to agricultural extension services. When it comes to the strategies implemented by these farmers, sanitation and hygiene was only implemented by 49%, while strategies such as monitoring was only implemented by 16% of these farmers. From this, these are some of the challenges faced by these farmers, and 74% of them faced decrease in egg sales, while 82% had reduction in profits, and 70% faced reduction in demand, and 67% had changes in consumers' behavior, which is maybe a situation where consumers grow fear of buying eggs that might be contaminated with avian influenza. These are the chi-square analysis results from the strategies implemented by these farmers. And sanitation had a p-value of 0.713, while PPE is 0.191, and financial support was 0.435. From these results, all the strategies have a significant value that is greater than 0.05, indicating that both genders implemented similar strategies. These are the binary logistic regression model results. Age increases the likelihood of these farmers facing more challenges, and it was statistically significant at 10% level. Gender also increases the probability of these farmers facing more challenges, and it is significant at 5% level. Marital status decreases the probability of these farmers facing more challenges, and it was found to be statistically significant at 10% level. Education level decreases the likelihood of these farmers facing more challenges, and it was statistically significant at 10% level. Training regarding outbreak of diseases indicated a negative association with the number of challenges, and it was significant at 5% level, indicating that when farmers have this type of knowledge, the number of challenges they face were likely to decrease. Poultry farming knowledge decreases the number of challenges, decreases the likelihood of these farmers facing more challenges, and it was significant at 10% level. Vaccinations indicated a positive association with the number of challenges, and it was significant at 5% level, indicating that when farmers implemented these strategies, the number of challenges they face were likely to increase. This might be because if they are overdosed or not applied correctly, they might lead to high mortality rates, therefore further challenges. But however, if they are applied effectively, so if they are applied in correct amount, they are very effective in preventing further challenges. Financial support increases the likelihood of these farmers facing more challenges, and it was significant at 10% level. For this financial support, most of the time it reaches farmers who have already experienced the challenges, or in the world we live in today in South Africa, corruption is a lot, so it might go to those people that they know rather than those people who need help. Reduction in egg sales increases the likelihood of these farmers facing more challenges, and it was significant at 5% level. Difficulties in accessing the market increases the likelihood of these farmers facing more challenges, and it was significant at 5% level. Changes in consumers' behavior also increases the probability of these farmers facing more challenges, and it was statistically significant at 5% level. Other challenges that were not mentioned in the study, such as increase in fish, indicated a positive association with the number of challenges, and it was statistically significant at 5% level, indicating that when farmers face these other challenges, the number of challenges they faced were likely to increase. Okay, the study concludes as follows. It fails to reject the second hypothesis of the study, as all the p-values of the strategies are greater than a threshold of 0.05, indicating that there are no differences in the strategies employed by male and female smallholder farmers. And the study rejects the first hypothesis of the study, as factors such as age, education level, reduction in excels, difficulties in accessing the market, other challenges not mentioned in the study, financial support, were found to influence the number of challenges faced by these farmers. Based on the main findings of the study recommends the following to the Department of Agriculture, agricultural stakeholders, education, and policymakers. Establishment of gender-specific programs for both male and female farmers, encouragement and promotion of education on poultry farming, but not only limited to that, also outbreak of diseases, so all farmers, regardless of their age, gender, marital status, and for them to provide vaccinations, which could be done by sending mobile units to rural areas such as Kukune to help these farmers easily access them, and improvement of market access infrastructures or creation of digital platforms to help connect consumers, farmers directly to consumers, promotion of public awareness campaigns to help regulate information during outbreaks and prevent public panics, and establishment of emergency funds and farm support services to all farmers. I would like to acknowledge the following God. I thank him for giving me the strength to go through the study and carry it out. To my supervisors, Prof. Senior and Prof. Beledi, thank you for your contribution in my study. To the community of Skukune District Municipality, thank you for opening your doors for me during my data collection process. May God bless you. Thank you. These are some.
Thank you very much, Johanna. Let's give her an appreciation of hand again. I'm going to take only 2 hands. Please, let's try to be specific. Can I take those 2 hands? This one there. And one here.
Okay, thank you for the nice presentation. My question is just out of curiosity. I see you used 2 different probability levels, which is 5 and 10%. What informed you? Thank you.
Oh, I use SPSS. So from the SPSS, they were automatically significant at 5% and 10%. It was unfortunate. I don't know if it was unfortunate, but then there were no 1% significant levels. I don't know if I answered your question, but I think I did.
Thank you very much. The other hand is here. Unfortunately, we are only taking 2 in the interest of time.
Okay, thank you. I heard you talking about the challenges on your background that these people got during the influenza problem. So I would like to know what is the problem that your study was trying to solve? What it is that your study solved, the problem statement specifically?
Okay, on the problem statement, it was like the challenges that were posed by the disease to the smallholder farmers. And in conducting this study, I've come to the realization that even though there might have been several studies on this disease, but then the information most of the time reaches, I would say, wrong audience, not the smallholder farmers. Because you can see the pattern is still the same. The farmers are still experiencing the similar challenges, although there have been researchers. So the information only reaches people like us, the researchers only. So it's not really effective. So in my study, in doing this research, I am planning to go back to these farmers where I've collected data and report on them so that they can get in this information, because I cannot expect them to go and research on this. And most of them, they are older farmers. I'm sure you saw in the table of descriptive. Thank you.
Thank you very much, Joanna. We can give her a hand as she descends the stage. And our next presenter is Tritin Gobeni. Topic, germination and growth response of Bambara groundnut to different concentration of seaweed extract and priming times, in priming times. Let's welcome him. Let's welcome him even though he's saying he's super ready.
Can you please come and help me with to adjust the mic? Thank you. Yeah, it's fine, thank you. Thank you, Chair. I'm Gobeniti, and I'll be taking you through a portion of my current study titled Germination and Growth Response of Bambara Groundnut to different concentrations of seaweed extract and priming times under the supervision of Dr. Mlatudi and Dr. Mangena. So Bambara groundnut is an underutilized legume crop that is native to Africa, which is widely cultivated by the small-scale farmers. So, however, Bambara groundnut, it has a slow germination, which is caused by its thick and hard seed coat, and its slow germination, it tends to lead to extended harvesting times. So, Bambara groundnut is a good crop which provides many advantages, making it a valuable crop. So, for instance, Bambara groundnut, it has the ability to withstand drought conditions, and in nutrient composition, it has approximately 65% of the carbohydrate, the 19% and 6.5% of the protein, and the fat, making it a balanced food source, and which helps to combat malnutrition. So with the seaweed extract, plant growth stimulants that I use as an eco-friendly nutrient source in an sustainable and organic agricultural practices. So these seaweed extract, they contain a variety of plant growth hormones, which includes the auxins, the cytokinins, and the abscisic acids, and the other phytohormones that are not mentioned here. So this seaweed, it can be used as a seed treatment, fall sprayer, and soil application. So with the seed priming is a pre-germination technique which involves soaking seeds in liquid substances such as water or other biostimulants, including the seaweed extract. So soaking Bambara seeds in seaweed extract, it presents a promising solution to overcome the difficulties of the slow germination in the growth of Bambara. So the aim of this experiment, it was to determine the effect of the seaweed extract and the priming time on germination and growth of the Bambara. And it was hypothesized that the seaweed extract and the priming time, it has no effect on the germination and the growth of this crop. And then this study was carried out at the University of Limpopo Department of Biodiversity in the glass house, and it was arranged in a factorial arrangement in an RCBD with 9 replications. And then it includes 2 factors, which are suet extract and priming times, with suets having the 4 levels, including the distilled water, the 0.15%, the 0.60%, and 1.20. And then with the priming times, it has 3 levels, including the 3 and 6 and 12-hour priming durations. And then these are the data that was collected in this experiment. So the collected data was then subjected to ANOVA using statistics 10, with the means being separated using HSD at P is less or equal to 0.05. So going forward here, I'll be presenting the parameters that were found to be significant. Okay, so here you are looking at the effect of suet extract and priming times on days to 50% germination. So looking on the graph presented to you here, we are looking at all the seeds that were treated with the suet extract, including the 0.5, 0.601, and 1.20, they significantly reduced the days to germination. And looking above the graph, we see the priming duration, where the priming duration, it reduced the days to 50% of the germination. Okay, with their interaction, we can see that most seeds that were treated with different concentration of the suet performed better compared to the seeds that were treated with distilled water. So to be specific, the 1.20% where the seeds were primed for 12 hours at the corner there with the black bar, it shows that this treatment was fast enough for the seeds to reach the 50% of the germination, which was followed by the treatments that were treated with 0.15 for 6 hours. So going to the plant height, once again we see that the suet extract, all the 3 concentrations, the 0.15, 0.60, 1.20, they did improve the height of this crop compared to the seeds that were treated with the distilled water. Looking at the priming duration, the 6-hour priming duration, it increased the height of the Bambara groundnut. With their interaction, we can see still here that the suet concentration, they significantly improved the height of the Bambara groundnut with with the seeds that were primed with 0.60% at the 6-hour priming duration, being the highest or achieving the highest plant height compared to the seeds that were primed with distilled water for the 3 hours. So moving forward to the interaction on the number of leaves, we can observe that the seeds that were treated with 0.15% for 12 hours, they significantly increase the number of leaves compared to all the seeds that were treated with distilled water for 3, 6, and the 12-hour priming durations. So looking at the interaction on chlorophyll content, we can see that the seeds that were treated with the 0.60% for 3 hours significantly increase the chlorophyll content compared to the seeds that were treated with the distilled water, including all the priming durations, the 3, 6, and the 12 hours. So the reduced days to germination that was observed can be attributed to the phytohormones that are present in the suet extract, and also the priming time, which sped up the cell activity and reduced the time that was needed to reach germination. And with the priming time, it contributed by allowing the enough time for the seeds to absorb the phytohormones and the nutrients that are available in the seaweed extract. So as seen that the seeds that were primed with 0.15% and 60% for 3 and 6 hours, they showed the higher effect on chlorophyll content, which can be attributed to the phytohormone cytokinin that is known to delay the chlorophyll breakdown and enhance the formation of the chloroplast. So with that being said, is that as the increase is being observed in chlorophyll, it also improves the photosynthesis, supplying more energy and improve the crop overall. So as a result, the plants managed to produce a greater number of leaves, which exhibited an increase in plant height, which is also can be attributed to the soil elongation that was caused by the increased levels of the these two phytohormones, the auxins and the gibberellins, that were also present in the seaweed extract. So the following parameters that was presented to you has been shown to be improved with the seaweed extract by previous studies. So in conclusion, I can conclude that soaking seeds with 0.5% or 0.6% at the priming duration of 3 and 6 hours, it can help to overcome the difficulties of germination and improve growth of Bambara groundnut. And I would like to acknowledge these two institutions, and special thanks to the Department of the Biodiversity for allowing me to use their resources. And these are the references that we used to compile this presentation, and I thank you.
Thank you very much, Sengobeni. Can we give him his applaud before we go to questions? Thank you very much. 2 hands. I'm going to take 2 hands. 1, 2. Unfortunately, this one I'm blue ticking it. So I'll only take those 2.
Hi, thank you for the presentation. My question, if I ask my question, in your presentation you said that in your study you used priming or the pre-soaking method. Why did you prime your seeds in seaweed extract instead of and not used the seaweed extract as a foliar application, like you explained that there are different methods to that seaweeds are applied as?
Thanks for the question. And as I've said in my presentation, that the problem of this crop is it's a slow germination. So in order to try to solve or to improve the germination of this crop is to start by soaking it into different concentrations of the seaweed and see which concentration performed better compared to the seeds that were used, that were treated with distilled water. So hence, as I mentioned, to improve the germination, I need to start by priming and observe which treatment improved the growth of the crop overall.
Hello. Okay, what I can ask, what is the difference between the seawater and the distilled water component, since it has the different effects on the leaves and the chlorophyll content and the plant height?
Okay, I didn't hear you well, but you're asking what is the difference between the distilled water and the seaweed extract. Okay, so is that the difference in the distilled water and the seaweed extract is that the seaweed extract contain nutrients and the phytohormones, as I've mentioned. So the distilled water is just a hydropriming, trying to soften the seed coat and immediately start the imbibition before planting the crop, the seed. So in terms of the difference in influencing the chlorophyll, is that with the seeds that are being treated with the seed water, the level of the phytohormones that are available, they increases compared to the natural hormones that are available in the seeds that were treated with this hard distilled water. So hence, the seeds that were treated with distilled water, with seaweed, significantly increased the chlorophyll, and with the chlorophyll being increased, it means that photosynthesis would also be improved, which is an energy source of the crop, complementing the leaves formation, the growth of the height, which contribute to the overall productivity of the crop. Thank you.
It's okay. Don't leave. He released himself. Thank you very much, Zengoveni. The next presenter is Koketso Boroko. And those questions that we blue tick them, please share with the presenters during networking sessions. They will help them. Koketso will be looking at geomorphometrical approach of soil erosion quantification in Lepellane catchment, Limpopo Province, South Africa. Thank you. Let's give him a hand before he starts.
Good day, ladies and gentlemen. My name is Rokoko Ketsokal. I'll be taking you through my part of my research. I'm doing first year of MSc in the Department of Geography and Environmental Studies. It's titled, I'm actually going to present a review. This was the topic of my study, but I'm going to be presenting the review of it. It's titled The Geomorphometric Soil Erosion Modeling in Sub-Saharan Africa, a systematic review of model applicability, validation gaps, and policy integration. This is my presentation outline. Soil erosion poses as a major environmental and socio-economic challenge, particularly in semi-arid regions like the Sub-Saharan Africa. It has the main drivers being climate variability, unsustainable farming, land use changes, and rugged topography. Now, this often leads to the loss of topsoil. It reduces agricultural productivity, sedimentation of water bodies, and also ecosystem degradation. However, despite its importance, erosion modeling in Sub-Saharan Africa faces key constraints. We see limited application of advanced geomorphometric and hybrid models, and even when models are applied, their outputs are rarely integrated into policy and land use planning. This reduces the accuracy of erosion risk assessments and weaken the environmental management procedures. The aim of this review is to systematically review the application, validation, and policy integration of geomorphometric soil erosion models in Sub-Saharan Africa. Now, to do this, we adopted a PRISMA methodology. It is a well-established framework for systematic reviews. It ensures a transparent process from identifying relevant literature, screening studies, assessing eligibility, and finally synthesizing the results. From data collection, we collected about 1,042 studies. From that, we excluded 607, which were conference proceedings, gray literature, extended abstracts, and editorial letters. And further, a screening process continued, whereby we fully excluded titles which were outside the geomorphometrical indices, and we were left with 108 studies which were considered for this study, and 108 of those underwent a thematic and bibliometric analysis in RStudio to produce these results. Now, we have different categories of this model. We have three categories, which is the empirical model, process-based models, and hybrid models. Now, under these empirical models, it consists of the commonly known ones, which is the Russell models, and the Ussel, and the Crims models, which these reviews uncovered that these models are very easy to use, hence they are mostly applied. They have low computational least, and they are best for broad estimates rather than precise results. But these models, they require local calibration, as these models are not, or were not originally developed within African context, so they need to be calibrated to suit the conditions of sub-Saharan Africa. Under process-based, we have the Kairos models, the web and USPED models, which these kind of models, they simulate real physical process models like the rainfall and runoff. But these models now, they require high resolution data and they require more computational lead. Under hybrid models, we have random forest models. We have Russell, which is the base of the soil erosion modeling. When combined with the AI technology, we have the random forest, which improved erosion prediction with an area curve of 0.89 in Iran. This was a study that was done by Tel M et al. In 2014. Now, we have Russell artificial neural network and GIS logistic projection models, which improved the accuracy of soil erosion mapping. This was a study that was done by Kosavi et al. At 2023. This is just a snapshot of what these models look like when they predict soil erosion. It ranges from high deposition areas until high erosion areas. So it accounts for both deposition and erosion of the soil. Now we have another critical driver of erosion, which is land use, land cover. Understanding how vegetation loss, farming intensity, and urbanization affects erosion risk is key to predicting and managing these erosion risks. I'm just going to highlight a few studies which were done under these to confirm the effect of the land use, land cover changes on soil erosion. There was a study that was done in Tangare, Ethiopia, by NISA, which they used land use, land cover classification from satellite images from 1986 to 2006. They used Russell modeling, and their key findings were that deforestation and expansion of agricultural land increases soil erosion. Another study that was done, now we bring it a bit closer home, in the Zambezi Basin River. It is where land use, land cover analysis with change detection techniques and soil loss estimation uncovered that urbanization and mining increased erosion in localized hotspot, and this was characterized or specific to their site. Now, if we look at this, this is the annual scientific production of papers that were produced from the year 2000 till 2024. We see a steady increase of these papers. If you focus more on 2015, this is when we started having a spike, and this was due to the release of satellite images such as the Sentinel-2, which made it easier and more less time consuming to do these erosion risks. And further, if you look from 2020, we see now as the highest spike of them all, and this was due to some advances in remote sensing and AI technologies. This is where also Google Earth Engine enabled large-scaling erosion analysis, so it allowed for researchers to predict soil erosion in large scales, and it was not even charged for that, so it had low economic requirements. And also around that time, there was when a satellite Landsat 9 was released, which had higher spatial and temporal resolution and allowed for precise erosion risk mapping. If we move to the regional gaps and policy implications, now this highlights that sub-Saharan Africa catchments are underrepresented in global terrain-based soil erosion. As we look at this map that was generated through the RStudio and thematic analysis, it highlights countries that are leading in such researches, and they have more knowledge in terms of how these models are applied. And these models, as I said before, they are originally from the Europeans, and they are not so specific to sub-Saharan Africa. Now, there's a localization need, which means that this model needs to be more content-specific calibration and field validation to suit local biophysical conditions as we bring it back now to the sub-Saharan Africa. Implementation barrier. Now, the spatial erosion data often stays in academia due to institutional fragmentation and poor cross-sector communication between academia and the policy implementers. In conclusion, geomorphometric models are powerful and evolving tools for soil erosion assessment. Their potential in sub-Saharan Africa is significant, but it remains underutilized due to validation gaps, data scarcity, and poor integration. Now, bridging the gap between model development and practical application is paramount. With proper adaptation, these models can guide sustainable land management, climate resilience, and soil conservation. Now, after this review, this study recommends that we should improve the model applicability in sub-Saharan Africa in a sense that we should adapt these models to sub-Saharan Africa's semi-arid condition using region-specific parameters such as the rainfall intensity and the soil erosivity factors. We should integrate these models into policies. The spatial development frameworks, environmental impact assessments, and land use, land cover plannings, they should embed the results which are from these researches that are done under this topic or under the soil erosion theme. We should establish validation networks. We have UAVs, LiDAR sensors, and field plot. So these offer a more precise measurement as it gives images with higher resolution. But the problem is that now these UAVs and LiDAR sensors imageries, they are not free and they are very expensive. So if we create a database whereby we can make this such images available to researchers, it would also help in a sense that it would promote more research that will be done in this field. We should also foster international collaboration. We should have open access to geospatial infrastructure in a sense that information is able to be shared in between countries to better address this. I would like to acknowledge the Department of Geography and Environmental Studies, the Center of Global Change, and Arid Region Water Research Center, University of Solplace. These are my references. Thank you.
Thank you very much. Thank you very much for the wonderful presentation, Koketso. Can we applaud him as we take questions? Can I have the 2 questions? Hands? 1, 2. Yes.
Hi, that was a very nice and comprehensive review. Congratulations. I only have like 1 question. I had 2, but I'm going to give you 1. On the objective, you mentioned something about the validation of the models, because you said applicability, validation, and policy application, right? I wanted to say that during your review, what current validation processes are there, and what's lacking with them?
Okay. The current validation methods that are there that we currently use, the first one is the field validation. I think there's no better validation methods that actually beats you actually going to the field and actually observing that whatever we are predicting on this map is actually happening on the ground, right? The second ones are these higher resolution imageries or satellite, right, which will provide us with advantages in a sense that it will reduce time, in a sense that we won't have to use transport to go there, you won't require to book or those kind of things, those kind of logistics. So hence we opt for those high resolution images because they also offer real-time monitoring of the situation. Thank you.
Thank you for your presentation. Mine, I think, is a clarity. I just want clarity. Like when you are conducting your systematic reviews, do you only rely on your Prisma flow chart to avoid biasness and enhance your precision of your outcomes? Of there are other like methods that you use to improve your precision of the outcomes and unbiasedness?
Yes, for this one we relied on the Prisma the Prisma method. As I said, it is a well-established framework, and for systematic reviews, it also offers a chance for other reviews to be based on it. It's not only limited to you creating your study on it. It allows for also future researchers to also use this as a filtering process for the selection of their literature.
Thank you very much, Koketso. Can we give him a round of applause for release? And the next presenter, according to our program time, I think we are still on time. I'm not talking about your real time, talking about the program. Our next presenter is Raisibe Mahlo. The topic, it's a systematic review as well, the selection, signatures, and application approaches on identifying candidate genes using the whole genome data in dairy cattle. Let's give a round of applause, please.
Greetings, my name is Matlo Raisibe, a second-year master student, and I'll be presenting a systematic review on the selection signatures and application approaches on identification of candidate genes using the whole genome data in dairy cattle. So this is how my presentation outline is looking like. So with the dairy cattle, they differ genetically due to both natural and artificial selection. And with this selection, it creates unique patterns in the genome, and that are those referred to as the selection signatures. Well, with this study, the objective was to systematically review published articles on selection signatures in dairy cattle. Several studies have been reported on the selection signatures in dairy cattle. However, according to the best of our knowledge, there is no comprehensive systematic review that exists. Therefore, this study is going to provide insights into the genetic mechanisms of dairy cattle. So the population exposure and outcome framework was followed to ensure that relevant articles were followed. Google Scholar, PubMed, Web of Science, and ScienceDirect, these are the databases that were used. Studies on dairy cattle, genome-wide data, and selection signatures were included in this study. However, the non-dairy breeds, non-genomic studies, and also reviews were excluded. Academic integrity was followed to ensure that no plagiarism was followed and data manipulation. So after the literature search from the aforementioned databases, 164 articles were retrieved, and after the eligible screening, 27 articles were included in this systematic review from the year 2010 until 2025. So figure 2 presents the world map showing the distribution of institutions by country for the 27 included articles. So from this map, it is evident that most of these articles are originating from Asia and Europe, suggesting that the temperate breeds were used, which is our host in, meaning that Africa has been underrepresented and the tropical breeds and smallholder dairy breeds were underrepresented as well. Now we are on figure 3, which present the genotyping platforms. These are the platforms that provide genotype data. So Illumina bovine SNP was reported in more than half of the reported studies, followed by the whole genome sequencing, genomic profiler, RAD sequencing. However, this Illumina bovine SNP, it is quite biased because instead of genotyping the entire genome, it genotypes a portion of the genome. However, the whole genome sequencing, it is able to genotype the entire genome, which means it is better than the Illumina bovine SNP chip. Now we are on the statistical methods that are used to detect the genomic selection signatures. So from this graph, it is evident that the fixation index, which is the first graph, it is shown that it has been reported in most articles, followed by the integrated haplotype score and the cross-population extended haplotype homozygosity. However, these statistical methods, they are also biased because they only select recent selection signatures, forgetting about older selection signatures which are still valuable in the future. So from this graph, the RSB, which is the relative strand bias, it is able to select both the recent and the older selection signatures. Figure 5 presents the most reportedly candidate genes. So after selection of selection signatures, this first gene, the PLAG1, was the most reportedly candidate gene, followed by, which is related to milk production, followed by a gene related to adaptation, and then the 4 consecutive genes that are related to immunity, and growth. After that, we are having genes that are related to conformation, to mammary gland functions, and reproduction. So now this is figure 6, which present traits that are identified from the selection signatures. These are traits that are associated with the previously mentioned candidate genes. So from this figure, it is also evident that milk production was the most frequently reported trait, followed by immunity and the environmental adaptation. So the systematically reviewed articles revealed recurrent selection signatures, such as the mentioned candidate genes, which are related to milk production, adaptability, immunity, and growth. However, the predominance of the fixation index, integrated haplotype score, and cross-population extended haplotype homozygosity, they suggest detection bias towards the common recent selection signatures. The limited use of the whole genome sequencing and the predominant studies in Asia and Europe, they underrepresent Africa. Therefore, in conclusion, the systematic review shows that the selection signatures in dairy cattle are predominantly associated with milk production, suggesting that the Holstein breeds are mostly bred for milk production as one of their economically important traits. This review further shows that the dominance of articles from Asia and Europe represent regions like Africa. Therefore, future studies should expand their focus to underrepresented regions, particularly in Africa. I would like to thank the supervisory team, the collaborators, and the Department of Agricultural Economics, and these are some of the references used. Thank you.
Thank you very much, ma'am, for the presentation. She did it in less than 6 minutes. Can we take the 2 hands? 1. That is the one.
Hi, that was very nice. It was nice to know about cattle, because the only time I see one is when it's in my plate, so it's nice. I had a question. You spoke about tropical breeds and underrepresentation. As someone who's not in your field, what would you say is the reason for the underrepresentation? Explaining to me, who doesn't know.
Okay, from the map that was presented, the articles are mostly from Asia and Asia and Europe, and these continents, they are having Holstein, they are not having our tropical breeds which are able to survive harsh environmental conditions and also which are disease resilient. Thank you.
Thank you very much. I guess that was the only hand. We can clap her as she... Oh, there is the last one. Oh.
Okay, it's just for interest's sake. Since you used papers that were both using genotype data and sequence data, I just wanted to know that how did you account, since the genotype data and sequence data, in terms of coverage for SNPs, they are quite different. So I just wanted to know, how did you account for the SNPs that were detected by both the 2 methods? Thank you.
Thank you for the question. Unfortunately, I didn't look into it. Sorry, apologies.
Thank you very much, ma'am, for the wonderful presentation. We are about to go to lunch, but can we clap for our presenters for being so beautiful and your own self for being a good audience? Our lunch will run for 40 minutes. According to the real time is 10 past 1. 10 to 2, we are back. Thank you.
heterosis. According to a study in 2013, milk is an essential component of the human diet that is consumed by 80% of the world's population, and also it's a source of a fundamental nutrient and minerals necessary for human health. Dairy crossbreeding on commercial farms remains low due to limited understanding of the genetic effects on yield and growth. Poor growth performance in purebred heifers compared to crossbred heifers. The objective of this review is to systematically review crossbreeding effects on dairy heifer growth and milk production. The study followed a PECOS framework of which the population used was Holstein, Jersey, and their crossbreeds from all over the world. Intervention or exposure, we used the crossbreeding effects of which we were looking at heterosis and the environmental conditions affecting growth and milk production. With comparison, we were comparing the milk yield of Jersey, Holstein, and also their crossbreed. The outcome is growth performance and milk production, looking at the traits of milk, such as milk yield, protein yield, and also somatic cell count. And then the design used a peer-reviewed publications. The method says strategy. The databases that we use are Google Scholar, ResearchGate, ScienceDirect, and PubMed. The keywords we use: crossbreeding, Holstein, Jersey, dairy heifer growth, and milk production, along with the boiling operators and/or. The data ranged from 2000 to 2025, of which gray literature was not used. This is the PRISMA table, which contains the total number of articles that were identified, of which there were 69. After screening and eligibility, only 20 articles were included in the review. Data was extracted looking at the publication, the year, location, breeding system, production system, the breed, sample size, and also the traits. Growth measures were looking at the weaning weight, birth weight of the heifer, with the milk production traits were looking at milk yield, fat yield, protein yield. And the risk of bias, we used a strobe checklist to ensure credibility of the findings. This graph shows publication by year, of which 2011 there were 4 articles that were published on crossbreeding effects on dairy heifer growth and milk production, of which in 2004, 2000, 2010, 2012, 2014, 2018, 2021 until 2025, there were no literature. And then publication by country, we can see that Turkey had the most published articles, of which there were 2, followed by Europe and other countries. This is a table 1 which shows the characteristics of the included studies, of which it shows the author, the year, location, breed, breeding system, the production system, sample size, along with the traits. Table 2 shows the growth traits, of which they also follow the same procedure of the author, the year, location, the breed, breeding system, production system, and also the growth traits, of which we have body weight, weaning weight, yielding weight, breathing weight, and etc. Of which in 2017, we can see that the study consisted of the body weight, weaning weight, yielding weight, and also other studies did not consist of those traits, of which it's a research gap that most studies should be conducted that will include the weaning weight, yielding weight, and other traits. Table 3 shows the milk production traits, of which Holstein have the highest milk production, because we all know that Holstein produces large quantities of milk as compared to other dairy breeds. And the table shows that Holstein have the highest milk production of 5,000 and something, and also Holstein have the least milk production, because Holstein only produces milk which contains of fat yield, fats and proteins instead of the quantity as compared to the Holstein. Crossbreeding effects on milk production is that crossbred animals, they tend to have better milk components despite their slightly lower yield. And also crossbred animals, they generally show lower somatic cell counts, indicating that there's an improved other health and milk quantity. Crossbreeding effects on growth traits. It was shown that in 2019, a study was conducted which involved that over 189,000 animals. They showed that crossbred calves had a comparable improved growth performance compared to the pure breeds, which is fundamental for pasture-reliant operations. Another study conducted in 2017 reported high birth weight, of which it was 19.99 kgs, weaning weight of 69.45 kg, and yearling weight of 113.21 kg in a crossbred of Holstein and Jersey under semi-intensive production system, indicating a good pre-weaning and post-weaning growth. The benefits of crossbreeding in dairy cattle is that it produces animals that performs better in low input system, and also it enhances the growth, fertility, milk yield, fat and protein content through heterosis. The limitation of crossbreeding on dairy cattle is that it leads to loss of breed purity, and also the performance may differ across generation and environments, making outcomes less predictable. In conclusion, crossbreeding, especially Holstein and Jersey, it leads to improved growth, fertility, improved milk yield through heterosis. So well-planned breeding programs tailored to local systems, they boost farm profitability and also support sustainability and strengthen global food security. So future research need is that more studies should be conducted in areas whereby there are less studies that have been conducted as of yet to improve global applicability, and also exploring long-term effects of crossbreeding on other health and disease resistance. I would like to thank the research was supported by the Department of Agricultural, Environmental and Animal Production, and also the TADA Research Group and the University of Limpopo. These are some of the references used for this review. Thank you.
All right, thanks Maredi for a wonderful presentation. Can we have 2 hands for questions? Where is the mic? This side.
Hi, Timothy, and nice presentation. I just wanted to know, so crossbreeding, you spoke about like how it will influence the growth and milk production, but how will that fit in with climate smart and climate change? So have you read anything about what the impact would be of crossbreeding? Would we still then have some sort of adaptation or heat tolerance, because heat stress is obviously a big thing in dairy cattle? So will the crossbreeding between, let's say, Holstein and Jersey improve the production while sort of managing heat stress and stuff like that as well?
Yes, and remember that Holsteins are susceptible to heat and all that, and also Jersey is adaptable to various climates. And so crossbreeding those two, it will produce an offspring that will be able to adapt to all those climates, and changes. Thank you.
Thank you very much. That was a very nice presentation. You'll pardon me a bit because I'm not really from this field. I'll just want to know, what motivated you to focus on crossbreeding in dairy heifers rather than the pure breed systems?
With pure breeding, is that it focuses on only 100% Holsteins or 100% Jerseys. So doing crossbreeding, it will lead to a production of an offspring that will comprise of both the traits of a Jersey and a Holstein, of which we all know that Holsteins, they have high milk production in terms of quantity, and also with the Holstein, with the Jersey, they produce milk of high quality in terms of fat and yield. So using pure breeds will lead to what can I say? Will not have the same effect as using a crossbreeding method. Like the results will not be the same.
All right, okay, thank you. One last one. You used the Prisma and Pico. What is the rationale? Why not one of those? Why both?
The Prisma one, I used it because I wanted to actually see which articles I can use on my study, checking the eligibility, because I cannot use all the articles because we focused on Holsteins and Jersey. So we needed to check which articles had the keywords and which ones they were focusing on certain traits and all that. Then with the PICOS was used to, I don't know. I'm sorry. I think I'm done. I'm sorry.
All right, thank you, Maredi, for the presentation and for answering those questions. Can we now welcome Matsidi Sorapezwa to come onto the stage? She will be presenting on soil quality shifts and land capability constraints in abundant crosslands of Mpanama village, Limpopo province.
Thank you, Chair. My name is Matsidi Sorapezwa, and I'm a second-year master's student, and I'll be... Okay. My name is Matsidi Sorapezwa, and I'm a second-year master's student, and I'll be presenting a portion of my current study titled From Fertile to Fragile, Soil Quality Shifts and Land Capability Constraints in Abundant Croplands of Mpanama Village, Limpopo Province. And my co-authors are Dr. Mbanjwa and Professor Dlamini. Soil fertility and land capability are key determinants of agricultural productivity. However, land degradation threatens these factors, particularly in semi-arid regions like South Africa. Over 60% of South Africa's land is degraded due to unsustainable farming, climate change, and land abandonment. Mpanama Village amplifies this issue, where cropland abandonment has led to soil erosion, bush encroachment, and declining fertility. Therefore, this study is important since there aren't many studies on soil fertility and land capability in rural communities. The aim of this study is to identify sustainable practices for soil restoration and optimal land use. And the objectives were to assess the soil fertility in 2 land uses by analyzing selected soil properties, and to also determine the current land capability classes to identify environmental limitations. This is a map of my study site. The study was conducted in Mpanama abundant cropland. The area experiences an MAT of 17.8 degrees Celsius per annum and an MAP of 500 to 600 milliliters. And the soils that were found here were, the texture was sandy and sandy loam. So the first thing that was done when we got to the field was to conduct a soil survey. In the soil survey, some physical parameters were determined. These include the soil depth, the color, the texture, as well as the permeability. And we also, after the soil survey, we did a soil classification in order to determine our soil family and soil forms. Then we went on to collect our soil samples. The soil samples were collected using a spade at a depth of 0 to 15 and 15 to 30 centimeters. Then the soil samples were then transported to the lab where parameters such as total organic nitrogen, total organic carbon, and bark density, phosphorus, and potassium were also determined. Then this data was then statistically analyzed using the GenStat software. The land capability classes were determined using the physical parameters which were determined in the field and also using this key above. The results showed that there were 3 soil forms which were found in this abundant cropland, namely Wesley, Tukulu, and Chiombo, with Wesley being the dominant soil form in this area, and it displayed the most limitations based on its diagnostic horizons. So for the purpose of this presentation, I will only be focusing on the fertility results of Wesley soil form. So here I have 3 graphs which represent the bulk density, nitrogen, and also the carbon. So the first graph shows the bulk density in the 2 land uses, abundant and undisturbed, and it was found that the bulk density was higher in the abundant cropland. This may be attributed to the fact that in these abundant cropland, agricultural practices such as tillage were occurring. This then resulted, and there was also a reduction in organic matter due to these agricultural practices, which then lead to the bulk density being higher as compared to the undisturbed land use. And on my right, I have a graph of the total organic nitrogen, and below we have a graph of the total organic carbon. And it was found that these two, they show similar patterns of having higher concentration in the undisturbed. This may be attributed to the fact that in the undisturbed, since we have more vegetation, it means that there was higher organic matter inputs. This then results in the high nitrogen and also the high carbon. So since there is high nitrogen in the undisturbed, meaning that there will be more there will be a release of nitrogen through mineralization. And also this can be said the same about the total organic carbon. So here I have my results for phosphorus and potassium. So in the Wesley soil form, it was found that the potassium was higher in the abundant croplands as compared to the undisturbed. This may be attributed to the past fertilizer application in this area, which explains the high potassium concentration in this in the abundant as compared to the undisturbed. And it may also be due to that, since the tillage practices were occurring here, they would then expose clay minerals such as elite and vermiculite to weathering, which then results to the release of potassium. And for the phosphorus, it was found to be higher in the undisturbed as compared to the abundant. This may be attributed to the fact that phosphorus is an immobile nutrient, meaning it clings to surfaces such as organic matter. And we know that in the abundant land use, there was higher organic matter. Therefore, this then explains the accumulation of phosphorus being higher in the undisturbed land use. So here I have a map which shows the land capability classes which were found here. We found 2 land capability classes, which was class 3 and class 4. Both these classes are considered to be suitable or for cultivation or arable, but they have limitations. They have limitations in terms of depth, texture, and also the depth and texture, and also the slope, and also the fertility. So the land capability class 4 has more limitation as compared to class 3. And if you look at the map, you will see that in the areas in which we have land capability class 4, it is the areas in which we found the Wesley soil form, which then supports our fertility results, which say that Wesley is the soil form with more limitations. In conclusion, I can say that land use shows significant effect on soil fertility, and soil fertility in abundant cropland shows higher bulk density and higher potassium, largely due to past fertilizer use and low organic matter. Therefore, sustainable practices such as reduced tillage, crop rotation, vegetation regrowth, and residue retention are essential to restore soil health. And the rehabilitation through prioritizing suitable land use, agroforestry, and continuous soil conservation measures are recommended. I would like to thank my supervisory team and also the University of Limpopo. I thank you.
Thanks, Matshidiso, for a wonderful presentation. It's now time for questions and comments. Can we have questions and comments from the floor? I see one hand, 2.
Hello. Ooh, hi. That was a very nice presentation, by the way. I liked it. I just needed confirmation on something. Maybe you mentioned it, I missed it, but you. Your sampling points, how did you which method did you use to select those if you did sample?
Okay, we use a stratified sampling method.
All right, next question.
That was a lovely presentation. It's funny how Panama also falls under my region of study. I just want to ask something. Title said from fertile to fragile. So I wanted to know, how do you define a shift from fertile to fragile in terms of measurable soil indicators?
Okay, I had 2 land users, which was my undisturbed and the abundant cropland. So I was using the undisturbed as a sort of my control. So I was comparing the results in the undisturbed and also the abundant. That's how I knew that there was a shift or a change in the soil fertility by comparing the 2 land users.
Do you have data, like previous data, to show that previously this is the state of the soil and currently this is the state of the soil?
No. For the purpose of this study, this is a land assessment study, so we are looking at the current state, since in my main study they are trying to develop a community garden. So what I'm looking at is what is there now and what can I best recommend for what they are trying to do, which is develop a community garden.
Okay, but I'm not satisfied. I wanted to know the shift. That's the part I wanted to know. How did it shift?
All right, one last one, Matshidiso. Stratified sampling. Why stratified random sampling, and how did you apply it?
Okay, I think for that it was because in the area that we were, there weren't many variations and the area was big, so we decided to divide the area into smaller sections since there was more uniformity in the area. There weren't many differences.
All right, thank you, Matshidiso. You are released. All right, can we call upon the third presenter, Leshabana Mabina? They will be presenting on systematic review of advanced deep learning, GIS, and remote sensing approaches for post-flood disaster management assessment.
Good day, ladies and gentlemen. I am Mr. Mabina LM, and I'll be presenting a systematic literature review under the title Deep Learning and Geospatial Approaches for Post-Disaster Damage Assessment, under the supervision of Prof. H. Chukori, Prof. F. Matiba, and Mr. Mashao. Global warming alter with our planet's hydrological cycle, increasing the risk of flooding from extreme precipitation event due to Clapeyron effect, Clapeyron relation, which indicates that a warmer atmosphere can hold approximately 7% more moisture for every 1 degree Celsius, 1 degree Celsius of warming. This can be witnessed in the cut-off flows, cyclones, and monsoon, which have the potential of generating heavy rainfall that cause flooding. So now we are going to look at the case study of Durban 2022 and 2019, where the eThekwini area has received a lot of rainfall, which has resulted in massive flooding that induced landslide that damaged buildings, main infrastructure such as buildings, roads, and there were reports that there were power outage due to power lines being washed away by the massive landslides. As you look at this picture, we can see that indeed there were those disasters where we see our building damaged, and this one at the Umdoti area, north of Durban, where we see a building that has been collapsed, and we see here a big fold that there was a massive landslide there. So this study aimed to review the current literature review on the advancement in geospatial technology for post-flood damage assessment integrated with deep learning approaches to assess the severity of the disaster. This review will examine the spatial distribution of scientific publication related to geospatial post-disaster damage assessment, surface displacement method to identify and measure deformation or landslide, building damage assessment using deep learning, image classification, building height assessment, and power outage. The study conducted a literature search on websites such as Google Scholar, Springer, and Web of Science to make sure that the study is replicable and provide transparency. So the study has followed the PRISMA guideline and resulted in 176 papers which were taken to our studio for bibliometric analysis. On our result, looking at country scientific production, we see that more contribution were done in China, gradually decreasing to America and Canada, and the gray shaded side shows no record of contribution, which highlights a geographic gap, and the study speculates that this could be due to technology, access to higher learning areas, and funding to research. Looking at this study considered papers from 2010 until 2025 June, and looking at our annual scientific production, it shows that 2024 was the highest, has been the highest year where we have found more papers related to the objective and the topic of the study. On our result, we have seen a massive shift from traditional method, which were time-consuming, to more remote sensing method, which we have filtered the remote sensing data according to their strength and their weaknesses. So we looked at images such as radar and LiDAR, and we focused more on radar satellite, since optical imagery such as Landsat and Sentinel-2 have a problem when they are cloudy condition, and this flooding impacts. This flooding events occurs when there is a cloud, when there are cloud covers, which deem them unfit to assess damage. So, furthermore, on radar, we have looked at satellites such as TerraX, ALOS, and Sentinel. So, TerraX and satellite imagery such as TerraX and ALOS have a problem when it comes to spatial resolution and temporal resolution, which led us to focus more on Sentinel-1, since it has a temporal resolution of approximately up to 6 to 12 days and a spatial resolution of roughly 10 meters. So in assessing the landslide, we have found techniques such as persistent INSAR, differential INSAR, small base INSAR, and Fisher information enhanced maximum likelihood INSAR, which are used in order to measure the permanent scatter and the distributed scatter. And this method are also used to measure the subsidence of land. On building height, we have found methods such as shadow analysis, which are less effective when in high-rising buildings with overlapping shadows, and prismatic interferometry, which needs 2 satellite imagery, 2 radar sensors beaming at one area in order to measure the building height. So these two methods, the shadow analysis and the static, have been used to measure building in building in higher rising areas such as in cities. However, they have not shown the effectiveness to measure buildings in lower rising areas, lower buildings. So to overcome this limitation, studies such as Lee et al. Lee et al. 2020 and Wu et al. 2023 have proposed a dual polarized band of VVH to measure building height in the cities where there are high-rising buildings, and in the outer skirt where there are low-rising buildings simultaneously. So in building classification, we have classified our classification method in equivariance and invariance. So in equivariance, we found, in invariance, we have found classification method such as convolutional neural network, recurrent neural networks, recession neural networks, and on the equivariance, we have found that there is less study done in the equivariant category in order to monitor change. So to measure our power outage, several nighttime light data have been found, such as the Luagi Sentinel-2 using different bands, and DSP ALOS. This satellite imageries, this nighttime light data have a problem with spatial coverage and temporal resolution, which led us to focus more on visible infrared radiometric suite provided by NASA, which provide data daily, nighttime data daily with a global spatial coverage. And it's important to note that all nighttime light data struggles with the booming of light and saturation of light, which can further falsify the extent of light coverage on the surface. To overcome these limitations, the studies have proposed the viewing zenith angle where to provide a continuous monitoring of electricity availability on the surface. I'd like to acknowledge my supervisors and the University of Limpopo, the Department of Geography and Environmental Studies, and EW-CETA for funding. In conclusion, we have found a lack of model coupling to achieve one objective. Models such as, which falls under interferometry, such as building height model and PS INSA. I thank you. And this is a list of my reference. And last but not least, I'd like to thank Babina Noko for the wonderful job they did yesterday.
All right, thanks, Mabina, for a wonderful presentation. Can we have 2 hands for questions or comments? 2 hands for questions or comments. I see 1 hand there.
Thank you. Thank you for the presentation. It was a nice one, but not bad. Okay, when you were presenting the slide of the building classification, where you found is it the equivariance and the invariance?
Yes.
So yeah, I have a question there. Would you, because maybe you didn't clarify it, or maybe I missed it. Would you advocate for the use of the equivariance classification technique or the invariance technique? And if you would advocate for the equivariance classification technique, why that over the invariance technique?
Okay. If we are looking at the invariance, we'll have to go back to the words of Geoffrey Winton when he said the pooling operation in the convolutional neural network is a big mistake, and the fact that it works so well, it's a disaster. Which states that if you look at this, to check damage, you need the classification that analyze change. So the invariance means that the input changes, but the output doesn't change. So if I come to you now wearing the spectacles, you see me as me. But now I come not wearing spectacles. So you still recognize me as me. However, if you go to the equivariance, when you want to check change detection, it will analyze these 2 changes, these 2 smaller changes that you see me wearing glasses and me not wearing glasses. So it simply means that the input changes. And if the input changes, the output should change.
All right, thank you for the response. One last question. You are from the School of Agriculture and Environmental Science, right? What are the implications of your findings to the agricultural sector and farmers in particular?
In farmers?
I'm not quite getting your question, because this one we are specifically dealing with flag.
What is the contribution?
Oh, you're asking.
Yes.
Okay.
what is the contribution of your findings to the agricultural sector or farmers in particular?
Okay. In from from the systematic literature review and how I understand the question and the contribution of this study, how the study will contribute to agriculture in areas where they experience this these disasters such as landslide. We will be able to identify landslide occurring in a farm and measure the magnitude and the velocity of the eroded soils. Thank you.
All right, thank you. You are released. The next presenter is Lebogang Rateke, who will be presenting on phytochemical analysis of Bambara groundnut selected plants as novel feedstuffs. Can we please welcome Lebogang to the stage?
Good day, everyone. My name is Ms. Rateke Lebogang, and I'm from the Department of Agricultural Economics and Animal Production. I am supervised by Professor Busisiwe Gunya and Mr. Sanele Giana, and we conducted a study on the phytochemical analysis and nutrient composition of Victor Septarian selected plant plants as novel feedstuff. And this is the outline of my presentation. This is a picture of Vicna Septarian, which is also known as your Bambara groundnut. Those who've never seen it, this is a picture of it. There is an over-reliance on conventional feedstuff in the livestock industry. When I say conventional feedstuff, I am talking about your soya bean, I am talking about your maize. And the prices of these conventional feedstuff has been increasing over the years. The reason behind the increase in prices is because of their production, it's declining. And you know that when production declines, the little amount that we produce, when we take it to the market, we're gonna sell it at a higher price, right? So those higher prices, our farmers, especially communal farmers, they cannot afford to pay. So they resort into feeding their livestock low quality feed. Therefore, we really need to have an alternative that is locally available, an alternative that is also sustainable. And I present to you Bambara groundnut. It is that alternative. However, there is a knowledge gap when it comes to their nutritional and anti-nutritional profiles, especially the other parts besides the seeds, the leaves of it, the roots, the stems, and also the shell covers. We don't know their nutritional and anti-nutritional profiles, and that's where my study comes in. Bambara groundnut, it is a climate resilient and underutilized legume. When I say climate resilient, I mean this plant can grow under drought conditions. It can grow in poor quality soils. Beyond its adaptability, this plant, it produces a substantial amount of biomass in terms of the leaves, the stems, and the roots. And most of the time, this biomass are normally thrown away because even now, when you can go to the market and say you request a Bambara groundnut, they are going to give you the seeds. They're not going to give you the stems or the leaves or the roots. So what if I can tell you that there is a potential for us to valorize those residues into valuable feedstuff? When I say valorize, I mean that we can add value to them, or we can transform them into something valuable. In this case, we can transform them into livestock feed. And therefore, this will contribute to a sustainable livestock and a circular bioeconomy. The aim of my study was to evaluate the phytochemical and nutrient composition of Bambara leaves, stems, seeds, shell cover, roots as a novel feedstuff. And my objective was to compare the nutritional and anti-nutritional composition of Bambara leaves, stems, roots, shell cover, and also seeds. My hypothesis is in line with my objective. We collected these, the Bambara groundnut leaves, stem, roots, shell cover, and also the seeds from a communal farmer at Ramolepe village. And then once we collected them, we separated them into seeds, stem into stems, leaves into leaves, etc. And after that, we sent those samples to the NutriLab at the University of Pretoria, where they performed a proximate analysis following the AOAC procedures, and they gave us results on the micronutrients, ash, DM, GE, and also tannins. And then we took those results and we analyzed them using the SPSS software. We also conducted a Taki test, and then the means were considered different if P was equal to or less than 0.05. For the results, table 4.1, it shows the nutrient composition of different plants of Bambara groundnut. From this table, when it comes to the crude protein, we can see that our seeds, they recorded the highest crude protein, and followed by our leaves. And then when it comes to starch, also our seeds, they recorded the highest, and our shell covers, they recorded the least. And then when it comes to the ash, our roots, they were like, you know what, we're gonna come first, and then they recorded the highest, and our seeds recorded the least. And when it comes to the screening of the phytoconstituents, only the tannins were found available or present from all the samples. Figure 4.1, it just shows the total tannin content in different plants of parts of Bambara groundnut, and we see that our leaves, they're having the highest amount of the tannin, and then our seeds, they recorded the least levels of tannins. The leaves, from our results, the leaves, they recorded the highest dry matter, and then these results, they contradict with the results found by another author this year in 2025, whom reported that the seeds, in their case, recorded the highest dry matter. Remember, ours, it was the leaves. And the reason behind ours having, our leaves having high dry matter was because of low inputs, in which caused a reduced transpiration and moisture. Remember, I said that we collected our samples from a communal farmer in Ramolepe, and we asked the farmer if he used any artificial irrigation, and then he said no, he only relied on natural rain. So this is the reason why the leaves had the highest dry matter. And our seeds, they recorded the highest crude protein, in which was in line with the study by Boateng back in 2013. And the observed similarities between the two was likely due to the growth environment. Boateng, Bambara groundnut, it grew in the field, so also our samples, they were also grown in a field, not in a greenhouse or in a controlled environment. Our leaves also, they reported the highest gross energy, making them a more usable as a feed for livestock. And so also our seeds and leaves, they recorded the highest either extract over the other plants, and on top of that, the seeds recorded the highest starch, and our shell covers, like I said, they also recorded the least starch, carbohydrates, energy. And then these results are in line with the results found by an author called Hlanga back in 2021, in which he also found starch to be in that particular level. But however, a study by Yao back in 2015, he found slightly different, slightly higher than ours. It was around 50%. And then this observed slight difference was likely due to different genotype, the environment, and the cultivation practices. So, like I said, our seeds, they recorded a high starch. This makes seeds a suitable source of digestible carbohydrates for livestock, particularly monogastric. When I say monogastric, I'm referring to your poultry chickens. Okay. Oh yes, and our roots, they recorded the highest crude fiber, and then our seed, Yona, it recorded the least crude fiber, and then the low crude fiber that was observed in the seed of the current study, it goes hand in hand with the ones that are found by a study by Barlow and Rano back in 2022, and the leaves, when it comes to the tannin levels, the leaves, like I said, they recorded the highest tannin levels. They recorded the highest tannin levels, and then our seed recorded the least tannin levels. Yes, our leaves, they recorded the highest tannin levels, but those tannin levels are still within the acceptable levels of tannin. And then we know that moderate tannins can enhance nutrition and health in monogastric. We are concluding that seeds, since they have high protein and energy, they are ideal for growth and production. And leaves, since they have high fiber and dry matter, they can act as an excellent roughage. And then our roots, since they roots and shells, they reported highest minerals, therefore they can be used as a mineral source. And then a take-home message is that all plant plants can be strategically used to enhance livestock nutrition and feed efficiency. So we don't have to throw those leaves, just use them for something. They are abundant in nutrients. And we recommend for you to use the seeds as an energy and protein source, and use the leaves as a fibrous feed source in ruminants, especially where roughage is very scarce. And also, we can explore the shells, the stems, and also the roots for inclusion in low input livestock feeding system. And also, maybe we can use them as composite for soil improvement. These are references that I used. I would like to acknowledge my supervisor, Professor Gunya, my co, Mr. Sanele, and NRF for the funding, and also the Department of Agricultural Economics and Animal Production. Thank you guys for lending me an ear.
All right, thank you, Lebrang, for a wonderful presentation. It's time for questions or comments. Can we have 2 hands for questions and comments? I see 1 hand there. Another one. You can start there.
Okay, thank you, Ratsege. I was a little bit concerned on your screening of phytoconstituents. When you said that flavonoids, I think it was flavonoids and alkaloids that were not detected. So how many standard chemical tests did you try out to see if indeed these phytoconstituents are not present?
Okay, thank you for the question. The lab technician back in UP, they did the thing, they tried it twice, and then in that trial only the tannins were recorded from their results that we got.
I mean, how many chemical tests? There are maybe 3 or 4 chemical tests to see if, to detect or to test the presence of alkaloids, and there are more than 2 chemical tests to test for the presence of flavonoids. I want to see like how many did they try, or they tried only one chemical test and then concluded that this phytochemical is not present in those plant parts.
Honestly speaking, I don't know how many tests they conducted at the NutriLab. All we did was we received the results from them. We told them this is what we want, and then that's what we got. But then I'll do a follow-up with them and ask them how many tests did they do, the chemical test, how many did they do?
Okay, because I think there might be biasness
Ne?
if you only try one chemical test. Yeah.
Okay, thank you.
Thank you for the wonderful presentation. But I noted that you mentioned that there is a nutritional gap in Bambara groundnut. So I want to ask, what is the gap, or to be specific, this nutritional gap? Because Bambara groundnut, it's already being researched in terms of the nutrient content or composition, and it is already reported in the literature. So if this was your problem statement, what is the nutritional gap, to be specific, in Bambara groundnut?
All right. Thank you, sir. Okay. My project, I was researching I was not only focusing on the seeds. The seeds are the ones that are mostly talked about or published. But when it comes to other parts, like your leaves, your roots, your stems, and so also your shell covers, there's absolutely no known nutritional and anti-nutritional composition. So I think I will be one of the first people to publish that, because I conducted my literature review, and then I found nothing on the shell covers, I found nothing on the roots, I found nothing on the stems, only the leaves, like few of them, but then most papers are on the seeds.
Okay, that's fine, and thank you, but I will recommend that to be specific, because when you say there is a nutritional gap, you are being wide, because in terms of the seeds, it is widely researched and published. So I would recommend that next time you should be specific or on this plant material. Yeah, thank you.
Oh, thank you. I thought I mentioned it, but then it's okay. Thank you very much, sir.
All right, thanks, Lebogang, for answering the question. The last one, I have to be consistent, because I asked previous presenters. I also need to ask you, these alternative diets, did you test them on the livestock or animals themselves to measure or to evaluate their uptake?
Well, this project that I'm presenting currently is part of my, it's a sub topic of one of my master's project. So on my master's project, I'll be feeding it to Bushveld chickens for 90 days, and then I'll be taking growth performance results, and so also I'll be checking for meat quality. So I'll be testing it next year. I'm coming back, and then I'll give you guys the results.
All right, thank you. You are released.
Thank you, ma'am.
All right, I would like to hand over to Dr. S.T. Hadebe to come and chair the next 4 presentations. Thank you very much for your cooperation. You've been, you did well. Thank you.
Thank you, Dr. Lefopan. You also did well. We can clap hands for Dr. Lefopan. Okay, I'm going to be your chair for the next hour or so. Just some house rules. I'm going to be sitting around here. If you see me standing, it means you've got about a minute. I'm just asking that you start wrapping up if you see me standing. Okay. The next 4 speakers, the next one is Prudence Mohapa. If you can start coming up. Prudence's study, it reminds me of when I was still very young. If you pass, they will wrap you an indigenous chicken, and then they will give it to you, and you'll be so happy. Now we call it a hard body prudence. Now we don't eat it now. We're not part of the poor. Ladies and gentlemen, I have to apologize to you. It seems like there was an error when slides were being uploaded. So in the meantime, may I please call Machete. Machete, please come up. Sorry that I'm just calling you out of the blue like this. And whilst Machete is coming up, we are having another graveyard slot after this. Those who were supposed to present from 1500 onwards, please also start loading your slides so that we don't have to delay ourselves further. Machete will be speaking to us about drinking water and tailing dust contamination. Let's clap hands.
Thank you, Chair, for the opportunity. Standing before you is Flora Machete. I'm under the supervision of Dr. M. Gleche and Prof. B. Gunya. I'll be presenting on the influence of tailing dust contamination in drinking water on the reproductive performance of dewars in subtropical regions. This is the outline of my presentation. Households in rural areas that are depending on goats for provision of meat, income, social, religious, and cultural roles. In mining areas, tailing dust contributes to water contamination. Clean water is essential for livestock health and productivity. Although goats are known to be tolerant to mine tailings, there is a concern about the contamination of water. Therefore, the information on impact of heavy metals in drinking water on reproduction of goats is limited. Consumption of water with heavy metals can damage cells, cause various disorders and reproductive diseases. The aim of my study is to determine the farmer's perception on the effects of heavy metals contamination in drinking water on the reproductive performance of dewars in subtropical regions. The objective of my study is to evaluate how household farmers perceive the effect of tailing dust contamination on the reproductive performance of doers. The hypothesis aligns with objectives. The study was conducted at Wapalaura local municipality, which has an average rainfall of about 450 millimeters per year. Then it experiences the warmest months in December and January with an average temperature of 34 degrees Celsius. 12 water samples were collected using water bottles with screwing caps, 6 from each region. Then the water samples, sorry. Should I restart? Okay. Sorry. 12 water samples were collected from 2 regions, 6 from each, and then they were stored in, the water samples were stored in cooler box with an ice at 0 degrees Celsius. Then heavy metals were measured using ICPE 900 at the University of Limpopo. A total of 200 households were randomly selected in Hamakushani, which is located 6 kilometers away from Bosveld, and Hamashishimale, which is located 21 kilometers away from the mine. Then the data was collected using questionnaires in local languages, Xitsonga and Kilobedu. The statistical analysis was used to analyze the data, and the relationship between household demographics and water quality was determined by chi-square test. From the results from the analysis that was conducted, it was found that the water sources in contaminated areas had high level of chromium, copper, magnesium, and lead as compared to uncontaminated areas, and then the concentration of dissolved oxygen was high in contaminated areas, unlike in uncontaminated areas. This indicates that the goats in contaminated areas were exposed to to poor quality water, which affects their health. Therefore, with the participation in contaminated areas and uncontaminated areas, we found that the education and ownership of cattle was significant, with high performance in uncontaminated areas, and gender, age, and training were non-significant, as well as the ownership of small stock animals. From the variables as presented in table 5.3, water was the main constraint to goat productivity. Therefore, in table 5.4, the river was the main source of water, was ranked as the first source of water in these 2 regions. Lactation was the main challenge lactation length was the main challenge that contaminated areas farmers experienced, and inter-kidding period was the main challenge that farmers in uncontaminated areas experienced. The lactation length was prolonged by the accumulation of heavy metals in the tissues, which reduced the milk let down. And during puberty, the accumulation of heavy metals in the body, they caused a prolonged stage of puberty, which in turn influenced the age at first mating and age at first kidding. Therefore, the accumulation of tailings in the body also resulted in high interkidding period. Therefore, in conclusion, the goats in contaminated areas are at higher risk of delayed age at first mating and prolonged lactation length. Therefore, mitigation strategies are required to be implemented so that we can improve the water quality. Further research should examine the behavioral, physiological, and hematological responses of goats in mining areas. This study will contribute to the body of knowledge as well as farmers around contaminated areas, since they are the most affected farmers as compared to uncontaminated areas. Therefore, the infrastructures required for all the materials required for the study were available at the University of Limpopo, and the study followed the standards of ULREC ethics. Therefore, I would like to acknowledge my supervisor and my co-supervisor. This is part of the references that were used for this study. Thank you.
Okay, thank you, Ms. Machete, for your very brief presentation. It didn't even take 7 minutes. That gives us quite some time for questions. Can I see hands? There's one hand there. There's a second hand. So we'll attend to those 2 hands. Contrary to what the previous question asker was asking, once if you do it right is enough, good people. So even if we get 1 question, it should be enough.
Okay. Thank you, Chair. To the presenter, you mentioned that there were some socio-demographic factors that were having some significant effect on your results, like the age and their gender status. What could be the reason there?
Okay, thank you, sir. On this one, education and ownership of cattle, they were the ones which were found to be significant. Reason being, in uncontaminated areas, we had more people who were educated, unlike in uncontaminated areas, we had more people who were educated, unlike in contaminated areas. Therefore, even the cattle ownership was high in uncontaminated areas, unlike in contaminated areas. On the cattle ownership, cattle are more susceptible to tailing dust or to the heavy metals as compared to small stock, because cattle cattle feed more. They require more water, they require more feed, therefore they end up consuming more metals, more heavy metals as compared to small stock. Therefore, with gender, age, training, and the ownership of small stock in both areas, they had almost similar number of participations, meaning that for these parameters, the numbers were almost equal. Sir?
Can we get the second question? Okay.
Yeah, so thanks for your presentation. So I would like to know from your study, how did you assess the health of the animals? Because I heard you mentioning something about you were assessing the water, that it's contaminated and it does affect the health of the animals. So I want to know which aspects of health did you investigate and how did you do it? That's the first question.
Can I answer the first question and then we'll go to the next one?
Yeah.
Okay, thank you for the question. The reason why we collected the water samples was to analyze the type of heavy metals which are found in Palabora. Then we did not conduct a study on the goats. Therefore, what we did was to get what farmers think about tailing dust, and then after that, with the heavy metals found on water, I went online to check the previous studies which were conducted and relate those effects on my study.
Yeah, so this leads to my second question. So wouldn't you think that, for example, you mentioned intercalving a period, so it is a bit longer. So I think you're associating it with water contamination.
Yes.
So wouldn't you think other factors such as shortage of feed, for example, because in Palabra it's too hot and yeah, there is there is a shortage of refuge. For example, even now it's very much bad. So wouldn't you think that could be the case, or parasites or other factors?
Yeah, there are other factors that influence the interkeeding period, but from the results that I found, I associated the heavy metals to be part of the contributors to the prolonged interkeeding period.
Okay, let's clap hands for Ms. Machete. I'm not trying to suppress further questions, colleagues, but if we can just stick to one, and then if you want to get to the next question, just indicate to me. I will tell you how we're doing in terms of time as well. Okay, because sometimes some questions are brief and can be allowed for follow-up. Okay, we'll have to call back Prudence to the floor. I think the problems have been solved. So please, you can come.
Good afternoon, everyone. My name is Mokapa Prudence, a second-year master student in animal production. I will be presenting a part of my master study titled Effect of Breed on Air Quality Traits of South African Indigenous Chickens. This is an outline of my presentation. Eggs are considered the most affordable and widely accepted animal products. They are an important source of nutrients for humans and a nutritionally complete food that is consumed worldwide. Egg quality consists of those characteristics of an egg that affect its ability to be accepted by consumers. Consumers have different preferences when it comes to egg quality. Some prefer large eggs with a firm albumen, which is a key indicator of its freshness, as well as a dark-colored egg yolk. So therefore, it is the responsibility of poultry farmers to provide high-quality eggs and also maintain that egg quality in order for consumers to accept them. Indigenous chicken play an important role in the livelihoods of most rural families in South Africa, providing meat and eggs as reliable sources of protein. They are known and also valued for their ability to adapt to harsh climatic conditions, disease resistance, as well as the ability to scavenge. Figure 1 and 2 is showing a picture of the Potchefstroom kokok as well as the Bushveld chicken, respectively, which are known and valued for their dual purpose ability to produce both meat and eggs. Although these indigenous chicken contribute to the livelihood of farmers and are also tolerant to harsh climatic conditions, they have low productivity. This may be due to a number of factors such as high mortality rate in kids as well as poor nutrition. Therefore, this poses a problem of security in the supply of these animal proteins to the population. Indigenous chickens in the majority of African countries have received little attention. There is lack of comprehensive research on these indigenous chickens. Therefore, efforts to improve these indigenous chickens through selection should be a priority. The poultry sector plays an important role in agriculture, with these indigenous breeds playing a key role in both the rural and small-scale farming system. Breed has an effect on egg weight as well as eggshell properties. Egg weight is considered the most important egg quality parameter by consumers. However, there is no information provided on the effect of breed on egg quality traits of this bushveld as well as the Potchefstroom Gokok chicken breed. As a result, this study's aim was to provide information that will assist both the researchers and farmers in discovering the best South African indigenous breeds with better egg quality traits. The objective of the study was to determine the effect of breed on egg quality traits of the Potchefstroom Gokok as well as the bushveld chicken. The study was carried out at the University of Limpopo Experimental Farm in Limpopo Province, South Africa. 50 Potchefstroom Gokok indigenous chicken as well as the bushveld were used in the study. They were raised under an intensive management system on deep litter from the first day until they started laying eggs. And then 400 eggs were collected from each breed. A completely randomized design was utilized in the current study. The external egg quality traits were measured as described by the indicated author. An electronic weighing scale was used to measure both the egg weight and the shell weight, and the egg length as well as the egg width were measured using a vernier caliper as indicated by the diagram. The internal egg quality traits were measured as described by the indicated author. A yolk separator was used to separate the egg yolk as well as the egg albumin, and then the egg yolk was measured using an electronic weighing scale. The albumin weight was obtained by subtracting the yolk weight as well as the shell weight from the total egg weight. This is shown by the diagram below. Data was analyzed using statistical package for social sciences, version 30.0. T-test was used to achieve the objective of the study, and this is the general linear model that was followed. Table 1 shows the effect of breed on egg quality traits of the bushveld as well as the Potchefstroom Gokok chicken breeds. It was found that breed had an effect on the external egg quality traits such as your egg weight, shell weight, egg length as well as the egg width, and the shell surface area, as well as the internal ones such as your yolk weight, yolk or albumin, and egg volume. However, breed did not affect the external egg quality traits such as your egg shape index, shell ratio, and unit surface shell weight, as well as your internal egg quality traits such as your yolk ratio, your albumin weight, and albumin ratio. Looking at the table, it is evident that the bushveld had higher mean values for all the egg quality traits except for the albumin ratio, which was higher in the Potchefstroom Gokok chicken. The findings of the current study were consistent with those of the indicated author, who reported that breed did have an effect on the external egg quality traits such as the egg weight, egg length, egg width, shell weight, and shell surface area, but breed did not have an effect on the shell ratio and unit surface shell weight of the Potchefstroom Gokok chickens. The findings of the current study differed with those of the indicated author, who found that breed did not affect both the egg width as well as the shell weight of the indicated chickens. Our findings also differed with those of the study that was published in 2015, who reported that breed affected the albumin weight of the indicated breed. This may be due to a number of factors, such as your breed differences, nutrition, as well as the environment. The current study found that the bushveld chicken performed better in terms of quality as compared to the Potchefstroom Gokok chicken breed. This means that breed does have an effect on egg quality traits, and it may be used to improve the egg quality traits through selective breeding. Therefore, farmers can use the bushveld chicken breed in their breeding programs in order to improve the egg quality traits of their chickens. It is recommended that research be conducted to investigate the effect of breed on egg quality traits of other South African indigenous chickens in different regions of the country, since the current study focused on only two breeds, which is the bushveld as well as the Potchefstroom Gokok chicken. Research should also be conducted to study the influence of other factors, such as your age, your environment, genetics, as well as nutrition on egg quality traits of chickens. I would like to acknowledge my supervisors as well as the Department of Agricultural Economics and Animal Production at the University of Limpopo. These are some of the references that I used in my presentation. Thank you.
Thank you, Ms. Mohapa. Again, less than 7 minutes. I think I like this trend. We'll go home early if things continue this way. Okay. Can we get hands? And please ask one question where you need a follow-up once we are done with the other speakers, then we'll come back for your follow-up. I'm not seeing hands. I think I should change my rules. Oh, Prof. Tad, you can ask as many questions as you want. I'm changing my rules for you.
Thank you, Chair, but I will stick to only one question. Prudence, you dwelled much on the effect of the breed on the egg quality attributes of the indigenous chickens. Are there extended studies relating to the genetics on the influence of the egg quality traits?
Most of the studies that I've seen focused on the effect of breed as well as age on egg quality traits. I haven't seen a lot of studies focusing on the genetics part. But then that's why I recommended that research be conducted to investigate genetics on egg quality traits.
Okay, sorry, Chair, this is a follow-up. How is the extent of heritability of egg quality traits that you are talking about? This could also relate to the genetics, the question that I'm asking about. The extent of heritability of the egg quality traits.
May you please clarify your question?
I'm going to rephrase again and say, what is the heritability of egg quality traits in the indigenous breeds? I'm asking that because this is the basis of selection as you have been advocating for.
Okay. We conducted this study in order to investigate these egg quality traits so that we can be able to breed these chickens so they can pass on the genes in terms of egg quality to the coming generation. So if they have desirable egg quality traits, this means that they can be bred with other breeds in order to produce breeds that have better or desirable egg quality traits.
Okay, thank you, Prudence. Before we clap hands for you, does it mean now I can shift from the hard body I don't like and eat the eggs? Okay.
Yes, definitely.
That's the good news. That's the applicability of the study that I'm taking away. Okay, our next, oh yes, let's clap hands, please. Our next speaker is Charity Makaleng, and then she's going to be speaking to us about Bonsmara beef cattle. So from hard body to beef cattle. Oh yeah. As she comes up, can we please clap hands?
Greetings to you all. My name is Charity Makaling, and today I'll be sharing you the findings of my first objective as a PhD student, titled Evaluation of Growth Performance of Stud Bonsmara Beef Cattle Across Generations and Agroecological Zones in South Africa. Bonsmara cattle is a locally developed breed that is being developed to thrive under different climatic conditions in South Africa. The Bonsmara breed, it's very well adapted to high adaptability, fertility, and beef yield. However, despite the importance of the breed contributing about 50 to 60% of the beef production in South Africa, the gene pool of the breed over the generations has been declining due to change in farming practices such as enhancement in technologies, breeding strategies, crossbreeding management practices, and overall climate change. Therefore, any change in the gene pool can lead to a drastic change in the genetic diversity of the breed, leading to inbreeding depression, overall resulting in reduced performance, adaptability, and overall productivity. Therefore, the study finds that it was important to determine or analyze the effect of agroecological zones on the growth performance of those stud breeders across different generations. The slide shows an overall geographic regions in South Africa with their respective climate, land forms, soil types, vegetation, and biomes. And figure 1 just shows the demarcation of where those agroecological zones are demarcated. The study focused on the stud Bonsmara of beef cattle raised under different standard husbandry practices in accordance with the Breeder Society, looking at the performance of the animal or looking at the vaccination, proper vaccination programs, deworming, deeping, feeding regimes, housing, and breeding. The study focused on the weight of the animal at birth, weaning, yearling, 18 months, and 24 months. The study used an observational longitudinal study design to compile the study. For data analysis, a purposive sampling method was employed across all agroecological zones and generations, deriving a pedigree information consisting of animal ID, sire ID, date of birth, the sex of the animal, the class, the region, also the live weights. The software that was used to conduct the study for data cleaning and editing, it was Microsoft Excel and Minitab, a version 18.1. An Android 4.2 software was used for determination of generational intervals. General linear model and ANOVA was used for least square estimation, and also Tukey HSD at 5% level was used for mean separation. And the model that was used is depicted on the slides. The study considered factor levels and variables in the study. It used a population sample size of 606,079 bulls and cows of Bonsmara cattle from looking at factor levels such as the year, the birth, the province, generation, and the agroecological zones. And the records that were recorded are the ones that have been previously mentioned. The results shows the growth parameters of the Bonsmara breed across these generations. As you can see highlighted with the blue marker, we can see that the Bonsmara animals that were raised in the arid regions performed low for almost all the growth parameters in birth weight, weaning weight, yearling weight, 18-month weight, and 24-month weight. This may be due to the warmer climatic conditions in the arid environments where the breed is susceptible to diseases and tick diseases that are caused by ticks, ultimately affecting the performance of the breed. As you can see, the dry humid regions performed well at the earlier stage of the animal, when the animal reached 18 months old and also 24 months old. And also, in the humid region, we found that the birth weight of the breed was very good in performance. This may be due to different management practices and also may be due to the fact that as season changes, there is high humidity in the humid regions, enable it good forage for the breed to eat and then so that it can grow better. Thank you. And then this slide shows the trends in overall production of the Bonsmara breed across different generations. As you can see on figure 2, 3, 4, and 5, we can see that the growth rate of the Bonsmara breed is increasing over generation 1, 2. However, in the third generation, it reaches its peak and start declining. And then the severe declining, it's mostly visible on the weaning weight. This may be due to factors such as inbreeding depression on the breed at an earlier age. However, as you can see on the 18-month weight, we can see that the breeding, it increases, the breeding performance increases on the 6 generations on the 18-month. This may be due to a feed lot within that generation because as the animal is being introduced to feed lotting. This slide shows the effect of these different factor levels on the several weight, such as the weaning weight, yearling weight, 18-month weight, and 24. So figure 6, it shows the effect of these factor levels, season, province, and agroecological zones on the weaning weight using principal component analysis. So it shows that season had a severe effect on the weaning weight of the animal as shown by the line here. So, however, on the yearling weight, we can see that season has a severe effect on the weight. However, it's not as severe as the one in weaning. That goes back to the previous slides that shared that weaning weight was severely affected by agroecological zones. The same thing can be said for the plot on the 18-month weight and the 24-month weight. We can see that agroecological zones has an effect on the 18-month-year-old. However, it was not as severe as the season. That means changes in season severely affects the weaning weight and the 18-month weight of the animals. So in discussion, we can look at the animals' environmental adaptability to these climatic environments and also the genetic adaptability to these climatic environments. As been said, that the arid zone showed the arid zone shows less performance in the early stage. However, in the later stage, it was better. So we can say that the animal is severely affected by the different physiological stage. In the early stages, weaning weight is decreasing, and then in the later stage, we can see that the 24-month-old, the weaning weight is increasing. As implications of the study is in line with that of Versace, that in 2017, that stated that the influence on the performance of the animal depend on the physiological growth of the animal. At weaning, the animal is more susceptible to diseases, and the animal needs to adapt to environment to show that it's growing properly. Therefore, it's essential for farmers to clearly define their breeding objectives. Are they breeding to sell at weaning, or are they breeding for breeding purposes? So it's important for that prior to the farmers or breeders raising their animal at those selected different agroecological zones, it's important for them to define what are they breeding for. And then in terms of genetic adaptability, the Bonsmara exhibited positive growth trends over the generation, even though on the weaning weight over generation, it decreases. This shows there's a need to determine the inbreeding depression, which may be a severe cause of the decline in the weaning weight of the animal. A take-home message is that growth performance severely is affected by different agroecological zones. So therefore, there's a need to continue monitoring those growth traits across generations over time so that we can find a way to identify opportunities to reduce effects such as inbreeding depression. Furthermore, I would like to acknowledge the following institution, private and public, and the people who have funded my research. With that, these are the references that I used for the study, and I thank you.
Thank you for not stealing my time, Ms. Makaleng. Very close to 10 minutes, and that's how I like it. Can we get hands for questions, please? Okay, there's one hand here. Okay, there's a second hand there.
Okay, I will start.
Sorry, is it possible to start that side, and then we'll come back to you, Mr. Mpai. I'll give you as much time as you need.
Thanks for the great presentation. I just want to make sure, did you use phenotypes like performance data, or did you use estimated breeding values? And if you use performance data, did you somehow account for G by E interactions, or then somehow account for the impact that the environmental differences between the agroecological zones would have on the performance?
We've used, okay, can I have the question again?
I missed it somewhere. I don't know if I'm too tired at the moment, but I'm just asking, did you use performance data? So was it actual phenotype, so the kilogram performance data of the weights, or did you use estimated breeding values? And if you used performance data, did you account for the genotype or environmental interactions or the environmental impact that the different agroecological zones may have?
Okay, so this is the findings from my first objective, which is a PhD study. The first objective used the performance data as raw as it is. However, going forth, the study is going to, on the second or the fourth objective, is going to look at the phenotypic data of the study to improve the accuracy of the state of the study. Yeah.
Okay, thanks. And then is there a certain agroecological zone that you would recommend the Bonsmara is the best for, following the initial results?
Yes. Looking at the results, we can see that the Bonsmara is very adaptable to most of the agroecological zones. However, in the humid region, in the arid region, it's the region that shows less performance. So I would say, if you would really want to breed with the breed, look at your breeding objectives. For instance, if you want to breed for production or for breeding purposes, to raise the calves, you must breed at the humid regions, because there is a high humidity, which of course it increases vegetation or availability of forages. So overall, the Bonsmara breed is very highly adaptable. However, but then in the arid regions, the performance is less compared to other regions. However, it's very much adaptable to most. Yeah.
Okay, thank you. Mine is going to be a follow-up to what you just answered. Okay. So let's say I'm a stubborn farmer in humid region. So, and I want to improve the performance of my stud Bonsmara in later stages. Your yearling weight, your 18-month weight, and 24. So what are some of the recommendations, management practice-wise, that you can give me?
Okay, management practice-wise, I think you should, hence I mentioned mostly the importance of breeding for, different objectives. If you want to breed for increasing probably the meat or looking at the high yield, proper strategies such as better management strategies, for instance, the type of feed that you feed your animal, the type of environment that you expose your animal to. For instance, are your animals walking too long to reach the feed, or they're not, or the fact that they're grazing? So I think if you really want to improve your production, one of the key important, I think, is better records. Record your animals better so that you can know which one is growing better, which one is not, and then you can cull the ones that are not. So yeah, implement all those better management practices, such as dipping your animals, making sure that they're vaccinated, making sure that you do daily checkups on them, and then you check their growth, you monitor everything. So I think the most important thing is just to monitor your animals. And then as a farmer or a breeder, then you can decide which ones are growing and then which ones are not. And also supplementing your animals, of course.
Okay, thank you, Ms. Makaleng. When you were busy talking about the number of bulls and cows there, I heard some Jacob Zuma numbers that you were saying. But thank you so much, and let's clap hands for Ms. Makaleng. The last speaker, sorry. The last speaker for this session. After this session, then we'll get a new chair, you'll get into a new graveyard slot, is Fano Makoka. Is that person around, or am I getting an early break? Okay. Going once, going twice, going, gone. Okay, then it means that this is the close of this session, and the next chairs, which I think should be coming up, Dr. Mchech, is it you? Okay. Then I in the famous words of Dr. Lefpan, I am released. Let's clap hands for Dr. Mchech.
Good afternoon, colleagues, and to our presenters. Welcome to the very last presentation session for our 3 days research day event. And let me check with the presenters. Have you all loaded the slides? If not, please proceed and load your presentations. As Dr. Hadebe has indicated, I'm Lesego Motshekga, and I'm going to co-chair this very last presentation session with Dr. Sateke. Hi, Doctor. Then we have Dr. Kopa. Let me convey the sincere apologies for her. She couldn't be with us due to other administrative duties that she's currently undertaking. All right, I'm going to observe the same protocols that Dr. Hadebe has said, 10 minutes presentations and one question at least, unless if time permits, then we are going to entertain the second question. Let me proceed to our presenters. For this session, we have about 12, and I'm going to usher in the first 6, then Dr. Sateke will continue with the last 6. The first one is Rito Matebula, who will be giving us a talk on investigating rangeland vegetation dynamics in selected semi-arid regions. Can we welcome him with a round of applause? Round of applause to chair him.
Greetings to all. I'm Mateo Lareto. I'll be taking you through my study, which is titled Investigating Vegetation Dynamics Along Distance Gradients in a Semi-Arid Rangeland of Capricorn District, Limpopo Province, South Africa, being supervised by Dr. B. Gusha and Dr. L.M. Motshekga. Rangelands are known to occupy on about 40 to 50% of the Earth's terrestrial surface. They consist of grasses, shrubs, and forbs. However, in South Africa, rangelands are often viewed as unsuitable for meeting the needs of livestock production, even though the reasons for the changes observed in vegetation over the time are complex and have risen from the combination of climate-related factors, such as land use. In rural households, concentrated human activities such as livestock grazing near homesteads continue to exert direct pressure. These activities then reduce grass cover, alter species composition. They then encourage the encroachment of less palatable species. Most of the studies have concentrated on land degradation and soil disturbances around watering point. However, our study aimed at assessing the vegetation dynamics along distance gradients from homesteads. Shown on the picture, figure 0.2, it's a communal rangeland. It's a picture of a communal rangeland of livestock grazing nearby homesteads. Our study was conducted at Tsima Village, Capricorn District, at Limpopo Village, at Limpopo Province, South Africa, and the climate of the region is a semi-arid. Then the biome is known to be savanna at the lower field bioregion. The vegetation types of the area are known to be the Polokwane Plateau bushfield, and they are also associated with the Mamabolo Mountain bushfield. As we move to our data collection, a total of 400-meter transects were established at distance 0, 100, 200, and 300. Within those distant gradients, plots were then established, which are a 10 by 10 meter plot at each gradient distance, of which all the vegetations which were contained were sampled. Then, as for woody composition, a 2-meter rod was used to measure the lowest breathable height, the height, and also the short canopy diameter. For herbaceous composition, a 0.5 by 0.5 quadrat was placed 5 times randomly inside the plot. Then all the herbaceous species were then harvested, then taken out for biomass estimation. Then for basal cover and equipment, a levee bridge was then used. Then a rod was dropped through the 10 holes which were contained in the levee bridge. Then if the rod strike the vegetation tuft, it was recorded as a strike. Then if the rod touches the ground, it was then recorded as a non-strike. Then all the proportion of the 9 strikes were then used to estimate the basal cover. So in terms of data analysis of species richness and diversity, they followed the Mahinics for richness, then the Shannon winner for diversity, the Pilos evenness, then the software which the R version was also used, then the independent test was used to assess the effect of distance on vegetation composition and also the biomass at significant level at P less than 0.05. As we move to our result of woody species composition, at distance 0, we find that the woody species which were prevalent or dominant were the Aluphorex and the Optionia ficus indica. Then as we move on from the distance from 100 to 200 to 300, we have seen that, we observed that the species such as Bacterial robusta and Dichrostrasis cinerea were starting to be more prevalent. So if you see that, if you see as the results I've indicated, the Aluphorex and the Optionia ficus indica are known as cactus species. Cactus species are known as species which are well adapted in environments which are disturbed, whereas Bacterial robustas and the Dichrostrasis cinerea are species which are known to be prevalent in encroaching areas. This suggests that, according to our finding, this suggests that even though the species which were in the disturbed area, there were still species which were tolerant to the disturbed area. However, as we move along the distance, The species which were more prevalent were the encroaching species. Looking at this figure, the figure shows the tree density, the tree height, and the lowest breathable height. Then, in terms of the tree density, there was no significant difference in terms of the tree density, moving to the adjacent, and the near, and the mid, and the far. However, there was a significant difference in terms of tree height. And also there was a significant difference in terms of the lowest breathable height. According to our findings, this suggests that the effect of selective harvesting as well as selective browsing played a huge role in terms of how the people in the communal area were utilizing the woody species. Then as we move to the longest crown diameter, there was a significant difference in terms of the longest crown diameter, and there was no significant difference in terms of the shortest crown diameter, as well as the tree crown diameter. However, according to our findings, we have found that due to selection by livestock and selection by human harvesting, this also had played a huge impact because, as you can see that, for instance, in the shortest crown diameter, at the adjacent next to the household, it was low, but as one moved to the far distance, it tended to be high. In contrast with the tree crown diameter, with the tree crown area, at far it became low. However, even at the adjacent next to the household proximity, it was low. Then in terms of the wood diversity indexes, our richness was slightly slow at the adjacent and became more lower until the mid, then started to be high. This shows that the selective harvesting of fuel wood and the selective browsing of animals played a huge role at the proximity of household as compared to the far distance. However, it's in contrast with the findings of the diversity. Our study has shown that besides having lower richness, but in terms of diversity, at proximity of household, the diversity was high, and however, it tended to be higher again at the mid and the far. The evenness, the same thing, it followed the same trend whereby it was higher at the next to the household, then it becomes lower a little bit at near, then becomes higher again at the mid, then it becomes higher again at the far distances. Our findings suggest that there's no effect which can cause which can play a role in terms of the disturbances. However, meaning that we can still find the highest diversity and the evenness, even though there are disturbances within the communal area. As we move to the table 2, which shows the species composition and the ecological characteristics, it has shown that Aristida ranocloa was more prevalent from distance 0 until 100 and Congesta. However, some of the species which were Temenda triandra and Chloris variegalta were low in abundance. As we move to the diversity of as we move to the diversity indices, the richness was higher and was lower as was higher at far, then became higher again at far again. In terms of grass biomass, the biomass was lower at adjacent and became higher again. The forbs were lower, that the basal cover became higher again. Then when we compare that, is that there's no effect of which we can see that the grass biomass was lower, but however, we have seen the highest basal cover, which means that having a lower basal cover, it doesn't entail that you may Having a lower grass biomass doesn't entail that you will have a lower basal cover. In conclusion, our findings suggest that there was a shift in terms of moving from homesteads, and the disturbance-tolerant species dominated closer to households, such as Aluphorex and Vachelia robusta, and Dicostrasis scenaria dominated at far. And vegetation structure was more affected than density. Diversity and evenness did rise as one moved away from the distance. However, heavy grazing favored Aristida species, which are pioneer species, which then reduced the palatable species. We also recommend that sustainable land use can be called upon, which can support and the participation of youth and women in terms of giving out the knowledge of vegetation dynamics. I would like to thank my supervisors and Mr. M. Kasheka, University of Limpopo, and the Limpopo Department of Agriculture. These are the references. Thank you.
Okay, thank you, Rido. Question time. Can we entertain questions from the floor? Questions, please. There's one question over there.
Thank you, Mateula, for a good presentation. I just want clarity on the data that you used to draw your whisker plots, the graphs, vis-a-vis the statistics used, those letters. Which data did you use to plot those graphs?
I used the ARS software.
Data, is it a mean, median?
Or I used the mean and median.
Yeah, that's where I'm getting lost. You use both?
Yes, I used both.
How?
Or I was comparing the mean and the median, and also looking at the highest and the lowest in terms of the box and whisker.
Okay, I don't think I'm... Because my assumption was that you would have used the median to plot your whisker plots, isn't it? And then how did you end up having those letters, statistics, when median was used? That's where I'm getting lost.
The letters were there to show the significant differences.
Between the means?
Yes.
Okay, I won't go further, thanks.
Thank you for the question. Without any question, for the interest of time, let me release the presenter. Thank you very much. A round of applause to Mr. Matebula, as we are also ushering in Mobe. Mobe, are you with us? Okay. Mobe will be talking to us on an exploration of socio-economic drivers of adoption of climate smart agriculture among Mankweng spinach farmers. A round of applause to Mobe.
Thank you, Chair.
Thank you. They say a great conversation begins with a meaningful greeting. So, Chairperson, members of the panel, and fellow researchers, I greet you with gratitude and respect. For those who don't know me, my name is Mobitesia Msibudi, a master's student under the School of Agriculture and Environmental Sciences, and I'll be presenting my honor study, which was under the supervision of Dr. Elgil Dwaba. Much appreciated. So in the midst of climate change, innovative solutions are crucial to ensure food security and agricultural resilience. Climate-smart agriculture, which aligns with Sustainable Development Goal number 2 of Zero Hunger and Sustainable Development Goal number 13 of climate action, is actually a viable solution. But the question is, who adopted it and who did not? As shown on the screen, my study focuses on the socio-economic drivers of adoption of climate-smart agriculture, CSA in short, among smallholder spinach farmers in Mankweng. So without wasting any time, let's get to it. Every solution begins with a challenge. So for us to improve adoption of CSA, we first have to tackle the problem at hand. Information about adoption of CSA in Mankweng was very scanty. However, smallholder spinach farming in Mankweng was still vulnerable to climate change. So there could be adoption of CSA. And this low adoption of CSA could be a result of lack of access to credit, CSA knowledge, and CSA farm inputs. And before collecting data, I thought adoption of CSA among smallholder spinach farmers in Mankweng could rise if climate change policies are flexible and tailored from local to economic level. Every study has a purpose, but before we go to the purpose, a question may say, why spinach or why CSA? Spinach is cheaper to plant and cheaper to buy, targeting two pillars of food security, namely food availability and food accessibility. Also, spinach is a very vulnerable leafy vegetable as compared to others to climate change. CSA ensures agricultural resilience or reduce the responsiveness of agriculture to climate change, I should say. So CSA practices are nothing complex. We are talking about crop rotation, intercropping, interspacing, efficient irrigation, application of manure, mulching, or yes. So going back to the purpose, this study was motivated by the need to facilitate and provide insights of adoption of CSA by smallholder spinach farming in Mankweng to increase resilience, productivity, market access, and income level. Hence, this study was held at Mankweng, a semi-arid climate with high precipitation in summer as compared to winter. Also an area where agriculture is one of the income-generating activities. This study aimed to analyze the socio-economic factors influencing adoption of CSA by smallholder spinach farmers in Mankweng. It had two objectives. The first one, to profile socio-economic characteristics of smallholder spinach farmers in Mankweng. The second one, to analyze the relationship of those farmers' characteristics with their adoption of CSA. The null hypothesis, socio-economic factors do not have an influence on adoption of CSA. Moving on to research methodology and analytical techniques, this study took place in Mankweng, given the coordinates whereby 65 smallholder spinach farmers were sampled using snowball and judgmental sampling techniques to collect primary data using structured questionnaire. The first and second objectives were addressed using descriptive statistics and binary logistic regression model, respectively. This was the specified model where P is the probability, B0 is the constant, B1 to B10 are the coefficients, and UI is the error term. The table of variables. The most important part, the results. So we'll start with the descriptive results. These results were prepared by the statistical packaging of the social sciences, and they read as follows. So from the 65 sampled spinach farmers in Mankweng, the dominant gender was female at 65.5%. The most at 60% were having secondary education as their highest, and men were married at 47.3%, followed by the single ones. We also noted that only 34.5% adopted CSA practices, and only 9.1% had access to credit, only 43.6% had access to extension services, only 40% had access to CSA information, only 12.7% had access to CSA farm inputs, while on the other hand, only 32.7% had experience of climate shock. We also noted that the ages of the farmers were ranging from 27 years to 73 years old, with a mean score of 51, which is approximately 51 years, at 11.848 standard deviation, showing medium variability in the ages of the farmers. The farm incomes were ranging from 500 rands to 6,400 rands, with a mean score of 2,263.60 cents at 970.56 standard deviation, showing high variability in the farm incomes. And then from the 34.5% of the people who adopted CSA, we noted that they adopted 1 to 8 practices with a mean score of 2.95 and a standard deviation of 2.064. And then from the 43.6% of the people who had access to extension visits, we note that they only received 0 to 5 visits per month with a mean score of 1.5 at 1.142 standard deviation, showing high variability in the number of visits received per month. So I want you to ask yourself, how much CSA farm input do you think the farmer received from 1 to 2 visits if extension visits are not only for teaching CSA? We are now on the binary logistic regression results, and the table presents. From the 10 observed independent variables, we note that only 5 were found to be significant, namely gender, farm income, access to credit, access to extension visits, and access to CSA farm inputs. And then the model above the table showed the relationship between those significant socioeconomic drivers and the probability of adopting CSA. So taking a closer look, we see that gender has a negative influence on adoption of CSA, meaning that an additional female among the farmers will reduce the probability of adopting CSA. And then farm income has a positive influence, meaning that if the farm incomes increase, then the probability of adopting will also increase. Then access to credit has a negative influence, showing that the more they have access to credit, or the more they take credit, the more the probability of adopting decreases. This is to say that the people around our alma mater are afraid to take credit and invest in something new they don't know much about. So access to extension visits has a positive influence, showing that the more they receive visits, is the more the probability of adopting will increase. And then access to CSA farm inputs have a positive influence, showing that if they receive more CSA farm inputs, then the probability of adopting will increase. Going to the model summary, we note a Nagel-Gerach R-square of 0.573, meaning that 57.3% of the variation in the probability of adopting CSA is explained by those significant socioeconomic drivers. So this is to say that this model is fit to predict the probability of adopting CSA. In conclusion, this study rejects the null hypothesis. Why? Because gender, farm income, access to credit, access to extension visits, and access to CSA farm inputs had a significant influence on adoption of CSA. Adoption of CSA among smallholder spinach farmers in Mankweng is relatively low. So this study recommends local organizations to engage to ensure programs respond to unique challenges faced by farmers. There should be provision of incentives for adopting CSA among small spinach farmers. And then the quantity and quality of extension visits should be increased. This study was shaped by proper research, and these are the key sources that shaped this presentation. So this is my take-home message from my academic background. Hacemos el camino caminandos, meaning we make a path by walking. We is me and you. Remember, it doesn't take one step by one person to open a path. Pay attention, it doesn't take one step by one person to reach our destination. Just as Prof. Signolo once taught us, development is an ongoing process. Let us unite and be consistent in finding solutions for Africa through research.
Thank you very much, Mobi, for a very interesting talk. Question time. Can we have questions from the floor?
Thank you for the wonderful presentation, ma'am. So I have a question. So I heard you mentioning two of the Sustainable Development Goals. So can you please explain what they advocate for and how they align with your study? Thank you.
Thank you for that question. Sustainable Development Goal number 2 of Zero Hunger seeks to end hunger, improve food security and nutrition, and then ensure sustainable agriculture. So my study aligns more with specific target 2.3, which seeks to double agricultural productivity and income level of smallholders, peanut farmers. And it also aligns with a specific target 2.4, which seeks to ensure sustainable food production systems and implement resilient agricultural practices. So, and then Sustainable Development Goal number 13 of climate action seeks to take urgent action to combat climate change and its manifestations. And then my study aligns with specific target 2.3, 13.2, yes, 13.2, which seeks to integrate climate change measures into the national policies, strategies, and planning. So like I said, my study promotes improving productivity, income level, and resilience. Thank you.
Thank you very much for the presentation and the question. You are free, my dear. Thank you. Our next presenter is Priti Rachweni. Let's welcome her on the stage. Rachweni will be presenting phenotypic relationships and predictive modeling of total skin length in the Nile crocodile.
I'm going to try to keep this short, like my height. Greetings to the House at large. My name is Priti Rachweni, here to present my findings on phenotypic relationships and predictive modeling of total skin length in the South African Nile crocodile, also scientifically known as Crocodilus niloticus subspecies corviae. The Nile crocodile is one of the most commonly farmed with crocodilian species in South Africa, with its contribution making about 30% to the global skin trade. The crocodiles are kept for the production of skins and meat, with the production of skins contributing about 80% to the total crocodile value, and the 20% attributed to the meat, crocodile oil, souvenirs, and crocodile blood capsules, and even other products. The Nile crocodile skin is ideal for heavy-duty products, including belts and shoes, even furniture, handbags, and traveling bags. However, the crocodile industry is faced with the challenge of producing skins of inferior quality and poor yield, which then means that farmers incur profit losses. It has been stated that the market price of crocodile skins is greatly influenced by the quality, the width, and the length. However, its preference for certain crocodile skin sizes is typically dependent on the intended use. Some industries, like the garment industry, usually prefer larger skins. However, there is no information on the model that can be used to predict total skin length from measurable skin traits of Nile crocodile. Hence, the objectives of this study were to determine the phenotypic correlation between total skin length and related measurable skin traits of Nile crocodile, as well as to establish a model for the prediction of total skin length. And the hypotheses are in line with the objectives. The study was conducted at Lalele Crocodile Farm in Mudimullo Mokopong municipality, whereby a total of 180 skins from 180 Nile crocodiles aged on an average of 35 months old were used. The 180 skins from 180 crocodiles just implies that we used whole skins. A cross-sectional study design and a census approach were used, whereby the cross-sectional study design implies that only the data was collected only once, and the census approach means that we used all the available skins. Ethical clearance was acquired from the University of Limpopo Animal Research Ethics Committee, and that is the project number, and slaughtering was done at Tabagwena Abattoir, which is a certified veterinary institute that exports meats to the European countries. This diagram just shows how data was collected. In the middle upper figure, it shows how the skin traits were measured using a measuring tape, and how the skin thickness was done using a vernier caliper, and the skin weight was measured using a weighing scale. And at that corner is just me, very, very happy that I'm still alive. The statistical package for the social sciences, SPSS of 2022 version 29.0 was used to analyze the data, and Pearson's correlation was used to determine the phenotypic relationship, while simple linear regression and stepwise multiple regression analysis were used for predictive modeling. This table shows the correlation coefficient values between total skin length and measurable skin traits of Nile crocodile. If we look at the part highlighted in red, we can see that skin thickness showed the lowest but positive correlation with total skin length. And TTL, which is the part highlighted in green, shows the highest and significant correlation with total skin length. This implies that any changes to TTL may also cause a change in TSL in the same direction. This table shows the simple linear regression models for the prediction of total skin length from skin traits of Nile crocodiles, whereby if we look at the highlighted part, we can see that TTL, which was very, very highly correlated in the previous diagram, also had the highest coefficient of determination, contributing to 76% of the variation in total skin length. Similarly, this is the figure that just shows TTL again. You see that R squared is 0.76. And then we can see that the data is clustered at the regression line, meaning that the relationship was linear and positive. Table 3 shows the stepwise multiple regression model for the prediction of total skin length from measurable skin traits of Nile crocodile, whereby the first model, which included all the measured skin traits, was the best in predicting total skin length, contributing to 90% in total skin length variation. Model 2 and Model 4 were also very high with the coefficient of determination values, meaning that they can also be possible alternate predictors of total skin length. With the first objective, there was lack of literature, so the current study did not discuss this objective. So my fellow researchers in animal science, this is your time to shine. With the second objective, several studies have reported on the prediction of length measurements. By length measurements, I'm referring to measurements such as the total length and the snout to vent length from morphometric data. This includes the studies published in 2011, 2013, and 2021, which showed that head length was one of the significant traits in predicting total body length of the mentioned breeds of crocodiles. Furthermore, according to a study published in 2012, the combination of ventral length and ventral width, which is basically just belly length and belly width in my case, was also the best in predicting total length of spectacled caiman. The current study concluded that TSL had a positive relationship with all the measured traits, and this means that improving these traits may subsequently improve total skin length. The study also concluded that the total skin length as a single trait could be the best predictor of total skin length. Breeders may use this trait to predict total skin length given that it is simple to measure. However, all the other traits as a combination can also be best predictors. Further research can be done on the phenotypic, as you've seen by the lack of literature, and also the genetic correlation using crocodiles of varying ages. I would like to thank the following institutions for their contribution with the research infrastructure for the current study. As they say in crocodilian language, in a wild crocodile. Thank you.
Thank you very much for Rachweni's presentation. She's very brave, working on crocodiles. Oh, can we have questions? We need at least one question from the floor. Can we please entertain the crocodile lady? One question, please. There's a question over there.
Thank you very much. Such a wonderful presentation. Just a quick one. How to apply your results for crocodile breeding program?
Oh, okay. I believe as researchers, we have the responsibility to go back to the farmers. So if we can go back, from my results, we can advise that they select for for crocodiles with longer tails. As we've seen that the total tail length was the one that contributes the highest to the variation in total skin length, meaning that the other factors are contributing very less. So if farmers were to select for crocodiles with longer tails or for skins with longer tails, then they will have skins with higher yields, thus mitigating the problem of poor yield. Thank you.
Thank you very much. I saw another question, but for the interest of time, please interact with her after the session. Accepted. Thank you very much. All right. Our next presenter, Kagiso Diale, who will be sharing information on inbreeding depression in productivity of Toggenburg dairy goats of South Africa. Diale, Kagiso, are you here? Okay, it seems like Diale is absent. We are going to proceed to Priska Masemola. Priska, are you here? Okay, Priska will be sharing with us soil amendments with cover crops improves amaranthus yield and reduces the severity of soil-borne fungal diseases. Can we welcome her with a round of applause?
Thank you, Doctor. Greetings, everyone. My name is Masemola Priska. I'm a master's student, and I'm going to present my ongoing research titled Soil Amendments with Cover Crops Reduces the Severity of Soil-Borne Fungal Diseases and Improves Amaranthus Yield, supervised by Prof. Kena and Prof. Ice. Amaranthus is a widely cultivated species belonging to Amaranthaceae family. These plants have been known for its high nutritional value, and then this plant is also easily accessible. Therefore, however, there is a challenge with the production of amaranthus yield due to soil-borne fungal diseases, Fusarium oxysporum and Rhizoctonia solani, which causes the diseases such as Fusarium wilt and Rhizoctonia root rot. Therefore, different approaches have been proposed in agricultural fields whereby they use synthetic fungicides and other chemicals to suppress these diseases. However, there have been reports whereby these chemical fungicides lead to fungicides resistance and then environmental degradation. As such, my study therefore aims to propose an alternative method of using the cover crops as a sustainable approach for eradicating these diseases. Cover crops are those crops which are usually grown for beyond primary gain. They are grown for the purpose of fertilizing the soil and then suppressing the diseases. Cover crops, they have an advantage of attracting the beneficial microorganisms and therefore outcompeting the toxic microorganisms. Therefore, the amendments of legumes and grass cover crops are going to be studied in this research. Therefore, the usage of cover crops, cover crops have been used in extensive research for the suppression of soil-borne fungal diseases. However, there's no study whereby the cover crops are used for the suppression of soil-borne fungal diseases in amaranthus. Therefore, the study aims to investigate the effects of cover crop amendments on soil-borne fungal diseases and improve amaranthus yield. The objective of the study is to evaluate the effects of cover crop amendments on fusarium wilt and rhizoctonia root rot severity of amaranthus under the greenhouse conditions. Therefore, the phytopathogens aforementioned were prepared under the laboratory whereby the pure cultures of these two pathogens were prepared using the potato dextrose agar for 7 days at the temperature of 28 degrees Celsius. And it was replicated 10 times using the CRG method. In preparation of the cover crops and the soil inoculation, the cover crops were terminated from already established fields, and they were ground to powder using the laboratory grinder. And then the 1 kilogram of the soil with the amendment of AGRO mix at 3:1 was used. Therefore, the mixture of the soil with AGRO mix was amended with the cover crops, and then they were left for a week for the soil and then it was amended with the 10 gram of the crushed cover crops. And therefore the fungal strains that was prepared under the laboratory were mixed with the amended soil mixture, and then they were left for 2 weeks to colonize the soil. And then the study was replicated 3 times using the RCBD method. Then for disease suppression in the greenhouse, for the assessment of the damping off, the post-emergence damping off, as well as the fusarium wilt were assessed at different days intervals. Starting with the pre-emergence damping off, it was assessed at 14 days to check for the damping off. And then for the rhizoctonia post-emergence damping off, it was assessed at 30 days, whereby we were checking for the root rot severity of the diseases. And for the fusarium oxysporum, it was also assessed using the scale method from 0 to 5. And in overall, for the post-emergence damping off, as well as the fusarium wilting, it was assessed using the method by Salman and Boeras 2023. And the ruler was used to measure for the roots and the heights of the shoots. Representing under the results, it's the graph showing the different cover crop amendments, whereby we were checking for the damping off severity, and we can see that there was lowest severity of oats, lowest severity of damping off under the oats cover crops, and then all 3 cover crops were significantly different from the rhizoctonia solani Solani treatment. Then coming to the disease severity, when we were looking at the Rhizoctonia root rot severity, we can see that clover and oats were not significantly different, and also stolen rye was different from both clover, oats, and Rhizoctonia solani treatments. But in overall, we can say that all three cover crops suppressed the severity of the Rhizoctonia solani root rot severity. Then when it comes to Amaranthus root height in centimeters, we can see that in terms of oats, it suppressed in terms of oats, we can see that the height was increased under the oats cover crops compared to the others. And then now moving on to the shoot height, we can also see that the similar trends occurred whereby oats also improved the shoots. Under the results, this is from the picture one, we can see the evidence of the Rhizoctonia damping off. By taking a closer look, we can see that there are 2 pods whereby there were no growth of any Amaranthus. This is the result of the damping off, but in terms of looking at other pods, the cover crops clearly improved the growth of Amaranthus. Then this is for the Fusarium wilting. We can see the yellowing in other pods, and then this is because of Fusarium oxysporum. And then here we can see the clear results when we are differentiating between Rhizoctonia and amendments of oats cover crops. I chose oats because it was the one which shows the highest suppression of the Rhizoctonia. And then therefore we can see that there is no there was a stunted growth under Rhizoctonia root rot of Amaranthus, but when it comes to oats, we can see that there was more development in terms of roots and the shoots development. Then in terms of discussion, we can say all cover crops clearly reduce the disease severity of Amaranthus plants. Then there was reports whereby they say Schoolman et al. Reported the Schoolman et al. Say that oats contains, they can release root exudation whereby they attract the beneficial microorganisms, and then also they result in more disease diversity in the soil, and therefore they outcompete the toxic microorganisms, and therefore subsequently increasing the roots and shoot height. And moreover, when it comes to the stolen rye reduced the Fusarium wilting, and then it was because of they released the secondary metabolites in the soil, which suppresses the amount of the Fusarium wilting. And therefore the data was analyzed using the statistical analysis system shown on the slide with mean separation using LSD at comparison test of 5%, and then the graphical representation of treatment means were generated using the GraphPad prism. And therefore in conclusion, we can say ultimately cover crops are sustainable, environmentally friendly approach for management of soil-borne fungal diseases. Notably, oats and stolen rye can effectively mitigate this Rhizoctonia solani and Fusarium wilting. Furthermore, this aligns with the Sustainable Development Goals 1 and 2. And in future research, research can be conducted to investigate the difference in fungal strains of these 2 pathogens. Furthermore, the genetic variations of these cover crops in suppressing specific pathogens. I would like to thank my supervisors and my mentor, Mr. M. Magagula, as well as Center for Global Change and ARC for funding. These are my references. Thank you.
Thank you very much, Priska, for a very nice presentation. Question time. Can we entertain questions for Priska? Okay, there's a question over there.
Okay, thank you. I heard you talking about cover crops, but I didn't hear you mentioning a specific one. Can you please mention? Okay. Yeah, you can go ahead.
Okay, I'm not sure if you missed it, but I mentioned the maize oats, stolen rye, and clover.
Can I add another question?
A
Or,
brief one,
What?
a brief question.
Oh, okay. What mechanism do these cover crops use in order to suppress this soil-borne disease? And are you sure that whatever result you got are because of those cover crops? Thank you.
Thank you for the question. Yes, I'm very sure that from different literatures where we saw that they have the allelopathic mechanisms for suppressing the diseases, and then they also, as I mentioned, the root exudates for suppressing the diseases. And then there was a different researches conducted from my supervisor's PhD students. They also conducted the research regarding the suppression of soil-borne fungal diseases.
Thank you very much. If you are still interested, Mr. Matiba, please engage with her after the session. Thank you very much, Priska. A round of applause to her. Let's welcome Carol Ramullo. Carol, are you in? All right, Carol is coming. She will be presenting the influence of household wastewater consumption on the health status of goats. Let's welcome her with a round of applause.
Good afternoon, everyone. My name is Ramullo Carol from the School of Agriculture and Environmental Science, and I'm under the supervision of Dr. Mdleje and Prof. Manyelo. And my presentation title is The Influence of Household Wastewater Consumption on the Health Status of Goats. This is my presentation outline. Household waters from domestic activities such as cooking, laundry, and washing is used as an alternative drinking water source for goats in rural areas facing water scarcity. However, this consumption has been linked to poor health outcomes, reduced body weight, lower body condition score, and diminished productivity in goats. Despite the adaptability, resilience on wastewater compromises the goats' welfare, block management, feeding strategies, rising long-term sustainability issues. However, the overall impact on goat health, productivity, and rural livelihood remains inconclusive and under-researched. The aim of this study is to evaluate the influence of household wastewater consumption on goat health. And the objective is to determine how the influence of household wastewater consumption on the objective of this study is to determine how wastewater influences the health status of goats. This was hypothesized that wastewater has no significant influence on goats' health statuses. Materials and methods of these studies. The study was conducted at Janfes in the two villages of Ra, Moreletsa, and Mudibeng. This is a comparative study, and it was based on a comparison of clean water and wastewater, and about 86 participants were used for this study respectively in those villages above, and the goats' availability and willingness of the owners was taken into consideration. The experimental design of this study and data collections, we used questionnaires, household demographics, water quality, seasonality, goat health, and also we use Sepedi just to make sure that they understand what we are asking, which is their home language. Statistical analysis of this data, I used SAS 2023 and chi-square test examined the relationship between household demographics and water quality, and the access was done on household perceptions on freshwater versus wastewater accessibilities. A general linear model was used on this study and binary logistic regression, and the model below is the one that was used to help with this data analysis. With the results, the socio-economic results indicated that goat keepers using wastewater were predominantly females and similar to non-water. The socio-economic results indicate that goat keepers using wastewater were predominantly females, similar to that non-water users with no significant gender difference. Age distribution was balanced in both groups, with the majority being 18 to 50 years. The non-wastewater had no significance and the difference was observed. However, education levels varied significantly, whereby households using wastewater had fewer individuals with no formal education compared to the non-water users with a higher proportion that attended secondary school. And the tertiary ones were also at a 17.7%, which was lower compared to the ones that were in secondary school. And the training showed that a highly significant difference, with 60% of non-water users have received training compared to those with wastewater users. And regarding livestock, goats ranked first as an important livestock amongst the villages, and then their wastewater usage was lower compared to the ones that was a non-wastewater use, which is fresh water. And then in terms of ranking the constraints on goats' productivity for goats consuming waste water and non-wastewater, it was observed that goats' productivity showed both water and non-water households identified that water availability as the most critical constraints, with a mean rank of 1.4 for wastewater users and also 1.24 for non-water users. This difference was statistically significant, highlighting that access to water remains the top challenge. Water quality followed as the second most important constraint, with the mean rank of 2.42 and 2.19, also showing a significant difference. Other constraints, including parasites, diseases, and also theft, were constantly ranked lower and did not show significant difference. This suggests that while health and security issues are relevant, water-related challenges are the primary limiting factor for goat productivity, especially in wastewater-dependent households. When it comes to ranking major causes of poor water quality on goat productivity for goats consuming waste and non-wastewater, the results showed that pathogens were the most critical factors affecting goat productivity, and also the results show that pathogens were the most critical factors affecting goat productivity for both wastewater and freshwater, with a significant lower mean ranked amongst wastewater being 1.4 compared to wastewater being compared to non-fresh water being 1.83, indicating a stronger association. Food crops were ranked second in wastewater and third in non-wastewater. Although the difference was not significant, hair was ranked third for waste and second for non-wastewater. Also not significant, the thing that was also not significant was detergent. However, it showed a significance different, ranking fourth in both groups with a poor water quality and statistical significance effect. And when it comes to ranking the seasons for both water quality on goat productivity for health, consuming waste and non-waste, the results show that ranking the seasons with poor water quality goat productivity shows difference between households using water and those that were not using wastewater. For wastewater using households, the cool dry seasons was ranked the most, and also the hot dry season was not at its highest. And then the rainy season's results were not significant along with the post-rainy season. And when ranking for major water sources for goat productivity and goat consuming waste and non-waste water, the water sources that were used were grey water, fresh water, and borehole, and it was found that the grey water is not significant compared to fresh water, and the grey water is significant compared to fresh water and borehole water. And then when it comes to ranking for major waste feeding supplements on goat productivity for goat consuming waste and non-waste water, the sources that were taken into consideration were laundry, hand basin, and kitchen, and only laundry was found as a non-significant factor, whereas the hand and kitchen laundry was found as a significant factor, and the hand basin and kitchen were found as the non-significant factors. And then ranking the health status for consuming wastewater, it was found that in food intake, water intake, and weight loss, there was a significant difference, unlike in the body condition score and the diarrhea, where there was no significance, and wastewater and non-wastewater ranks are shown in the table as it's presented on the slide. And then when it comes to the odds of ratio estimates, lower and upper confidential interval of households using wastewater and those using non-wastewater, as shown in the slide, the predictions show that training and education were non-significant, whereas water sources were significant, whereas water sources were not significant. Water intake and weight loss were significant. In conclusion, higher adoption is observed among farmers lacking education and training, and wastewater is strongly linked to poor health, which means that it will have long-term effect on the productivity and welfare of goats. And as a recommendation, I recommend educational initiatives, water security methods, and more research to be done on wastewater when it comes to consumption for goats.
Thank you very much, Ramullo. Question time. Can we have questions, at least one for Carol? There's a hand at the back.
Thank you for the nice presentation, Carol. Mine is simple. I want to know, how does your study align with the theme?
Okay, thank you for the question. The theme is from lab to market, and this is a continuous study whereby the findings will be used to inform farmers of how wastewater can affect their goals in terms of productivity, because we aim to ensure that farmers do not lose any more livestock, and the results from the lab using this continuous research will help them reach their target market and sell and feed their households.
Thank you very much, Carol. For the interest of time, if you have other questions or comments, please take it outside. And thank you very much. I have come to the end of my chairing role. For this very last session, I'm going to hand over to Dr. Sateke. Thank you for being good audience and presenters. Over to you.
Good afternoon. I see that the
there is one adjudicator who is not here. I mean, they are not here, all of them. So it looks like it will not be fair that we have a presenter. Oh, Dr. Matebula is there. Thanks very much. And then our presenter would be Papu Mamija. Okay. Then it means we're finishing now. Twarelo. Thanks very much. You can just give a round of applause.
Dumelang, Sanbonani, Abusheni, Ndumachere, good afternoon, ladies and gentlemen. My name is Sarlon Zanungwatu. I'm a Master of Science candidate in Agricultural Economics in Department of Agricultural Economics and Animal Production. So I'm so excited to take you through my research, my final year research project titled Investigating Factors that Influence Small-Scale Broiler Production Revenue in Greater Zanin Municipality, which was actually supervised by Prof. A. Bellet and V.M. Bengwa. I'm sorry for that. Actually, this study focuses more on the socio-economic factors that can affect the small-scale broiler production revenue, and those socio-economic factors may be access to credit, age, marital status, and many more. Small-scale broiler farming is considered as one of the prosperous and easy-to-initiate business. Therefore, the small-scale businesses, large-scale businesses, medium-scale businesses actually provide us with animal-based protein for the households. Hence, this production is the fastest-growing animal in agricultural subsector, especially in developing countries such as South Africa, Namibia, Zimbabwe, and many more. In South Africa, commercial producers actually account for 80% of the broiler production, whereas medium and small-scale broiler producers account for 20%. So it is very crucial to actually understand these factors in order for the policymakers to make effective policies and development programs that can help in transitioning small-scale farmers to commercial production. Broiler chicken in South Africa forms part of the staple food as they consumed in larger quantities by different members of the households. The small-scale broiler farmers have two main goals, which is actually to ensure that they reduce food insecurity and also to supplement household income. However, inadequate education, lack of credit access to extension services, and poor production resources are the main challenges which are actually hampering or impeding the growth and the development of small-scale broiler farmers. Therefore, assessing all the socio-economic factors that influence the small-scale broiler production revenue in Greater Zanin is the goal of this study. The aim of the study is to generate information regarding factors that influence small-scale broiler production, and the study intends to identify and describe socio-economic characteristics of small-scale broiler farmers in Greater Zanin Municipality, and also to analyze the factors that influence production revenue of small-scale broiler farmers in the study area. The hypothesis was actually derived from the objectives. The data was actually collected in the greater local municipality, and the researcher actually used the primary data, which was used to collect data using the structures questionnaire from 67 of the small-scale broiler farmers. That 67 participants was actually derived using Yusuf et al. 2016 formula. So actually, one may ask, how did I actually get to these participants? I actually used snowball sampling technique, which actually small-scale broiler farmers actually recruit me to the other small-scale broiler farmer. Two of the analytical techniques was used, which is the descriptive analysis, inferential analysis. And these two analytical techniques which was used to address the objective. So the first objective was addressed by the descriptive statistic, and the second objective was addressed by the multiple linear regression model. There we have the model specification, which shows Y as the dependent variable, which is the small-scale broiler production revenues, beta 0, which is the intercept, and we also have the explanatory variables, or I can say the independent variable, and the error term. I actually managed to add 3 of the descriptive results on this slide because of time. So, as I'm someone who's actually respecting time, so I'll actually just explain 3 of them. So, table 4.1 shows us age of the farmers, which shows that less than 20 years farmers accounts for 1.5%, whereas most of them are they are 31 to 40 years, which actually contributes to 40%. And table 4.2 shows gender of the farmer, which shows that we have more of the females, which are actually venturing or succeeding in these small-scale broiler farmers, that they actually accounts for 61.5%, whereas male accounts for 38.5%. And also shows that figure 4.1 shows the land size of the farmers, which actually shows that most of the small-scale broiler farmers have 6 to 10 hectares. The results here shows the table 4.3 model results, which shows 6 out of 11 independent variables which are statistically significant. And the dependent variable is small-scale broiler production revenue. And those 6 variables which are statistically significant is the age of the farmer, broiler farming experience, gender, access to credit, land size, and also employment status. We are coming to the discussions. As I've already discussed, table 4.1, which represents the results of the age of the farmer. Table 4.2 shows the gender of the farmer. Figure 4.1 shows the results of the land size of the farmer. So I'll just explain 3. I'll just explain table 4.3, which was actually shown in the previous slides. So I'll actually explain 3 of the variables, which are the access to credit. The second one is the broiler farming experience. The third one is the age of the farmer, which are all positively significant to the small-scale broiler farmer, to the small-scale broiler production revenue. Access to credit is regarded as one of the key elements in raising agricultural productivity, because if a farmer can be able to access credit, they may be able to afford some of the agricultural resources, such as purchasing quality of their day-old chicks and many more, and that can actually ensure that the farmer can be able to generate more of the revenue. And also age of the farmer. This shows that when it says the age of the farmer found to be positively significant to the small-scale broiler production revenue, it simply shows that when the farmer is getting more older, that's why they're eventually getting more interest in participating in the broiler farming, because of the dissatisfaction of the income, more of the responsibility on their shoulders. They also want to actually ensure that they provide adequate healthcare and education to their kids, and that can ensure that they can make them to be more interested in ensuring that they succeed in the small-scale broiler farming, and that can also ensure that the small-scale broiler production revenue also increases. So, Gyan, Sahin, and Horn in 2021 observed that age is one of the factors that can affect the probability of a farmer being successful in farming, as I've explained. In conclusion, I'll conclude that the proposed research hypothesis was actually rejected because, as the findings of the multiple linear regression model showed, that some of the factors were actually having an influence on the revenue of small-scale broiler production, such as age, access to credit, and others. So the findings of the descriptive statistic indicated that the women predominate among greater than municipality scale broiler production based on how they are categorized as female constitutes to 61.5 and male accounts for 38.5. So in my own perspective, I'll say the government will have to actually intervene in ensuring that they invest in the research and development where they get to fund the researchers who are actually addressing the matter of the gender inequality. Because in our country, or I can say in South Africa, the gender inequality is very persisting. So the recommendation, I'll say, farmers should receive significant regular training in managing broiler production, such as workshops where they get to understand or they learn more about the challenges, marketing strategies, and also the threats in the broiler production. And also, I'll say easy access to government extension services, which they can actually educate the small-scale farmers about the climate change or raise awareness about the climate change and what kind of the strategies they can actually adapt to avoid the higher mortality in their broiler production. Also, lastly, I'll say access to agricultural resources such as quality of day-old chicks, vaccinations, adequate housing, and many more. So I'll definitely take this chance to acknowledge my supervisors and also all the stakeholders that have participated to ensure that this study is successful. Here are my references. Thank you everyone.
Thanks very much. Can I have one question? There is a hand there.
You mentioned something about agricultural resources, so I want to know which resources should the government or the NGOs offer to enhance the production of broiler.
Okay, thank you for the question. I would say the agricultural resources which the small-scale farmers has to actually have or access to ensure that they enhance the productivity. I'll definitely say the vaccination. If the small-scale broiler farmers can have the vaccination, that will actually assist in terms of the veterinary medication to ensure that they control the diseases such as Newcastle. The other one, it was avian influenza. I'm not sure if I spelled it very well. And also another thing may be feed. So the small-scale the broilers will eventually need the feed in ensuring that they can grow properly. And also another one, I'll explain the adequate of the housing. So if the broilers can have adequate housing, it will ensure that they do not have to experience the predators or the animals that will eventually kill them, and that will actually assist in ensuring that there is a reduction in flock mortality. And lastly, I would say the small-scale broiler farmers will eventually need the finances. They will need the finances in ensuring that they can be able to purchase what I've already said or have access to what I've already said, such as ensuring that they have the vaccinations, ensuring that they can be able to purchase the feed, they can be able to ensure that they build the adequate housing. Yeah, that's hence I will say the agricultural resources that eventually.
Sorry for that. Thanks very much. We do not have time for other questions. We can just give a round of applause. Our next presenter, Malachi.
Good evening, everyone. My name is Maputi Malachi, a second-year master's student, and I'll be presenting a study extracted from my dissertation titled Germination and Seedling Morphology Responses of Terpre bean to Different Concentrations of Colchicine as well as Soaking Durations under the supervision of Dr. R.L. Mlatudi together with Dr. P. Mangen. Terpre bean is a diploid leguminous crop that has originated from the arid regions of southwestern United States and central Mexico. It is considered as a valuable crop due to its nutritional content. The grain of terpre bean is rich in protein, carbohydrates, vitamins, as well as rich in antioxidants. Colchicine, on the other hand, is an alkaloid originally derived from Colchicum autumnale bulbs, and its function is to disrupt the structure and the orientation of the spindle fibers, resulting in the process of polyploidization, which is the condition in which the organism has two or more than two chromosomes. In Southern Africa, in countries like Botswana, Zambia, Zimbabwe, as well as South Africa, this terpre bean is cultivated by smallholder farmers who usually use unimproved landrace varieties. These landraces are poor in quality and they affect germination as well as resulting in poor crop stands and yield. The widespread adoption of terpre bean is hindered by the absence of improved cultivars that can yield consistently high grain yields. On the other hand, the genetic improvement of terpre bean is also constrained by its limited genetic diversity, which can potentially be expanded through induced polyploidization methods, such as using chemical mutagens like colchicine. The process of polyploidization in plants is usually a slower process, hence there is an interest in using induced methods for faster results. The effect of this colchicine specifically on terpre bean germination as well as seedling morphology is not known. This terpre bean is important and serves as a rich source plant-based protein, and it contains calcium, iron, copper, and zinc. It is a drought-tolerant crop, and it has the ability to thrive in high temperatures, making it a suitable crop for water-scarce regions in the face of climate change. The induction of polyploidy in plants is also recommended because it can bring substantial changes in morphology, physiology, as well as the genetics. And over other mutagenic chemicals, colchicine is the most preferred due to its effectiveness in the morphologies. Therefore, the response of terpre bean germination and seedling morphology to colchicine is worthy of investigation. The aim of this study was to investigate polyploidy of terpre bean using different concentrations of colchicine for its growth and yield development, with the objective to determine the effect of different colchicine concentrations and seed soaking durations on terpre bean germination and seedling morphology, and it was hypothesized that they have no effect at all. The experiment was conducted at the University of Limpopo at the Department of Biodiversity and the Tissue Culture Growth Room, and the experiment was laid in a completely randomized design with 15 treatments, where the treatment factors were colchicine at 0, 0.1, 0.2, 0.4, and 0.8%, and soaking durations at 3, 6, and 12 hours. In order to conduct the study, a colchicine stock solution was prepared and the seeds were sterilized on the surface. They were then put in the beakers and placed on the orbital shaker for 3, 6, and 12 hours. After they were rinsed to remove the debris and then sown in hygromix as a growth medium and incubated for 14 days where data was collected. The data that was collected was then subjected to ANOVA using statistics 10 to compare treatment effects on terpre bean, and the means were separated using Tukey's test at 5% probability level. The obtained results, as shown in figure 1, showing the effect of colchicine soaking durations on germination percentage, it shows that there was a significant effect, and then where it shows the seeds soaked for 6 hours and 12 hours, achieving the highest germination, while the seeds that were soaked for 3 hours showed the lower significant at P is less than 0.05. Figure 2 shows the effect of colchicine soaking duration on mean germination time, where the seeds that were soaked for 3 hours germinated fastest at 3.3 days, while the longer soaking durations in between 6 and 12 hours showed the delayed germination. Figure 3 shows an effect of the interaction of colchicine concentration and soaking duration on primary root thickness, where the distilled water produced the thickest root, statistically grouping with the most low dose treatments. However, this high concentrations, particularly in between 0.4 and 0.8% colchicine, consistently reduced root thickness across soaking durations. Table 1 shows effect of colchicine concentration and soaking durations on seedling germination and seedling morphology, where the parameters were non-significant. Moving to the discussion, under germination percentage, which showed a significant effect, the results suggest that soaking times may be insufficient to fully hydrate temporary bean seeds and activate metabolic processes required for rapid germination percentage. And similar trends have been reported in legumes, where hydration has improved imbibition and uniform germination. And then with the mean germination time, the rapid emergence following shorter hydration may also be linked to partial soaking effects, which promote early metabolic activation without leaching inhibitory solutes. And the extended soaking might cause leaching of nutrients or oxygen limitation, slowing down the seedling emergence. And then with the interaction of soaking duration and colchicine concentration on primary root thickness, since I've mentioned that the colchicine disrupts the spindle fibers formation during mitosis, leading to altered cell division and growth, it is shown that the root meristems were sensitive, explaining the observed thinning at higher doses. And comparable inhibitory effects of colchicine on root morphology have been reported in beans and other legumes in studies conducted by Adami and Alui in 2004 and Badir in 2014. For non-significant parameters, colchicine effect since the colchicine effect are tissue-specific, they strongly influence root meristems, but less so above ground traits at early seedling stages. So the absence of significant differences could also reflect on physiological buffering, where colchicine concentration are insufficient to cause phenotypic changes beyond root development. In conclusion, soaking duration significantly improved germination performance with 6 to 12 hour treatments enhancing percentage, while shorter soaking reduced time, and the colchicine application at higher concentrations reduced primary root thickness, confirming its inhibitory effect on meristematic activity. The findings of this study suggest that moderate soaking duration at 6 hours with colchicine at 0.2% provide the best balance between high germination percentage and rapid emergence. I would like to thank the University of Limpopo and the Department of Biodiversity for providing all the resources that were needed to complete this project. And these are the list of references that were used in preparation of this PowerPoint presentation. Thank you.
Thanks very much. Now it's time for questions. Yes.
Dango for your presentation. My question is, do you think it's practical and economically feasible at a commercial level? And then does early germination time and emergence guarantees higher yields? Because we know that farmers are more interested in yield, so you might find out that your treatments result in improved germination time and emergence, but then when it comes to yield, it is poor.
Okay, thank you for your question. I have mentioned that this is a portion of my dissertation. So when it's full, it's also focusing on the growth and yield. So this is the partial part where it was significant at a germination level. So the second part of the experiment, that's where I'll be conducting on growth and yield. And that's when I'm going to see that it's practical and it can be applicable to farmers and practical in general.
You have a follow-up? Okay, no, thanks very much. Can we now have Lerato? Lerato. Can we have Moloko while Lerato is sorting some things?
All right, thank you. My name is Muloto Muloko, and I'm here to present part of my research titled Soil Health at Risk, the Insight from the Gully-Eroded Communal Land in Panama Village. Now, our soil at communal lands are at risk of gullies. So gully erosion is a part of water erosions that occurs when the concentrated water flow erodes the soil to the point that it creates the valleys which cannot be implemented or corrected by the tillages. Now, according to Lee Rocks at 2010, stated that more than 570,000 hectares of South African lands have been eroded by gullies, and specifically in Limpopo, more than 58,000 hectares of these lands have been eroded by gullies. Now, looking at Panama Village is the one of the villages which are affected by gully erosion. And then we can see that it has been affected to a point whereby the destructions cause the removals of soils and the damage in terms of the infrastructures, as you see on these pictures here. And then also these gullies have been incising throughout the village, which now threatens the livelihood and also the soil fertility. Now, why should we study the susceptibility of soils to erosion? Now, the thing is that we need to know which soils then are vulnerable to erosion. Now, understanding which soils are vulnerable to erosion will now help us to implement the erosional measures in order to improve the soil fertility and in order to implement the proper land uses. Now, the aim of this study is to investigate the susceptibility of soil to crustability and erodibility, and the objective was to determine the crustability, stability, and the erodibility of the soils along the gully. Now, the study area, this study was conducted at Panama Village, which in Feta Homutu was a municipality as shown on this map. Now, the field survey was conducted to assess the variations of soil forms along the gully banks, and the soil forms was classified using the natural and anthropogenic system for South Africa, 2018's version. Now, the stratified sampling strategy was then conducted, guided by the variations of soil forms along with the elevation change. And at each profile, a triplicate of soil samples was then collected from each horizons, the topsoils and then subsoil. Now, in terms of the analysis, the sift soil was then used to determine the soil organic matter using the ignition method, the soil texture, soil pH, and the soil EC. And also, the wet sieving method was then used and then used to infer the soil crustability. And the soil erodibility was then inferred from the soil texture, organic matter, and also the structure. Now, these are the soil forms which are dominating within the gully banks, which namely is the wheat bank, Dundee, and Kwaggafontein. And then these soils are characterized by lumicent with more than 80% of sand content. And then all of these soils have slightly acidic in terms of the pH. Now, how did the gully erosion influence the organic matter throughout the gully banks? Now, as you can look at these annotations here, you can see that mostly these soils have less than 1% of organic matter. Now, this can be explained by their coarse-textured soils, which now studies have shown that soils with less than 1% of organic matter are usually related to the coarse-textured soils or the sandy soils. And then also, the gully erosion now tends to increase the sandy soil or the sand content in the soils by removing the fine particle soils, which now tends to increase the sand content, which also reduces the organic matter. And then also the other thing is that through the concentrated water flow by these gullies, they tend to erode the plant litters and other organic compounds, which now reduces the organic matter into the soils. But if you can look at Kwaggafontein on the topsoil, it has more than 1.5% of organic matter. Now, this can be explained by the vegetation which was found on top of this surface, which now vegetation tends to increase the organic matter, and specifically on the topsoil. Now, looking at how gully erosion has influenced the total organic carbon. Now, total cations, Total cations, we're talking about the amount of the nutrients, which they are exchangeable base cations, which can be held by the soils to sustain the plants. Now, we can see also that all of them, they have less than 10 centimole per liter. Now, usually, literature says that soils with less than 10 centimole per liter of these total cations are usually characterized as weakly fertile, which now it means that they don't have much of the nutrients to sustain the crops. Now, this can be influenced by their high in terms of the sand content, which means that they have low nutrient holding capacity, and also during the concentrated water flow, this base cations tends to be leached away, which now tends to have these values that we're seeing, which have less than 10% of organic cation exchange capacity. Now, the table that is represented here shows the stability and the crustability and also the erodibility of the soil. Now, the K factor that you are seeing there is the one that is going to tell us about the erodibility of the soils. Now, soils with less K factor shows that they are highly susceptible to erosion compared to the ones that have higher K factors. Now, if we can look at the Dundee, it has the coarse structured with a medium stability, which is the one that is likely in danger in terms of the erodibility. Now, this can be explained by its coarse textured and low organic matter, which now tends to increase the crusting and also lead it to be at risk in terms of the erosion. Now, both Wheatbank and Kwaggafontein have slightly higher K factor, which now tells us that they are at risk, but then they have high resistance compared to the Dundee one. Now, here we can see that the low organic matter in cooperation with the coarse structured now tends to increase the erodibility of the soil. Now, in terms of the conclusion, we can see that the Dundee soil form has the high sand content, which is considered or can be related to its low organic matter along with low total cations, which now tends to have a low structural stability, which now makes it to be at high risk in terms of the erosion. And then Kwaggafontein even though it has slightly higher organic matter compared to the other things, but the decline on profile due to organic matter put it at high risk and also in terms of the erosion. Now, the erosional measures should be implemented in order to reduce the erosions in terms of applying the revegetation, in terms of reducing the concentrated water flow, and also to increase the stability of the soils. Now, not considering the soil properties will now lead to the influence that we're seeing here, that these are the pictures which were recorded at Mpanama village around the gully. This is because we're not considering the properties of the soils when we're trying to implement the erosional measures. Now, trying to understand which now soils are susceptible to erosion will now help us in order to implement the correct erosional measures, which now will reduce the concentrated water flow and improve the soil fertility. In terms of acknowledgement, I want to acknowledge my supervisor and University of Limpopo. Thank you.
Thanks very much. Can I have hands for questions? I need questions. Can I? Yeah, I see the two.
Thank you very much, man. That was a lovely presentation. It was well combined and it showed proper investment on the work. But based on what I saw there, for the soil erodibility, you only considered the soil profiles. And I wanted to know that why did you exclude other factors that are contributing to the soil erodibility, such as the slope as well as the rainfall that is affecting the area? Thank you.
Okay, thank you. I did not exclude other factors, but then for the purpose of this one, I just focused on the ones that were influencing the K factor, which is the erodibility. On the broader part of my study, I've incorporated the GIS in also to look at the effect of slope and other topographical influence, because they're the ones that drives this erosion. But then for the matter of this one, I just looked at the ones that influence the K factor, which is the physical properties and the biological.
Okay, thank you for a nice presentation. I'm familiar with the place, and upon visiting, I was concerned because it is reducing the availability of grazing lands. So correct me if I'm wrong to say, are this the erodibility and related to, or maybe challenged by the inherent characteristics of the soils?
Okay, yes. The soils, looking at them, they are very susceptible to erosion, which now they are people, because usually when they just implement the land uses, they do not consider the soils. And then usually this, looking at these results, they are the ones that are mostly the driving factor in terms of the erodibility, but they encompass with the other land uses and other things.
Okay, furthermore, since that some of the measures like the gabions that were established have also been destroyed due to erodibility of the soils, are there any other control measures that can be implemented in such soils to reduce the erodibility?
Yes. I like the example for these gabions. These gabions, when they were, according to my suggestions, when they were implemented, they did not incorporate the soil properties. But then, yes, the gabions are the correct measure in order to decrease the water concentration, the water concentration flow. But then incorporations with the revegetations, planting trees can also, in terms of stabilize the soils, and incorporations with those gabions, they can reduce the fertility, the gully. Thank you.
No, thanks very much. Lerato. Lerato will be our last presenter.
afternoon, everyone. I come by the name of Miss LV Malavati, and my students call me Miss Love. I will be ending the FSA 2025 Research Day, and allow me to take you through the work of Professor J.J. Tongwani, Professor V.M. Bengwa, and Dr. M. Ngwan. But before we bunch into the findings of this wonderful PhD, I would like to begin and ask you a question that, how many different products made out of maize have you consumed ever since the FSA have catered for us? Is it one, perhaps two or more? So there are many products that are sometimes even recessive, and this study will take you through those kind of products that we are underestimating. So the topic of the study is a systematic review and structural design of maize value chain in South Africa, where we have looked into the market transformation and the implication into the food security and economic development. With that, just take your seat and relax. We are going to take you through the presentation outline. So in South Africa, maize is the main culinary engine, which have been recorded to have 14.5 million tons, which have been reported in 2023 by the SAGES. And furthermore, we have consumed 11.7 million domestically. So among these two ratios, we are having a difference of 10 to 30%, which is being accounted to be a post-harvest loss. So that must be as our concern as researchers, that what can we do to minimize it? So there are critical challenges that the study have identified, where we have the lack of comprehensive product flow map, the poor coordination between white and yellow maize streams, and the supply-demand mismatches, along with smallholder exclusion from the value chain. And the study is motivated by the dual consumption pathway, because it has been recorded that white maize, it is for human consumption, and yellow maize, it is for the animal feed, but we are having an increased interchangeable based on the market condition, because today we can see that we are consuming a lot of products from maize meal. The study have used a systematic review following the latest PRISMA 2020 guideline to assure the transparency, where it have extracted databases from academics, government, and also the grade literatures. And to make it realistic, we have took from 2020 to 2025. So with that, the study started with 103 database sources, where from it have extracted 28 high-quality studies. And this have been done through the thematic analysis, where it have been organized using the famous software, which is the Microsoft Excel, to organize the data according to the themes such as the product system, the post-harvest processes, the value addition, the stakeholder dynamics, and the climate smart dynamics, and also the integration, as you can see from the diagram. Furthermore, allow me to take you through the result and discussion, where we are going to look into the 8 distinct maize value chains, which have been identified through the systematic analysis using the visual comprehensive framework to develop one maize value chain, which is going to summarize it. So the study have found that production integration have acknowledged commercial farmers to dominate both white and yellow maize sector, with climate smart practice, which have been increasingly adopted. And secondly, in the secondary sector, it have acknowledged major millers such as the Prema Foods, the Tigers, the Pioneers, which are controlling the transformation process. And lastly, in the tertiary sector, we are having market segments where we are having distinct pathways for human consumption and for animal feeds, with an increasing cross-section. Is it acknowledged in the current maize value chain? So the study will answer that. So the crucial finding is that market concentration in milling and feeding sector, it restricts smallholder participation and equitable value distribution, and that will be spared for our discussion later on. So the study have evaluated 8, as I'm saying, 8 maize value chains, which have already been developed. And from the first one, figure number 2, we are seeing a global maize value chain, which speaks about the transportation and also the import and export of it, and going to the second and third. But South Africa have adopted the Lewis design, which have been numerously being encountered or included in the Department of Agriculture reports, and that marked its milestone. And currently, we are developing the climate smart policy of 2024, which need answers. So with that, we are going to, we further looked into other four maize value chains to not make them biased, and to design the following maize value chain. So the maize value chain, it have been designed by me, the researcher and the supervisory team, where we have acknowledged that yellow maize, it is also used for human consumption. Where do you think the cornflakes that we eat in the morning, they come from? Yellow maize. But most of the literatures, they're saying yellow is used for animal feeds. So we have identified that we are having a cross-utilization where maize is also being broken into three sections, which is the maize brine, the maize germ, and the maize starch. And those products, they are making the very distinct maize product that we see. And during lunch, we can say we ate pap, which is from the white maize, because it was yellow. Because according to South Africa, it is wrong and it is a violation of human that we mix yellow and white maize. But for animal, we can mix them to avoid what? Wastage. With that, we are drawing to conclusion. We are having conclusion saying, the dual nature, it needs to be emphasized because white and yellow maize, yes, it serves as a distinct but yet complementary roles, with commercial farming dominating both sectors. But we need to also encourage the smallholder exclusion being highlighted. There's a need for inclusion of them in the market. And the potential is that there is a significant value chain potential for existing the process, and particularly the starch and the germ and the brine products, which is what we have been eating. Then lastly, on the hierarchy, we are seeing optimization. This is a cry to us that optimization and maximizing the byproduct utilization and reducing post-harvest losses is very crucial for our economy to be sustainable. For policy recommendation, it is a cry from us, the researchers, that inclusion participation of our smallholders into their policies, it needs to be there through the support of programs and the grants. Secondly, the processing investment, where in the processing investment here we speak about rural infrastructures. It is by time that we see tiger brands in our rural areas because a lot of farms are there. Then thirdly, we are living in climate era, so it will be a sin to leave this out. So climate resilient, it needs to be emphasized. And lastly, infrastructure development, which is the roads, they need to be emphasized for our farmers to reach the market. This has been aligned with the Agricultural Agro-Processing Master Plan and also the National Development Plan of 2020 and the United Nations Sustainable Development Goal, which have been even mentioned by Ms. One of the speakers who was here. And for me, the PhD candidate and proud alumni of University of Limpopo, I would say, strengthening South Africa maize value chain is not just about food, but it's about future securing, reducing poverty, and driving inclusive growth. And research provides a foundation for that. And a collective action, it builds the future. With that, these are the references that have been used for the study. And I would like to thank you very much for attending FSA and supporting University of Limpopo. Thank you very much.
Thanks very much. Can I have hands? Okay. Moluko, where is your mic? Oh, okay.
Thank you for the amazing PhD study. I'm interested in your findings. I also want to know what influenced the time period from 2020 until 2025.
Okay, thank you very much. We are emphasizing in research module, especially the fourth year model that I'm lecturing, which is the builder for all of us. It is very important for us to look into the recent literature for us to have relevant outcomes, because we are communicating to recent policies. We are communicating directly to the policies. Therefore, inclusion, the study, if you can look at it from a broader sense, it is a study which is a panel data analysis, a combination of time series and cross-sectional, where it starts from 1990 to 2023. But for this systematic review, for its relevancy, I needed to use the recent data set, which is even there in the reports, because they're already even reporting about all things, but as a reference. So it is relevant to report about things that are within 5 years and following the university policy.
Any other hand? We still have some time. Oh, there's a hand somewhere. Where? Oh, Mativa. Yes, you can ask.
Yes, thank you. I think there's been a lot of research on maize. And then if you look at the current trend in terms of agriculture as a whole, there's this thing of including traditional underutilized crops. So in your view, do you think it's wise for us to continue researching on maize? Looking at, consider climate change, by the way. We have your crops like sorghum and millet, which, if you look at the agricultural landscape, your cereal crops like maize, rice, and wheat are the ones that are gatekeeping other cereal crops from entering the market. So do you think it's wise for us to continue working on maize? Or we can, yeah, I think you get my question now. Thank you.
I wish they can project my precious maize value chain. But it is very, very, very important, because as I've studied the study, I said maize is a culinary engine. Being an engine, it means that on our daily basis, we are eating maize, starting from the pap, going to the oil, going to the cornflakes, going to your snacks and everything. So it is very important, because we have also identified there's been a loss of 10 to 20. And in the market, when you go to the other objectives of the study, which is the market trends, we are identifying that there is a shortage, so there is a cry. If maize is a staple food of South Africa, I would vote for maize to be researched over and over again, but in a different perspective. Remember, when we look into the criteria of choosing a topic, the topic needs to be unique. And the unique part is that for this study, we are now looking into the cross-utilization. So it is important very much. Thank you.
Okay, thanks very much. That was our last presenter. We can just give a round of applause, the last time. And then I'm told that Prof. Chikoro will come and give us, is it the marching orders or direction, probably? So.
Thank you very much. It's a very good evening, everybody. It's been an excellent 3 days that we have been here, and my task, I'm here on behalf of the chairperson of the research committee from the School of Agriculture and Environmental Sciences, Professor Dlamini, who you have seen. He has requested me to speak on his behalf, so I'm speaking on behalf of the chairperson. I would like to, first of all, thank the students for excellent presentations. I've been here the whole 3 days, and I have been amazed at the range and depth of the contributions that you have made. So I think maybe we should give a round of applause to us as students. I'm extremely proud of you, and I can also speak on behalf of the seniors who are not here. They have been saying the same thing as well. Well done, well done. Across from your fourth year, honors, master's, PhD, you have all done very well. Thank you very much. We are very much happy about that. I would also like to express the appreciation to, of course, you are being supported by supervisors, by advisors, by mentors, and so we cannot forget them. We need to appreciate them. We need to appreciate our supervisors, and some who are not formal supervisors, but who are assisting us in one way or the other. So we would like also to give a round of applause to our supervisors. And of course, to the university, to the Faculty Higher Degrees Committee that have put this all together. I think you can imagine the enormous responsibilities and tasks that are involved to try and even just to put the program together, for example, to collect the abstracts, to do all the logistical arrangements. So I think we need to also acknowledge the university, the faculty, and all those involved in the organization of this big, big event. Of course, maybe from me, I would like to encourage you, whatever it is that you have presented here. I've seen that most of the work that you have presented, it is at various stages of development. Some are still beginning, some are somewhere in midway, others are almost submitting for examination. I would really encourage you to consider publishing this work in the refereed literature, in reputable journals. Of course, as you might know by now that there are some journals which are not necessarily the best to go to. Please consider taking this work, this excellent work that you have been presenting here, to a wider audience, and a wider audience is through international journals, conferences other than this one. We need you to also go to other conferences where you will meet students from other institutions and professors from other institutions, and so on. So that I'm also encouraging you to consider that. Lastly, I've also been requested to announce that tomorrow is the last day of the FSA Research Day, and the program starts at 5 o'clock. The buses, I think, I think the communication will come again through emails, how the buses will take students from the usual stations. So please do come on time, and also remember to carry the name tags so that you can be admitted into the hall. I think the program will be here starting from 5 o'clock. If you have nice clothes, please wear them. Yeah, tomorrow we'll be we'll be having a gala dinner, and there will also be another program for that. Today, I think this is the end of today, also the end of the whole program. I wish to thank, oh, I forgot, Prof. I forgot to thank the chairs. One more, let us also thank all the session chairs who have been here, making sure that our program runs smoothly without any hitches. I had forgotten. Program chairs and the adjudicators. There have been adjudicators who have been listening. You are forced to listen when you are an adjudicator. You cannot even look at your phone because you must listen to the whole presentation, and you must judge them on different. Of course, tomorrow, please don't forget, there will be awards for students who have done but you have all done well. I've said it already. So if you don't win an award, please do not think that you have not won. You have all won. In my opinion, you have all won. But there will be some who will who will get some formal recognition tomorrow. And so that's why I'm reminded to also acknowledge the role of those who have been adjudicating. So I had forgotten critical role players, the adjudicators and the session chairs. Let us please do one more round of applause. The director is here. I didn't notice he's here. I think we must also have a special thanks to the director of the school who's sitting here. I'm looking here and I'm saying, that looks like the director. Please, let us acknowledge the director. Like I said, like I said, if we don't get support, which also includes a lot of financial resources, then we won't manage to have events such as this. And I'm sure the director is here, he has seen the work that you are doing. If we go to him tomorrow, Prof, we want to go for a retreat, we want to go for a workshop, I think the Prof will realize the value of what we are doing. Otherwise, I think it is time for me to go back and also time for us to call it a day. Thank you very much, everybody.